primary immunodeficiency disorders, then the risk of infection is low as compared to the autoimmunity, which is then becomes high. And again, the common genes that are involved are shown over here, and the risk of autoimmunity in these patients are also shown in this diagram. So this is something that's a good diagram, I find. So this showing an immunological mechanism associated with autoimmunity, which is why there is an autoimmunity in PI patients. So I tried to summarize this in these boxes. When autoreactive T cells and B cells fail self-tolerance checkpoints in thymus and bone marrow, it starts from there. So, and when they spill into the periphery, then we have a mechanism, peripheral mechanism, again to or checkpoints to control them. But if they are not intact, of course, and all this control is lost, maybe because of some extrinsic factors like viruses, toxins, etc. Then these clones, which were dormant in the peripheral in our periphery, they become then active clones, and then they causes autoimmunity. So if peripheral checkpoints is impaired, autoreactive B cells become activated and convert into plasma cells to generate autoantibodies. T cells and T cells regulation and immunophenotypic characteristics of the CVID. CVID is a very prototype of common primary immunodeficiency in which we see a lot of autoimmunity. So this is just showing how autoimmunity occurs in CVID. So there can be a B cell dysregulation in these patients, and as we can see in the lab, elevated CD21, low B cells and IgM, and also increased autoreactive B cells in the periphery, and there is decreased memory B cells and B cell receptor diversity. The CVID can be because of the disturbance of T cell homeostasis, maybe decreased T regulatory cells or skewed Th1 immunity, or there may be downregulation of the DR cells as occurs in LRB and CTLA4 deficiency, IgA deficiency. There may be upregulation of mTOR activity in PIK3CD, and these patients then they respond to mTOR triggers. The KP GF leads to expansion of P17. BAF augments H1 cytokine production by effects on the BAF receptor on T cells and promotes the survival of autoreactive antibodies via receptors on the B cells. So the management is very important. Right from the beginning, how to take it. So there is a short-term strategy to control the hyperinflammatory state. You do it with the steroids, immunosuppressant agents, or the chemotherapy, and then the long-term strategy where you have to decide if it is IG replacement or the allogeneic hematopoietic cell transplantation is needed, and there are other modalities according to the diseases that we have found. So this is just my concluding slide that in primary immunodeficiency there is diverse clinical presentation that can be severe allergies, infections, autoimmune inflammation, and malignant proliferation. And the diagnosis of the immune defect is very important, and all it depends upon the very good immunological tools. We need to have these immunological tools developed here to have the better diagnosis. So that we can have a mechanism-based immune strategies, because from diagnosis we can present in minimizing the long-term effects of recurrent infections, autoimmunity, and inflammation. So I say thank you with that, and then you have any questions.
Session is open for questions. Dear Dr. Raja.
I was wondering if in different conditions, the antigens, the self-antigens that are recognized are different.
Mm-hmm.
And if there is any relationship between those antigens and shared antigens with pathogens.
Yes, that can happen. This is the trigger mostly.
Are the epitopes known or antigens known?
Not exactly. I haven't looked at it in depth. Maybe it will get started comment section.
Okay, thank you. Thank you very much. That has always been the question. If we only knew about the specific epitopes, and then there is epitope spreading as well. Thank you very much for the very nice presentation. And I was wondering, the routine sort of work remains essential.
Yes.
In the first case, I may have missed a number of findings there, but immunoglobulin levels, immunization responses, and those things are available, say within few days, and this workup for me to follow once you have a almost confirmed diagnosis of immune deficiency, and then you send it across. And they take their time.
Yes, it took a long time to give us the, like, and we had, actually, I actually put only the significant findings out there. We've done, we have available response to vaccination, and response to vaccination now in the
Those are
hospital.
essential.
Yeah, I understand. So initially we did not have that, we had a lot of problems.
Missed in this slide.
Maybe, I should have, if it's,
And
yeah.
subsets were not there.
No, I should.
These are finding. Anyway, also
Yeah.
those things are important, and they are available at a number of centers within Pakistan.
Yeah, we had done lymphocyte analysis and GNT, and it's written in
I may
the
have missed
slide.
that.
Yeah.
The second patient, if you could emphasize that again, a routine workup which is available. So the complement C4 was lower,
In the first
was
place.
positive, and it's important to have the quantity. So, and the initial finding of cytopenias and all of these things were pointing to lupus initially. So that's the routine or the common things remain common, and then obviously.
Yes, I mean, the case was actually referred to us for the immunodeficiency workup, but when we did autoimmunity workup, it came out positive. But because of this presentation, our underlying immunodeficiency remained there, causing all this.
Thank you very much. And
माइक ऑन करें
thanks, Sabiha,
माइक ऑन करें सर
wonderful presentation. So, let me introduce the second speaker of this session. And he has already introduced the moderator, but I would just mention about his brief achievement. Professor Tahir Aziz obtained his FRCP from the Royal College at the College London. Currently, he is working as a professor of immunology at Shiva College of Medicine, Islamabad. He will be talking about autoantibodies as a diagnostic markers in autoimmune encephalitis. So, Dr. Saab, we are looking forward for your presentation.
Thank you very much for that introduction. And we are all indeed honored that you are here with us today. With your permission, sir, I would like to thank my teacher and my mentors and my guides here, Professor Rabi and Professor Naki Zafar are here. So I would like to thank them and like to thank all of my colleagues who are sitting here and are being part of this very important activity. So, and thanks are due to Professor Sabia, Dr. Vidya, and all the team for putting together this nice program. So thank you very much for providing me this opportunity to talk about autoantibodies, the diagnosis of autoimmune encephalitis, a relatively recent clinical service in Pakistan. And what I would like to demonstrate that what these markers do is provide a tool which can be easily used in most of the centers within Pakistan, and they can make a critical difference in the management of patients. So the next slide is just right here. So moving class of guidance, that's the snapshot. And if you look into the eyes, that is the journey of 1,000 years moving forward, and we come to this special appearance here. On the left, there is this medusa, right? Slide, you can see on our left in red, the antigens are on the surface of cell membrane, and on the right, the antigens are inside the cell. So antibodies are able to interact with the target antigens, and if we use the rat hippocampus as the substrate in the indirect immunophores, we can demonstrate that the antibodies were binding with And if we use the neurons in culture, living neurons, we could demonstrate using immunofluorescence that the antibodies would combine with the targeted on the cell surface. But in case of antigen, which is in this case anti-blue antibodies, the target antigen was inside the cell. The NMDR antibody encephalitis affects with the female preponderance. And here in the center, I have these small bars where we can see that 1 to 1.47 for male to female. The total positivity ratio out of 859 was only 4.8%, and the mean age at presentation. Generally, these patients would be in their third decade, and however, there is a great age range that is 3 to 81 years. The patients will be affected. Anti-NMDR and positive serum specimen, and in the CSF specimen, and in the CSF specimen, please note only NMDR antibodies were positive. Rest of the antibodies like VGKC and GABA were not there, which were detectable in the serum specimen. So CSF is a preferred specimen if you are suspecting anti-NMDR encephalitis, and you just have to use indirect immunofluorescence. And here we can see that positivity rate has gone up a bit, 6.4% using CSF, and male to female again, one is 2.2. A mean age at presentation, again emphasizing in the third decade of life with a wide possibility, wide age range possibility. This important bit of information just emphasizing that the 60% of the autoimmune encephalitis is likely to be anti-NMDR encephalitis. And for this, we had the initial description by Professor Dalmu, and he didn't have these luxury of having autoimmune markers, and he was just following the clinical presentations, and he documented the clinical. Professor Dalmu, and he works in New York, and initial publications were in 2010, 11, and onwards. And what I came across is that story of one of the patients. She was a journalist, and Susanna Cahalan, I thought it should be Callahan, but there it is. She wrote a novel as she suffered from NMDR encephalitis, a month of madness, and she didn't remember anything. And this book was based on interviews of her caregivers and doctors and other attendants, her family members, and she titled it "Brain on Fire." So it is such a severe disease that you lose all your higher mental function. There is a prodrome of fever, headache, vomiting, nausea, diarrhea, and upper, just quite common prodrome, but that is followed by psychiatric symptoms, memory deficit, atypical seizures, all the higher functions going away. Hypersexual behavior, irritability, can be extremely violent. And this is a shot from the movie. This book was converted into movie, so this Brain on Fire, a very interesting bit of information there. And again, 2 snapshots, and she suffered from it. Fortunately, she was cared for by Professor Dalmu and his team, and with persistent immunosuppression, she came out of it, and now following her pursuits in career. So that was about anti-NMDA, and the second one was at 35% was anti-VGKC antibodies, and these anti-VGKC antibodies, out of the total test, again in 859 serum specimens, the positivity rate is 2.9%, just emphasizing that these are relatively rare conditions, even in patients who are suspected to be suffering from autoimmune encephalitis. So even at these rates, lower rates, but if you come across this finding, this is a specific finding. You won't find non-specific false positive positivity. So if you find something positive, you would make a life and death difference in the management of these patients. So the VGKC antibody
producing patients, and they would suffer from a severe disease. And these patients, more of them are likely to be male. We came across something like total patients which were positive out of 859, 25 patients. And here we have more of male patients. Again, the age range here is in 2 peaks. VGKC is a complex of 2 antigens, Casper2 and LGI1. For Casper2, the mean age at presentation was 33 years. There is again a big age range, but for LGI1 it is 52 years. So that's something, small bits of information which should be kept in mind, especially by the neurologists who are taking care of these patients. So what do these patients present with? That was memory disorders, prodrome, and everything for anti-NMDA positive patients. What about anti-VGKC positive patients? LGI1 positive patients characterized by this. These are few histories, and that just emphasizes we need to have our own data as well. So anger, sleep disorder, cognitive decline, impulsive behavior, disorientation, and seizures, usually atypical seizures. So perhaps pointing to those special conditions where sometimes, I don't know, these labels are put that there is taking over of the body by extraterrestrial beings or something like occupied by jinns or something. So Casper2, again, sleep disorder, cognitive decline, history of falls, hallucinations, seizures. Those are always there, and these are not typical seizures. They are atypical seizures, just plucking something from the air all the time. So these seizures, atypical ones, are there in patients from NMDA, suffering from NMDA, LGI1 antibodies, and Casper2 antibodies. Two were males and 2 were females. Now, this is another group of disorders which is caused by autoantibodies, anti-aquaporin 4 antibodies. Again, these antigens are present in the cell membrane, and this is a disease which is affecting the optic nerve, so producing the defects in vision, and that's a quite prominent presentation with lesions moving on to the spinal cord and causing bladder problems as well. And this disease was initially described as neuromyelite optic, affecting the optic nerve and the spinal cord, and it was initially described by Eugene Devick in 1894, also known by Devick's disease, and prominent feature as he described was urinary retention, complete paraplegia, and blindness. And this was a progress in few weeks after the initiation of the illness. So Devick's disease can be, if it is not treated, 50% of patients with NMO, neuromyelitis optica, will be wheelchair dependent and functionally blind, and third will be dead within 5 years after the first attack. So if you can provide this information, this critical bit of information to establish a diagnosis, a specific immunotherapy, perhaps going up to plasmapheresis, will help these patients, and that would be a critical help. And we have these assays available from commercial sources. These cells are transfected with the genes producing the antigens, and again using indirect immunofluorescence. This technique is available in most of our centers, and we can provide this essential clinical service to the neurologists. So these patients would present with bladder symptoms and vision-related problems, and just looking at this data, the anti-aquaporin 4 positive individuals, we have tested something like 1,888, something 900 total tests in 4.5 years, and the positivity rate is 16.6%. Look at the substantial possible number of patients who were helped for their specific treatment. And here you also notice that 1:5 female preponderance. So total positive patients were 314, and males were 52 and females 262, mean age at presentation 33 years. And it is important to keep in mind that these patients would have these antibodies In addition, they would also likely to have other autoantibodies. So something in the spectrum of autoimmune disease, starting specifically from NMO-related manifestations for anti-aquaporin 4 antibodies, and here we have the nerve cell, and these aquaporin 4 proteins are water channels, mainly present in the body, concentrated in the body of neuron, and they may form multimers, and autoantibodies are formed against these multimers and damaging the cell membrane. While the myelin antibodies directed against myelin are anti-MOG antibodies, and they would damage the insulating cover of the dendrites. So neurons, they would lose their insulation, and the progression of the electrical signals would be disturbed, and they would ultimately affect the function of the nervous system. So this is, sorry, again, the same slide. So MOG1 would present with prominent visual field effects, more of those and less of spinal cord related lesions, and they would be seen in younger patients. My apologies for the repetition of this slide. So for anti-MOG antibodies, this is prominent in males, anti-aquaporin 4 prominent in females, and the MOG antibodies, when they are found, they are not, these two antibodies cannot be found at the same time. What we do is we perform tests for anti-aquaporin 4. If that is negative, then we perform this test, and it is positive in most of the patients who are negative for aquaporin 4 antibodies. So this is the same fact that anti-aquaporin 4 antibodies are likely to have other autoantibodies, while these other autoantibodies are rare when we have anti-MOG related disease. So MOG antibodies, again, using the very simple technique of indirect immunofluorescence, you just have to have the right kind of antigen substrate, that is MOG infected transfected cells. And MOG, this is the MOG related data, total test 632, positivity rate at 13.2%, more of males, aquaporin 4 more in females. So autoimmunity spectrum of disease, while in case of MOG, it is male specified 2.5:1, relatively younger patients, 23 years at presentation.
So, can you just wind up in next 2 minutes?
Okay, thank you very much. So there is another group of antibodies, which is the paraneoplastic related with tumors, and paraneoplastic syndromes are rare. These antibodies can appear earlier than the detection of cancers. When you find these antibodies and syndromes, you have to aggressively look for underlying malignancy. You can have cancer and antibody without neurological manifestations. So this has to be related with clinical findings, and most of the tumors which are related with these production of autoimmunity are these: neuroblastoma, small cell lung cancer, and thymoma. Neurological symptoms, rapid onset, precede the detection of paraneoplastic tumors. And for the detection of these antibodies, what we use is immunoblots, again available, an easy technique which is available in most of the centers. For our own data, 4.5 years, total positive were 102. Most of these antibodies were anti-titin antibodies which produce a myasthenia-like picture. And we can use rodent tissues and have these, we can use rat hippocampus and cerebellum, and can have these characteristic pictures. Looking at these pictures for different antibodies, anti-HU antibodies, anti-MA1 and anti-Ta antibodies, looking at these excellent pictures from in other published studies, I came across this idea that we can have our own sections prepared from rat brain, and that is the study that we performed, and our own frozen sections and characteristic appearance of anti-NMDAR antibodies. So other investigations are important, clinical findings are important, history is important, and taking all the information together and having this critical information, we can make a difference in a large number of patients. And it is critically important to recognize the treatable and potentially reversible autoimmune encephalitis. That's why I emphasize that there is a significant percentage fraction of patients who can be helped. So we may use these techniques. There may be some hesitation, but if we use this technique, we can help a number of patients. So thank you very much.
Thank you very much, Dr. Saab, for a wonderful presentation. Unfortunately, another speaker is just waiting online. So there are a few from the audience, if you have any questions, so Dr. Saab is available and you can ask during the tea break or lunch. So let me introduce Dr. Michael J. Masad. He is an immunologist with special interest in inborn errors of immunity. He is the director of the Research Center of Excellence in Immunity and Infection and scientific advisor for the National Newborn Screening Program for SCID in Lebanon, where he established a laboratory to study immune disorders with a focus on inborn errors of immunity. He is also a faculty member of the Department of Experimental Pathology, Immunology and Microbiology, Department of Pediatrics, Center for Infectious Disease Research, and Faculty of Medicines at the American University of Beirut, Lebanon. So, Dr. Masad, we are looking forward for your lecture. Now, floor is over to you.
Good morning. Thank you, Mr. Chairman, for your kind introduction. I'm trying to share my screen, but I'm getting a message that the host disabled my screen sharing. If only the host can allow me to share my PowerPoint presentation, please. Can you hear me? Hello?
Excuse me. Can you make him co-host?
Maybe while they allow me to share my screen, first want to start by saying that this is a pleasure to be with you today, even if it's virtual. And I really like to thank the organizers, especially Dr. Anis, for her invitation and for hosting me to talk about our approaches to diagnose and treat patients with inborn errors of immunity, which are otherwise known as primary immune deficiency diseases as well. Can you please, can you hear me? Can you hear me in the room?
Yes, we can hear you, and now you are a co-host of this session, so you can now share your slide.
Okay, I'm going to share my slides now. So, as I said, I'm specialized in inborn errors of immunity, otherwise known as primary immune deficiency diseases. And you all know that these are genetic diseases, and there are many approaches that people have used to study human genetics. One approach would be to take a large number of apparently healthy individuals and sequence their genomic DNA using next-gen sequencing approaches and identify what is later classified as polymorphisms in the healthy population. Another approach would be to take patients from a relatively common disease, such as cancer, sequence their genomes, and then identify polymorphisms that have a higher incidence in the patient population than the healthy individuals, and then classify them as driver mutations associated with cancer development. And these kind of studies are known as genome-wide association studies, or GWAS. Another approach would be to look for these very, very rare patients that have orphan diseases, in our case, inborn errors of immunity or primary immune deficiency, sequence their genome to find hopefully variants of unknown significance, and then use a battery of functional tests to determine the pathogenicity of the variant and its association with disease. So before I talk about inborn errors of immunity, I'd like to take a few minutes to describe the normal immune response in healthy individuals. The immune system is composed of a number of organs and cells that communicate tightly together to mount an efficient immune response to any antigen. And of course, the most physiologic antigen for the immune system are pathogens. However, any substance that can elicit an immune response is an antigen, whether it's a pathogen, an allergen, self-protein, or even some metals. Immune cells develop in the bone marrow from common precursors, where they all develop, with the exception of T cells, that develop in the thymus. And both thymus and bone marrow are known as the primary lymphoid organs. After they develop in the primary lymphoid organs, they exit and circulate the body in the bloodstream, but also in the lymphatic system, which is composed of this elaborate network of lymphatic vessels on which are interdisposed these small nodules called lymph nodes. And both lymph nodes and spleen are known as the secondary lymphoid organ, and this is the site where the adaptive immune response develops. One characteristic of the immune system that is very different from all other organ systems is that it's very dynamic, meaning that if an antigen enters the body at a certain site, local immune cells at that site try to eliminate the antigen, and some of them pick up the antigen, travel in the lymphatic system towards the closest draining lymph node, where they mount an adaptive immune response against that specific antigen. Then they can exit from the lymph node and the lymphatic system back into the blood and home specifically to the primary site of infection, but also to any other site of infection where the same antigen has entered in the body. That's why immunity is systemic. And this relies on the coordinated action of the innate and the adaptive immune system. The innate adaptive system the innate immune system is the first line of defense against any antigen. And actually the very first line of defense are the epithelial barriers of the skin and the mucous membranes of the lungs, the GI tract, and the genitourinary tract. These make a physical barrier that prevents any antigen from entering into the underlying tissue. But if this barrier is breached and an antigen goes through, then we have in all tissues what's known as tissue-resident innate cells. And those are mainly phagocytes, NK cells, mast cells, and some soluble components that phagocytose the antigen with the purpose of eliminating them. But by doing so, they also get activated and they secrete a large number of pro-inflammatory cytokines, such as TNF-alpha, IL-1, IL-6, that first activate the endothelial cells of the venules at the site of the infection, thus attracting more innate cells from the blood. First to come are monocytes and neutrophils, and they help eliminate the pathogen. Dendritic cells are a special type of innate cells that bridge the innate and the adaptive immune system. What they do is that they engulf the pathogen with the purpose of degrading it into small peptides and presenting these peptides on their surface in association with MSC class 1 and 2 molecules. And then they get activated, express homing receptors that take them to the closest draining lymph node, and they sit and reside there for a long time. Naive T and B cells keep circulating the body, entering from one lymph node to the other. When a naive T cell that has a T cell receptor that recognizes the antigen on the dendritic cell, then this T cell makes a stable connection with the dendritic cell that can last up to 3 days, during which both DC and T cell get activated. And then this naive T cell becomes an effector cell specific only for that antigen. And then it exits the lymph node, goes to the bloodstream, and home specifically to the site of the infection, where CD4 cells orchestrate the immune response against that antigen. And CD8 cells try to kill any cell that is infected with an intracellular antigen. These cells don't rely on dendritic cells to be activated. They can go to the lymph node, and then they recognize soluble forms of that same naive antigen, and they get help from this effector T cell to mount an immune response by secreting antibodies specific for that antigen. A defect in any one or more components of that system can result in an adaptive inborn error of immunity. And this immune response is very tightly regulated to prevent infection and decrease the incidence of tumor development without causing inflammation or autoimmunity. In case of immune deficiency, there is prevalence of infection and increased risk of tumor development, but sometimes it's combined with immune dysregulation, which favors the inflammation and increased risk of autoimmunity. So now inborn errors of immunity are extremely genus group of genetic disorders. Kids are born with them, and these disorders affect components of the immune system, either at the developmental level or at the functional level, or both, resulting in a wide spectrum of infections, sometimes associated with increased incidence of autoimmunity and malignancy. Underlying infections, because they are one of the first manifestations of inborn errors of immunity, but they are most of the time now associated and recognized to be associated with autoimmunity. And these inborn errors of immunity or primary immune deficiencies are classified into 10 different categories depending on the cellular component that is affected. And the most severe ones are immune deficiencies affecting cellular and humoral immunity, sometimes associated with syndromic features. And these are the ones characterized by T cell deficiency, sometimes associated with B cell or NK cell deficiency. There are antibody deficiencies, disease of immune dysregulation, and paradoxically, immune deficiency is associated with dysregulation because of loss of a special type of cells called regulatory T cells that orchestrate our recognition of self-protein from not. So these, if they are deficient, we have an increased risk of autoimmunity. Defects in phagocyte number and function, defect in innate immunity, inflammatory diseases, complement deficiencies, and inborn error of immunity secondary to bone marrow failure or phencopies of inborn errors of immunity. And these are the diseases where the patient makes autoantibodies against cytokines, for example, that eventually result in the development of an immune deficiency by lack of cytokine function. As I said, infections are the most prolific manifestation, but these infections are not the regular infections that healthy kids have. They are severe and recurrent. They are extremely unusual in type and organ they affect. They're mostly caused with opportunistic pathogens, and they are hard to clear and require often IV antibiotic and immunoglobulin replacement therapy. And the type of defect, the type of infection can really give an idea about the immune defect. For example, a defect in innate immunity can result in different bacterial and viral infections, especially those that affect the deep-seated infections that are found in the organs. Defects in humoral immunity or antibody-mediated immunity results in infection with encapsulated pus-forming bacteria and some viruses, or at the extracellular bacteria. And defects in T-cell-mediated immunity is widespread and lead to infection by viruses, intracellular bacteria, and fungi, highlighting the importance of T-cells in orchestrating the immune response. The most commonly seen infections are respiratory infections and upper respiratory tract infections, but skin manifestations are also prominent in inborn errors of immunity, GI infection, central nervous, eye infection, and bone and ligament infection as well. So since the infections are very prominent, the Jeffrey Mondale Foundation put out the 10 warning signs of inborn errors of immunity that is mostly used by clinicians to recognize that this patient might have an underlying immune defect rather than regular infections. And those include 4 or more new ear infections within 1 year, 2 or more serious sinus infection within 1 year, 2 or more months on antibiotic with little effect, 2 or more pneumonias within 1 year, failure of an infant to thrive, recurrent deep skin or organ abscesses, persistent thrush in the mouth or fungal infection on the skin, the need for intravenous antibiotics to clear the infection, 2 or more deep-seated infection, and last but not least, and this is one of the best ways to diagnose a novel case of inborn error of immunity, is a family history. If you have one patient in the history of the family that died early age or that was severely sick for a long time, that's a strong indication that the new patient might be having an inborn error of immunity. Infections are not the only manifestations. In fact, immune dysregulation is very prominent, and whether it's an autoimmunity where the immune system are no longer able to distinguish between self and non-self, resulting in loss of tolerance, such as Treg deficiency in the case of IPEX, or non-infections dysregulation as allergy, which can result in eczema, asthma, allergic rhinitis, or food allergy. Other manifestation is lymphoproliferation, non-infectious and non-malignant actually lymphoproliferation, where the immune response proliferation is unchecked, as in the case of XRP and ALPS, or an increased incidence of malignancy because of a failure of tumor surveillance, such as in the case of Prescott-Aldrich syndrome and other, but I'm stating the ones that have been studied most. The treatment of inborn error of immunity is two-pronged. One is to treat the symptoms, other is to cure the disease. To treat the symptoms, people use antibiotic to treat the infections at the time of infection, or in a prophylactic manner to prevent the development of infections. Steroids to reduce the inflammation that sometimes associate the infection. IVIG to complement the humoral defect in case of B-cell development failure or function failure and production of immunoglobulin. And those, as I said, are only treating the symptoms. However, to definitely cure inborn errors of immunity, you need hematopoietic stem cell transplantation or bone marrow transplant and gene therapy in some cases. Although these diseases are individually rare, their incidence is relatively high, especially in the Middle East and North African region, and as well in the Asian countries such as Pakistan, India, and other countries. And this is because of the high rate of consanguineous marriages that favor the segregation of disease-causing mutations within countries, even regions, and sometimes within families only. And those diseases are really not easy to study. That's because they are genetically very diverse. There are approximately 10,000 genes expressed in the immune system. So far, only 450 have been associated as monogenic causes of inborn error of immunity. So there are a lot still to be discovered. They are phenotypically extremely variable, meaning that mutation in the same gene may have different clinical phenotype in two patients, or mutations in different gene can have similar clinical phenotype, thus confusing the treating clinician and scientist. And they are technically very complex because biologic assays are essential to characterize them. But these biologic assays are highly specialized and personalized, and in most cases are not cost efficient to be introduced in clinical lab. That's why they are done mostly on a research basis. They require viable cells from the study participant, which we know sometimes is extremely hard to get because the patient cannot give blood or doesn't want to give blood, or because the blood needs to be shipped overseas, which can take up to 2 days to get to the destination, and then the viability is not optimal, and they are costly. The most comprehensive comprehensive approach to study them is a combination of genomic technologies and functional tests. The genomic technologies identify the causative variant in most cases. However, it doesn't give any information about the biologic effect of the variant. The functional tests are essential to characterize and mechanistically investigate the disease. However, they cannot identify the causative variant. So really a combination of both genomic and functional is extremely important to elucidate the disease and its mechanism. And the strategy that we have used so far to study the patient is first to recruit the study subject with the phenotype of inborn error of immunity. And as we get the subject in the lab, we immediately do exploratory functional studies. And this is extremely important because it will give us an idea if there is any defect in the development or function of one or more cellular component, which will guide our analysis of the genetics. And then in parallel to this, we do genetic screening using a targeted gene panel approach to sequence 300 genes only from the patient, which have been associated with inborn error of immunity or some novel ones that we add to our panel. If we find a known mutation in a known gene, then the study is complete. If we find a new mutation in a known gene, then we still do mechanistic studies to prove the pathogenicity of the mutation and its association with disease. If we don't find any candidate variant using the targeted gene panel approach, then we can do whole exome and genome sequencing depending on the interest we have in the project, followed by mechanistic studies to finish the study. And now for the rest of the time, I'm gonna share with you two cases that we recently studied in my lab. The first case is that of a female born to first-degree consanguineous parent. She's the only daughter of this young couple. Her first manifestation since birth was hemolytic anemia, highlighting the autoimmunity that develops sometimes earlier than the infection in the patients, depends on the pathogens around them. And we know that in the last 2 years, because of COVID and all the measures of protection, kids are getting less sick, and then their manifestation could be autoimmunity before the infections settle. However, she had some modest upper respiratory tract infections and oral thrush. At 6 months, she's very young, she had bronchiolitis, and at 7 months, she had a rotavirus infection with jaundice, nausea, and episodes of vomiting for which she was hospitalized. So when she was hospitalized, they did 2 clinical tests, but then at 8 months of age or 10 months of age, she was referred to our lab. And we did a detailed immune phenotyping of the patient. And on all the slides that I'm going to show you, the top panels are the patient and the lower ones are the control. So here on the y-axis, we're looking at the B cells. And apparently, she doesn't have B cells, but this is not the case. I'm going to get back to this point a little bit later. For now, keep that in mind. However, the percentage of her T cells are less than the control and the range for age. However, they developed into CD4 and CD8 cells normally. Her NK cells are normal. To determine the origin of these T cells, we look at the recent thymic immigrant, which is this population here that is CD31 positive or A positive. And the patient doesn't have any recent thymic immigrant as compared to the control in the lower panel, indicating that her thymic output is defective, and whatever she has in the periphery is probably maternal engraftment or the expansion of a few clones in the periphery, which could be autoimmune. Her T cells are all of the memory phenotype, as you can see here, and this is abnormal for a young kid. They should be all mostly naive. So she has a defect in the ratio of naive to memory cells, indicated continuous activation whatever T cells she has. And as I said, her B cells, keep an eye on the B cells. So she was referred to us at 10 months of age here, but before that she had 2 clinical immune phenotyping done in her local hospital, which showed low T cells at all time points. However, she had normal numbers of B cells, but because her hemolytic anemia was thought to be autoimmune in nature, they gave her rituximab, a monoclonal antibody, to deplete her B cells, which resulted in the complete depletion of her B cells. And when she came to us, we thought she didn't have B cells. That would have skewed our analysis towards C minus, B minus phenotype, but in fact, it was secondary to the biologic they treated her with. So she is a B plus and a T low cell. She has low T cell, as I said, at all the time points she studied. And most importantly, her T cells do not proliferate when stimulated with PHA or anti-CD3. This is done using the dye dilution method, which when the cells proliferate, they move towards the left on the histogram. And the control T cells in blue proliferated normally, whereas the patient's T cells in red failed to proliferate. They're completely dysfunctional. So at this point, we suspected that she was suffering from an inborn error of immunity, and we did the target gene panel approach on her. And we identify a novel homozygous variant in the IL-7 receptor alpha gene. It's a G2NA at positions 379 in the cDNA. It's actually the last nucleotide in exon 3, and it resulted in a change of valine to isoleucine at position 127 in the extracellular domain of the protein. So as I said, the patient is homozygous for this variant compared to the control here, and both parents are heterozygous for the same variant. So to determine if this variant is pathogenic, we looked at the expression of the IL-7 receptor alpha on the T cells from the patient here, and this variant seems to have completely abolished the expression of IL-7 receptor alpha in the patient as compared to the control. Most of the T cells express IL-7 receptor alpha. And the percentages of T cells from both parents that expressed IL-7 receptor alpha was normal. However, the level of expression on their T cell was half that of several contours, which is consistent with the heterozygous nature of the variant they have. So at this point, we were sure that this variant is pathogenic and hence is classified as a mutation. However, we were still surprised at how come a missense mutation completely abolished protein expression. As I said, it's the last residue in exon 3, so we hypothesized that it might be affecting splicing of exon 3 to exon 4. So we designed a strategy to amplify exons 2 to 4 from the cDNA of the patient, and this gave the expected 400 base pair band in the control. However, the patient band was shorter, and then when we sequenced it, it showed complete splicing of exons 2 to 4, bypassing exon 3, and thus taking the protein out of frame. So in fact, what the next-gen sequencing gave us as a valine 127 to isoleucine was not correct. It's actually a termination at position 74 in the protein, leading to a truncating and out-of-frame protein. Based on this, the patient was transplanted at our center, and then we followed the reconstitution of her T cells after transplant up to 14 months. And all of these graphs, the vertical line is the time of transplant, and this graph shows complete reconstitution of CD3, CD4, and CD8 cells. But most importantly is this red line, which are the recent thymic immigrant, that are the population that immediately came out of the thymus, that increased around 20-fold after transplant, indicating normal transplant, engraftment, and thymic outputs. The percentage of naive and memory cells completely normalized, and she had the production of NK cells and B cells 14 months after transplant. However, they were still a little bit low because the patient was still on steroid at 12 months after transplant, because at 7 and 9 months, she had episodes of autoimmune hemolytic anemia, and she was put on steroid. So at that point, at 12 months, we took some sample from the patient and we looked at the Tregs, a population of cells that is important to prevent autoimmunity, and found that her Tregs percentages are normal, but most importantly, they express the effector molecule CD25, CTLA4, and Aelios, indicating that they are functional. And in fact, like a month after we did this test, the patient was taken off steroids. And then we looked at the expression of IL-7 receptor on the patient's T cells, and most of her T cells express the IL-7 receptor, indicating that they are donor-derived. However, their level of expression was half that of several controls, indicating that the donor, who was her paternal grandmother, might have been a heterozygous for the same mutation. Nevertheless, her cells express IL-7, and they proliferated when stimulated with PHA and TCD3, however, a little bit less than the control, as I said, because she was still on steroid when she did the test. So this case shows how we could use some functional tests to get an idea what to look for, do the genetics, find the mutation, study the mechanism of disease based on the mutation, and then treat the patient based on these studies and follow the reconstitution after treatment. I'm gonna, for the next 5 minutes, I'm gonna talk about another case that we did in the lab. It's a case of Wiskott-Aldrich syndrome. But what was confusing about this family is that they are consanguineous, and people, whoever knows about WASP, know that it's an X-linked disorder. So the first boy who was referred to us was 5 years old. He's from consanguineous parents, as I said. He suffered from thrombocytopenia, eczema, and recurrent infections. And this is the triad that characterizes Wiskott-Aldrich syndrome in males. So we thought that, you know, this is Wiskott-Aldrich syndrome, but the parents are consanguineous. What's the likelihood of having an X-linked disease in a consanguineous family? Usually you look for autosomal recessive mode of inheritance. So that's why we didn't do functional tests. We were really stimulated by this project, and we did directly the sequencing. And in fact, we found a variant in Wiskott-Aldrich syndrome in the cDNA. It's an exon 2 C to a T change at position 134, which changes a threonine to a methionine at position 45 in the EVH1 domain of the protein. And I'm going to get back to that EVH1 domain a little bit later. When we told the clinician that this is what we found, he told us he has 3 other brothers, all suffer from different types of thrombocytopenia and bleeding episodes and some infections. So at this point, we got DNA from the siblings, and we did Sanger sequencing, not next-gen sequencing. We Sanger sequenced the region of the mutation that was found in the first patient, and we found that all 3 siblings had the same mutation as the one detected by next-gen sequencing. And the mom was heterozygous. As I said, this is an X-linked disorder. So it's a consanguineous family that has only 4 boys, and they suffer from an X-linked disease, all 4 of them. Now, to go back how we functionally studied the mutation, WASP is expressed only in hematopoietic cells, and it's found in the cytosol in this closed resting conformation. When the cell is not activated, the WASP is an autoinhibitory conformation associated with another protein called WIP, as well as adapter molecules. Upon activation of the cell, a series of events happen that leads to the opening of the molecule, and then this VCA domain at the end of the protein is the active site that mediates actin polymerization. And this is one of the earliest events that happens when T cells or B cells get activated. They polymerize their F-actin downstream of the T cell receptor or B cell receptor. And this is important for sex signaling, migration of the cells, shape change, adhesion to the cellular domain. And this actually protein is extremely unstable, and it's stabilized by interaction with WIP. WIP is another protein that binds to the EVH1 domain of WASP and mediates its stability. And actually, a lot of patients with WASP or XLT have mutations in the EVH1 domain that decrease the affinity between WIP and WASP, leading to the dissociation of the complex and degradation of WASP. So we thought maybe that the mutation in the EVH1 domain decreased the affinity between the 2 proteins and leads to the degradation of WASP. And in fact, this is what we found by immunoblotting. All 4 patients have either absent or completely negligible amounts of WASP as compared to the parents and healthy controls. So this showed that this mutation destabilizes the protein. However, what we were surprised to find was that WIP as well, the partner of WASP, its level was decreased by 80% or a little bit more in the patient, indicating that the association between the 2 proteins is important for both stability, and this was not described in the literature. WASP has been studied in more than 400 patients, and nobody reported that WIP also is decreased in WASP patients. So we showed that the association between the 2 proteins is important for the stability. And this stability is at the level of the protein, not the mRNA, because the mRNA of both WASP and WIP was normal. So we have a mutation that affects the interaction between WIP and WASP that results in diminished stability of both proteins. And then, as I said, the first event downstream of TCR signaling is F-actin polymerization. So we looked at the levels of F-actin at resting stage. It was not decreased in the patient as compared to the control. However, when we activate the cell, the control cells polymerize F-actin more efficiently than the patient cells, indicating that the contribution of WASP to F-actin polymerization has been abolished. And consequently, T cells from the patients fail to proliferate, or they proliferate poorly when stimulated both with plate-bound and soluble anti-CD3. However, their B cell proliferation is completely normal. So this is another confusing case in a highly consanguineous population where you have 2 cousins getting married. However, all their boys have an X-linked disease that we studied. So I want to end here and thank you again for hosting me. And I want to acknowledge people in my lab who did this work, mostly Dr. Mansour, who is a postdoc in the lab, and 2 RAs and graduate students. I have collaborators all over Lebanon at the American University of Beirut Medical Center and another hospital, the Rafiq Hariri Hospital. And also I have a long-standing collaboration with Dr. Talal Shatila and Raif Shah at the Boston Children's Hospital. And I'd like to thank the funding agencies for funding our work. Thank you very much.
Thank you, Dr. Masad, for such a wonderful lecture. There is only time left for one question. So,
Sure.
So if you have a question from the audience, only one short question.
Okay, sure.
Ji.
That was an excellent presentation. Really enjoyed the presentation. And I have just one general comment, and that is, we know that the innate immunity recognizes unprocessed antigen, and T-cell immunity, adaptive immunity, recognizes processed antigen. are there any differences whereby you only the innate system is affected? And if so, why?
Yeah, in inborn errors of immunity, there are diseases that affect only the innate system, and those are the defects in phagocyte development or function, neutrophil activation, and then macrophage migration. Those defects affect only the innate system, and then you can see the initial phase of the immune response, which is the clearance of those antigens in the organs affected. And then these pathogens can start become proliferating in the organs, causing abscesses in the skin or the internal organs. And this is an indication of a defect in the innate immune system before the adaptive immune system kicks in.
Thank you very much, Dr. Masad, for sharing your wisdom with us. And there is an announcement from the organizer that after this lecture there is a tea break, and after the tea break, then we will start with the free oral presentation. So thank you to all the 15 minutes, 15 minutes tea break. Thank you very much, everyone.
Thank you.
We will now start with, we will now resume the first session. This part will comprise of free papers. I will now request the honorable chairs to please take their positions. Thank you very much.
Okay, welcome everybody to the second part of this sessions after the tree. So the first presenter in this second part of this session is Bushra Mubarak. She is from University of Health Sciences, Lahore. She would be talking about association of interleukin 38 level and its polymorphisms with rheumatoid arthritis. So, Bushra Mubarak. You are having 5 minutes to wind up your lecture.
My name is Bushra Mubarak and I am from University of Health Sciences. The topic of my presentation is association of interleukin 38 and its polymorphism with rheumatoid arthritis. I would like to start with a brief introduction of rheumatoid arthritis. Rheumatoid arthritis is a chronic inflammatory autoimmune disease and it is characterized by immunoglobulin G and citrullinated peptide antibodies which cause or induce inflammation and leading to cartilage damage and bone destruction. Globally, rheumatoid arthritis affect almost 0.5 to 1%, and in Pakistan, its prevalence is 0.5%. As well as pathogenesis is concerned, there is cellular and humoral immunities involved, and there is a abrupt release of cytokines in rheumatoid arthritis patients and in animal models. The main cytokines which is involved in it is interleukin 6, interleukin 1 family cytokines and TNF alpha. The interleukin 1 family cytokines include 7 agonistic cytokines and 4 member which shows antagonistic action, means anti-inflammatory action, which includes interleukin 38, interleukin 36, interleukin 37 and interleukin 1 receptor antagonist. My topic is on interleukin 38, and interleukin 38 is interleukin 1 family, and it is its main function is anti-inflammatory. It inhibits interleukin 38 receptor signaling pathway in autoimmune diseases in like rheumatoid arthritis, SLE. There is a difference in cytokines dysregulation, and interleukin 38 also alters its expression in rheumatoid arthritis and other autoimmune diseases. Like Zirabi et al. Reported a study in which the interleukin 38 level in new cases of multiple sclerosis and systemic lupus, in systemic sclerosis, there is increased number of interleukin 38, and Z et al. Reported a study in which there is decreased number of interleukin 38, decreased level of interleukin 38 in rheumatoid arthritis. There are some polymorphism studied in interleukin 38, which includes RS1704-888 and RS674-3376. My topic is on RS674-3376, which is a missense SNP of interleukin 38, located on and in this SNP there is a mutation from A to C. Walker et al. Also studied this SNP in rheumatoid arthritis and reported that there is decreased severity of rheumatoid arthritis within this SNP. With this background, my main aim is to see the association between interleukin 38 and its polymorphism with rheumatoid arthritis, with two main objectives: to determine the level of interleukin 36 in rheumatoid arthritis and healthy control. And second objective is to determine the frequency of interleukin 38 polymorphism in patient of rheumatoid arthritis and healthy control. This study was conducted in University of Lahore, and it is a case control study which is conducted on 80 individuals, 40 RA patients and 40 healthy controls. And this is the inclusion criteria in which ACR criteria will be, those patients which fulfill ACR criteria is enrolled in this study, and those patients with co-existence with any other autoimmune diseases like SLE or any other, it will be excluded from the study. Sample 5 ml venous blood will be collected in EDTA vacutainer, and in EDTA vacutainer DNA extraction will be done, and then polymorphism will be done by allele specific polymerase chain reaction. And in 2 ml blood will be added in was added in gel vacutainer for serum separation for measurement of rheumatoid arthritis, ACCP, and interleukin 38 level by ELISA. This is the sequence of primers which is used for allele-specific PCR. And this is a gel picture for genomic DNA extraction from blood sample. And this is a picture for PCR amplification product of SNP, C and A, and my product size is 235 base pair. This is the result in which age and gender matched controls were taken with patient sample, and CRP rheumatoid factor and anti-CCP. In CRP is 70% positive in rheumatoid arthritis patient. Rheumatoid factor and anti-CCP were 100% positive in rheumatoid arthritis patient. As well as interleukin 38 is concerned, in cases 26.86 picogram per ml interleukin 38, and in control there is 19.27, and there is no significantly a difference was found between these two, patient and control. When genotype is done in patient, AA genotype is more as compared to controls, and there is a significant difference with odds ratio of 9.
Time is over, so you can wind up only 1 minute in 1 minute.
Okay. And a conclusion of the study is to main genotype, which is the homozygous AA, is a risk factor for rheumatoid arthritis in our study, and other two genotypes, which is AC and homozygous CC, is play a protective role in our patients. And this is the main conclusion. Thank you.
There is a time for one question. Yeah, Sabir.
By using a formula, I skipped that formula because there is was informed it is 7 minutes presentation. That's why. Yes, I calculated it. I calculated it, and there is 80 sample size, which divided into 2, healthy and patient.
Thank you very much for presentation. There is a suggestion that if you stratify your patients according to severity of disease, that is more likely to be related to underlying.
Also categorize the patient in with the severity of disease, but the main topic, as you know, there is a very short time. I just entered the result about interleukin 38 and polymorphism. I didn't really add it in.
Okay, thank you very much. So, next. Positive patients. So over to in time.
Assalamu alaikum everyone. So I'm going to start my presentation with my topic name, frequency of autoantibodies to nuclear antigens in rheumatoid factor positive patients. So this is the introduction. Anti-nuclear antibodies are autoantibodies direct against nucleus of the cells and its components, and it is the biomarker, a key biomarker in the diagnosis of autoimmune diseases. Rheumatoid factor is a protein found with various isotypes directed against Fc portions of the immunoglobulin G, and it is a key player in immune responses in many infectious organisms, and high affinity indicate more severe and persistent disease in patients with rheumatoid arthritis. The correlation in ANA and RF may be useful in the diagnosis of other autoimmune diseases in the patient having RA, rheumatoid arthritis. This is a picture. There are many ANA patterns, but the most common patterns of our study shown in this diagram: homogeneous pattern, centromere pattern, and fine speckled rod rings. So the rheumatoid factor binds with commonly IgM and IgG, and rarely binds with IgA, IgD, and IgE. So in this picture, IgM RF factor is shown and IgG RF factor is shown. This is a mechanism how RF factor is made and reacts with IgG and IgM to make complex, and after the complex form, it activates complements which produce inflammatory cytokines, and then it's produce inflammation, destroy bones. So the objective of our study is to determine ANA patterns in rheumatoid factor positive patients. Study design was retrospective descriptive study. We take 100 patients, fulfill our inclusion criteria which were positive for ANA and RF. The time period of the study from January 2020 to September 2021 at SIUT. ANA was done by commercially available kits by indirect immunofluorescence assay, and RF was done by nephrometry. The data analysis were done by SPSS version 20. This is a demographic of gender distribution. We took 100 patients positive for ANA and RF, in which 65% of the patients were female and 35% were male. In this pie chart, this is the main pie chart of our study, shows that the patient who were positive for ANA and RF both found to be 64% of speckled patterns and 28% of homogeneous, 5% of nucleolar, and 1% of mitotic rods, rings, and rims for each. This is a bar chart of ANA titers. The patients, 20 patients were found to be 1:80 titer, 38 were found to be 1:160. These are basically the dilutions we performed on our study population. The patients who were positive for ANA and RF also found that there were 23% of the population positive for double-stranded DNA, and 42% were negative. The rest, 36%, was not performed for this test. So the conclusion is anti-nuclear antibodies are key biomarkers in the evaluation of rheumatic diseases such as systemic lupus erythematosus, Sjogren's syndrome, and mixed connective tissue diseases. Anti-nuclear antibodies are closely associated with specific diseases and other non-specific in both patients and healthy individuals. Anti-nuclear antibodies are useful screening and diagnostic biomarkers, provide insight into the understanding disease mechanism. Thank you.
Thank you very much for the presentation. So, house is open for question. Dr. Sabiha.
Positive for rheumatoid factor and anti-nuclear antibodies. We took 100 patients and they were positive for ANA and RF both.
Okay, you just took the laboratory criteria,
Ji.
but not the rheumatoid arthritis patient.
No, no, no.
Okay, so your conclusion is somewhat, I could not understand. I mean, what is the conclusion that in the patients with rheumatoid fracture positivity, you have this ANA? And so what is the clinical significance of this?
Basically, the conclusion is we have patients who are positive for RF and ANA as well. So मेन चीज़ ये देखनी थी कि जो पेशेंट्स हैं जो जिनके अंदर RF पॉजिटिव आ रहा है और साथ साथ ANA भी आ रहा है, तो रूमेटॉइड आर्थराइटिस के अलावा और कौन कौन सी ऑटो इम्यून डिजीजेस जो हैं उन पेशेंट्स में हो सकती हैं. मतलब RF पेशेंट उनके अंदर है वो.
So how many were there them were rheumatoid arthritis patients? Rheumatoid arthritis के कितने थे इसमें से?
They were मतलब जो मैंने स्टडी की है उसमें जितने भी पेशेंट्स लिए गए थे 100 वो सारे RF फैक्टर पॉजिटिव थे. अब ये इसका डायग्नोसिस नहीं मतलब पता कि कितने RA के लिए थे और कितने से सिर्फ RF फैक्टर पॉजिटिव थे.
So thank you very much for the presentation, and I think this is the last one. There is one more. Okay, so we don't have. Okay, this is on the other page. So the third presenter of this session is Hamna Hanif. She is from Indus Hospital and Health Network. She would be talking about impact of consanguinity in a patient with primary immunodeficiency. So, Hamna Hanif.
Bismillahirrahmanirrahim. My topic is the impact of consanguinity in parents on patients with primary immunodeficiency. So this is the outline of my presentation, which I'll try to cover in 5 minutes. So what are primary immunodeficiency diseases? They are defects in the innate or the adaptive immune system that result in recurrent, severe, and unusual infections. They may be associated with autoimmunity or malignancies, and all PIDs, they may present in childhood or unusually present later in life. The topic is consanguinity, and what exactly is consanguinity? It is a union between two persons who are related as second cousins or closer. And as all PIDs are genetically determined, therefore consanguinity has an effect on the incidence of PIDs. In Pakistan, the rate of consanguineous marriage is the highest in the world. It stands at 65 to 70%. And at present, there is a lack of population-based studies on the prevalence of primary immunodeficiency diseases in Pakistan. So what was the purpose of my study? It was to determine the presence of consanguinity in the parents of patients suspected with or diagnosed with primary immunodeficiency, and to compare the age, gender, clinical presentation, and family history in all those patients, whether they had or did not have parental consanguinity. So it was a retrospective study that was carried out at the Department of Immunology at the Indus Hospital and Health Network. All the patients who were referred to the immunology clinic with a suspicion, or those who were later diagnosed with PID from January 2020 to September 2022, irrespective of their age and gender, were included in this study. And all these patients were assessed for the following demographics, clinical findings, and investigations, including genetic analysis, were available. So starting with the results, the total number of patients we included in our study was 44, out of which we had 61% were males and 39% were females, and majority of our patients, 86% were children, while 14% were adults, and the mean age of our patients was 8.6 years. Overall, parental consanguinity was positive in 30 of our patients, that is 68%, while it was absent in the remaining 32%. And out of 44 patients, we could confirm primary immunodeficiency in 13 patients, and in 31 it was still suspicious, and our diagnosis included congenital neutropenia, hyper-IgE syndrome, hyper-IgM syndrome, agammaglobulinemia, common variable immunodeficiency, Shiitake-Higashi syndrome, hemophagocytic histolymphocytosis, and Shosman-Diamond syndrome. And coming to the clinical findings, the most common clinical finding we found in our patient was chest infections. That was followed by skin infections and GI infections. Ear infections and meningitis were less common. And in investigations, in majority of the patients had normal immunoglobulins levels, and similar was with complement levels. And we also performed anti-PCP antibodies, and majority of the patients had low titers of anti-PCP antibodies. And coming to the lymphocyte subset, which we performed twice in every patient for confirmation at an interval of 4 to 8 weeks, the most common abnormality seen was low B cells followed by low NK cells. And when we compared the two classes of patients, those who had and those who did not have parental consanguinity, we found that it did not have any significant impact on the age of presentation of our patients. And similar was the case with gender. There was no relation of parental consanguinity with the gender of patients with PID. However, when we come to family history, we found that in those who had parental consanguinity was present, family history of PID or family history of recurrent infections consistent with PID was present in 60% of the patients, while that was only present in 36% of those in which consanguinity was absent. In total, family history was positive in 23 of our patients, and of these, 78% had parental consanguinity. And confirmed diagnosis, in the 13 patients in which we established our confirmed diagnosis, 77% of the patients, they did have consanguinity. And when we tried to find an association of consanguinity with the clinical findings of our patients, although our p-values were not significant, but the closest association we found was with chest infections, in which when consanguinity was present, 63% of our patients had chest infections, while when it was absent, only 36% presented with chest infections. And similar is the case with autoimmunity. Consanguinity, parental consanguinity did not have a significant impact on whether our patients with primary immunodeficiency presented with autoimmunity and malignancy. We had very few patients, only 3 patients in total who presented with malignancy, and that did not have a significant relation with parental consanguinity. Finally, how did we manage our patients? All of them were given antibiotics according to the culture and susceptibilities. Intravenous immunoglobulin was given to 9 patients. Recombinant granulocyte colony stimulating factor was given to 2 patients, and steroids to 3 patients. And in conclusion, parental consanguinity did have an impact on the incidence of primary immunodeficiencies in our local population. In our study, the rate was seen at 68%, which is compatible with the rate we found in our general population of 65 to 70%. And this warrants a need to do population-based study to assess the burden of these genetic disorders in our patients and to establish good diagnostic services to reach a confirmed diagnosis for effective patient management. Thank you.
Okay, thank you very much, Hamna Hanif, for the presentation. So, house is open for question. Ji.
Thank you very much. The inheritance of these primary immune deficiencies is very well described. So it would have been better if you had stratified it according to X-linked, autosomal dominant, autosomal recessive, and then tried to see whether consanguinity, what kind of impact it is having on our own patients, and just studied this factor, because the phenotype of the disease, chest infection or other things, they would depend on the underlying defect, not on consanguinity. There's no relationship with that. So the important thing is to have your population carefully identified, and if there is an X-linked disease, was there any impact of having family history or autosomal dominant or autosomal recessive? So there was a lot of thought which should have gone into it. Thank you very much.
Yeah, yeah.
So actually it's a very small size. You can see that we have 44 patients that we have seen. So that is the basic idea, to build on the larger population-based study on this small sample size, and then we can have a stratified.
So, thank you very much, all the presenter, and I would request Dr. Saab to just conclude these sessions and just have a brief overview of all the presentation.
Thank you very much. What a pleasure to have these talks and see the enthusiastic participation by the young presenters. There is so much to do, as correctly pointed out by Dr. Sabia, and we listened to Dr. Sabia about her project of autoimmunity and immune deficiency. That's so important, and there are so many gray areas. And then from Dr. Masad from UK, inborn errors of immunity, and we are seeing in our 3 centers or maybe 4 centers a number of patients with immune deficiency, and they are having international collaborations as well. And we need to do a lot, as has been pointed out. And for the young speakers, just these presentations are extremely important. So prepare for them, study, understand the subject, and then a lot of thought needs to go into the organization of the project. So that is way forward. Thank you very much, and it has been a pleasure having Professor Raza with us as the chair and myself as co-chair. Thank you.
Sir, thank you very much. So before we conclude this session, if you have any remarks.
So it's a matter of great pleasure for me to be here, and especially with Dr. Sabiha. I have a long association. We were working together on clinical trials while in some other studies, and we are also planning for the future. So the talks were wonderful, especially Dr. Tahir talk was one of the best I have seen, although it was beyond the time, but it was, you know that if a scientist start talking and if someone love the field, then he do not realize that what is the time bar. So that shows that how much he is having a love for this field. The other talks were also brilliant, and for the students who presented the presentations, and they were also up to the mark. And we have also given some remarks on them, and best of luck, and I really appreciate and I congratulate Dr. Sabiha for organizing such a wonderful event. Thank you so much, everyone.
Thank you very much, sir. Before we conclude this session, I will now request Professor Rabia Hussain to kindly give away the mementos to the honorable chairs. And thank you very much. Professor Dr. Raza, sir, please. Brigadier Tahir Aziz Ahmed, sir. Thank you very much, ma'am. Thank you. I will now request Professor Dr. Raza Shah to please give away the mementos to the invited speakers. Dr. Sabiha Anis, please, ma'am. Brigadier Tahir Aziz Ahmed, sir. Sir. Sir, आपकी as a speaker. Sir, thank you very much, sir. This concludes the first session.
Tahir, can I request you to please come and give away the shield to the moderator of a session? Thank you. Now I request Dr. Aisha to please come and take over for this.
Sir, thank you very much. This concludes the first session for today. The e-certificates for all the participating speakers who presented the free papers will be dispatched on their email addresses. I now invite the moderator for the second session, please.
Assalamu alaikum everyone, and welcome to the second session of today's morning. The session is about molecular and cellular immunology. I would like to call upon our respected judges, Dr. Junaid Iqbal. He's an assistant professor and research director at the Nutrition Research Laboratory at the Department of Pediatrics, Aga Khan University. His main interest is in stem cell research and involves in various research projects. He has authored several articles in peer-reviewed national and international journals. Please, Dr. Iqbal, take a seat. The second chair is Dr. Kiran Iqbal Masood. She is an assistant professor in the Department of Pathology and Laboratory Medicine at the Aga Khan University and Hospital. She obtained her PhD in microbiology and immunology from the Aga Khan University. She has received several awards and presentations, including Bill and Melinda Gates Awards, and there is a whole list of those awards. She is a co-supervisor and supervisor mentoring PhD students. She has authored several articles published in peer-reviewed national and international journals. I would like to request our chairs to please convene the session.
Thank you so much. So we'll start with invited lectures, and our first invited lecture is from Dr. Talat Rumi. Dr. Talat Rumi is currently working at Dow University of Health Sciences, from where she is working for last 11 years. She's an associate professor of molecular medicine and director of Dow Institute of Advanced Biological and Animal Research. She has vast experience in biological sciences. She has worked in a number of international institutes, including Case Western Reserve University, Cleveland Clinic Learner Research Institute at Ohio, University of Aberdeen, Scotland, Singapore Immunology Network, MDC Germany, and Genome Center, Cambridge, UK. She was awarded the prestigious Fulbright Scholarship, Wellcome Trust Award, Charles Wills Fellowship from UK, and various national and international grants and awards. Her current projects involve, she is involved in a number of projects currently, and the most notable is that she's involved at Dow in the development of anti-snake venom production and development of conventional DNA vaccines, toxoid monoclonal antibodies. She has published more than 30 research papers in reputed international journals and have more than 300 citations. So I would request Dr. Talat Rumi to come to the podium and give her talk. Thank you so much.
Assalamu alaikum everyone. Thank you, Dr. Junaid Iqbal, for the generous word. And first of all, I am very grateful to all the organizing committee of Pakistan Association of Pathology, and especially Pakistan Society of Immunology, for giving me this opportunity. Snake bite and venom is a neglected tropical disease and posing a public health challenge globally. In South Asian region, the burden of snake bite and venoming is quite high, and it's contribute approximately 70% global death. And on the other side, the availability, accessibility, and affordability of anti-snake venom is another challenge, and even it's a dilemma in our region. Millions of people are affected by snake bite envenoming, and it's become a significant source of death. WHO set a goal to halve the snake bite mortality by 2030. According to WHO report, 74,000 people are bitten by a snake every day worldwide, and approximately 5 million snake bites are reported yearly, causing between 81,000 to 138,000 deaths worldwide, with 400,000 amputation and permanent disability. WHO strategy for prevention and control of snake bite envenoming based on 4 objectives: empower and engage communities and ensure safe and effective treatment, increasing partnerships. For that, WHO has designed phase-wise plan. At the first phase, this is a pilot phase, they are engaging 10 to 12 countries where the snake bite envenoming rate is quite high. Then they will incorporate 35 to 40% and then 40 countries, and then in rollout phase they will involve all the countries affected by the snake venom. According to their phase-wise extended plan and the objectives and the goals, they have allocated budgets and implementing their target to reduce 50% snake bite mortality by 2030. The Association of Southeast Asian Nation, the snake bite incidence is quite high, and Pakistan lie in this region, and this graph represents that snake bite associated morbidity and mortality comes after dengue, which is quite alarming, and I will share some few more alarming facts and figures that Pakistan is mapped out in high incident region where estimated annual snake death is more than 10,000. It is another alarming figure that we live in the region where the snake bite associated mortality is 54,000 yearly, which is almost more or less equivalent to the global rate of the mortality. This graph actually depicts that global and regional snake bite envenoming mortality rate from 1990 to 2020 and forecasting to 2050, and we are lies in a green lines, and which gives us an idea how strictly the government, our government and NGOs has to focus the WHO guidelines. If we look into the percent change in death from 1919 to 2019, the question is that where we are. In 30 years, only 1% change in death in Pakistan. However, all the neighboring countries, if we talk about India and Bangladesh, they have controlled the mortality and morbidity by 45%, and other Islamic countries control it 70 to 90%. Why we are standing with this figure? Only one reason: the Pakistan, we are not producing indigenous vaccine. We are not focusing and not investing in the development of the biological product in Pakistan. This actually represents the percentage share of total economic burden associated with the snake bite morbidity and amputation and disability. ये सारा खर्चा करना की ज़रूरत ही नहीं है, सिर्फ एक जगह खर्चा करना है, development of anti-snake venom. If we does that, this whole economic burden goes down. Okay, so if we look into South Asia, there is 300 snake venom, and they are, and among that 70 are the highly poisonous. But I am focusing here the big 4, that is skate, cobra, soy scale viper, and wrestle vipers. They falls in the category 1 and responsible in high level of morbidity, disability, and mortality. Okay. In Pakistan, we have 4 anti-snake venom which are prevalent, and they are highly poisonous snake venom, and we need to produce anti-snake venom against that. WHO reported in 2017 approximately nearly 40,000 deaths occur annually, with 10,000 deaths. But this is just a tip of the iceberg, because we are the nation who don't believe in reporting. We have a lack of integrated and proper surveillance system. So it means that the morbidity and mortality associated with this snake bite is quite high. In this graph actually represents the month where high incidence of snake bite observed, men's, farmers and children mainly affected, and this really gives an idea that we have to develop anti-snake venom, and we totally based on the importing snake venom from India. It's why? But this supply is very irregular. The reason is that Indian manufacturer in Asian country, Indian manufacturer are not producing regularly, and they have certain issues. They seize their production facility because of the price control reason, poor animal husbandry, shortage of venom. Additionally, the most important point, intra-species variation occurs in snake venoms. So maybe the Naja cobra is similar here in Pakistan part and India, but due to on the basis of location, their venomics and the proteomics is entirely changed. So to produce effective antivenom, we have to develop our own antivenom in Pakistan. Right. At the moment, DHS, when it started our project, and now we have submitted our drug to Drug Regulatory Authority for the registration. We have completed 3 trial batches, and we have completed all the stability data, and we have completed all the preclinical testing, and because it falls in the biosimilar development of anti-snake venom, so it does not require to perform the phase 1 and phase 2 clinical trial. We will perform phase 3 clinical trial or maybe post-marketing surveillance. In Dow University of Health Sciences, we have a huge plant that deals with the upstream processing and downstream processing. Upstream processing is the development of hyperimmune plasma, and downstream processing is the development of the finished product, means the purified immunoglobulins. We have a CGMP and GLP and GMP compliance facility, and in 2020 Drug Regulatory Authority Pakistan has been awarded drug manufacturing license with the design and approval of vaccine and antisera and various other blood products development. We believe and our landmark is to work as a manufacturer and to serve the healthcare industry. This is the institute, Dow Institute for Advanced Biological and Animal Research, which deals the all aspects of biological and biomedical aspects related to research, R&D, and mass scale production of hyperimmune plasma. This is the major component of this institute. We have a central animal house facility where we house more than 100 horses, which we immunize with the venom in a different group under the supervision of expert veterinary doctors. That we call equine management system, otherwise we cannot produce such biological product. We have a basic manufacturing facility. The 2 component is very important for us: venom collection and production processing facility, and plasma collection and processing facility. We have a huge quality operation system which deals with the quality control and quality assurance, and we have a facility for the preclinical testing, and experts and a trained skillful staff are available in the facility. The whole system is governed by more than 230 SOPs, and we focused and obliged to work under the 20 approved ACUG guidelines that have been developed and approved in the University of Health Sciences. As I mentioned, that we are working and producing antivenom against these 4 poisonous venom, and it is very important when we choose venom to elicit the immune response in horses. Its complete characterization, profiling, and identification is required. So for cobra, which is this species is strain is Naja, and we test phospholipase A2, neurotoxin, and cytotoxins. For Russell viper and soy scale viper, we evaluate phospholipase A2 and snake venom proteases. And this is actually the important steps that require for the processing of the venom. This is the 6th step: quarantine of the venom, milking of the venom, and and its quality control analysis. And after and when we look into the quality control analysis, we go for the physical examinations and the protein estimation, all the biochemical analysis, enzymatic and non-enzymatic analysis, and also we perform functional analysis. After having all data, we calculate the suboptimal dose of immunogen that play important role to elicit the immunogenic response in horses. These are the steps which I already described. So venom safety profile is very important. For that, we have to set the reference specification according to the WHO guideline to meet the quality parameters. So this is the protein estimation, 6 batches of the venom. This is the cumulative data of the 6 batches of the venom of the Pakistani venom from crate cobra, Russell viper, and soy scale viper. Additionally, utilizing the proteomics tools, we isolate the protein, we characterize the proteins, and we identify the proteins. This is the assay for the phospholipase A2. Without these characterization, we cannot use any venom as an immunogen. And we also perform protease tests. Functional test, if we look into the functional test, we perform hemorrhagic activity. I will not go in detail due to the time constraint. And we perform aggregation inhibition activity, we perform procoagulant activity, and these are the results that we acquired in our laboratory of all immunogen. This is the most important data, the median lethal dose, which gives you an idea what dose you select to elicit the immune response as immunogen. In our region, the crate is the most toxic venom, and SSV is the least toxic venom. We further perform various other toxicological analysis and teratogenic tests and different acute and subacute and chronic toxicological studies we perform incorporating various laboratory parameters. This is very important data. It actually gives the therapeutic window of the venom, the safe dose and the dose which start toxic response, which helps us to identify the immunogenic concentrations. Here we are showing the cobra. 1 to 8 milligram can be used in horses to immunize and elicit the immune response. In crate, only 0 to 5 milligram to 2 milligram, and same RSV and SSV 1 to 8 milligram venom can be used to elicit the immune response. We in one schedule of immunization, we have a primary immunization and 2 to 3 boosters, and every year we perform 4 to 5 immunization schedules, and we produce yearly 100,000 liter hyperimmune plasma to produce antivenom. actually, we have the 4 different venoms, and we divided horses in 4 different groups. And after immunization, we collect blood from them. And here is our veterinary doctors who are involved in immunization of the horses against the venom. And now, when the horse body acquire optimal titer of antibody, then the step is the blood collection and extraction of the plasma. And after plasma, we identify the titer, and getting the titer, we go for the different these are the different quality control testing of the plasma, because this is the raw material for the production of anti-snake venom. Okay, I will let me skip this slide. An important testing for the plasma in our laboratory is the protein estimation. That is very important, and we have to look into the equine, any bioburden. Is there any bacteria or any toxin and anything? So we have to evaluate in the laboratory techniques, and similarly we test the plasma if there, because we are acquiring plasma from animal body, so there should not be any bacteria, any endotoxin, and any virus in the product. That also confirmed, and when all are negative, then the plasma is ready to provide for the downstream processing. Before giving to the and submitting the batch for the downstream processing, these are the study that we perform for the titer utilizing ELISA. And this is the neutralization assay data, which is one of the our specification. This is our batch disposal record, batch dispatch record, and then we enter into downstream processing. Downstream processing is carried out in Dawn Institute of Life Sciences, which are licensed drug manufacturing facility in the Dawn University of Health Sciences. And at this institute, these are the steps for the purification of the IgG. We actually, based on the capillaric acid, we perform this portion utilizing the capillaric acid and filtrate and tangential differential flow, and ultimately we go for different quality control testing. This is the few glimpses of our commercial facility. This is the our aseptic solution preparation area. This is the aseptic filling area, and this is the lyophilizer Teflon. This is the country best and the biggest biotech facility in the Dawn University of Health Sciences. And this is our cap sealing machine and labeling machine. And these are the important quality control parameters that we have to perform when the finished product acquired. And this is the anti-snake venom potency. So the potency against the batch claim of the venom. And all 3 monovalent we acquired 95 or 93 to 100% potency and safety against the batch claim of the venom. And our goal to improve our production to 500,000 vials. Currently we are producing 50,000 anti-snake venom vials. And I would like to conclude. This is my team, and these are the veterinary doctors, and our proteomics, Dr. Osman is sitting here, and all the quality assurance, production manager, and the researchers are sitting here also. They are contributing a lot. Thank you very much.
This session is open for the questions. We could take 2 questions, I think.
Rawalpindi for past about 40 or so years. Is your snake venom any different from theirs? That is one. And the second thing is that They are producing it and they have been distributing it throughout Pakistan. Obviously, there is a capacity limitation. And perhaps your case was really that not enough being produced. I don't think that facility is closed. They've had extensive experience, and they are into these big four. So any difference? And please also note that there is so much of importance which is attached to GMP certification and continued monitoring of your production facilities for the efficacy and safety. Thank you.
Regarding NIH, yes. The difference, there is a slight difference in the method. We are utilizing the capillary acid method. The NIH is using little bit different protocols in the downstream processing. And the second point, yes, they were involved since long, but their production is not continuous. Thereby, during all that time, Pakistan is importing antivenom Bharasira. Even I cannot see in the market any product from NIH, but I can easily get the Bharasira. This means that their production is interrupted and not sufficient to fulfill the Pakistan's requirement. At the moment, to treat the snake bite victims, we required 300 to 500 vials, and their production facility at the moment not able to do that. Thereby, DHS has taken the initiatives, because the most of the anti-snake venom cases are present in Sindh, especially in the Indus Delta region and the interior Sindh, Thar, Badin, Mithi. These are the regions. So this is the facility has been developed with the support of Sindh government through PC1, and DRAP is also really helping us to develop a second institute in Pakistan who can contribute in development of antivenom.
Dr. Saab, this is a great effort because Sindh has a real problem, the snake bite, and at SIUT, in those very months that you have mentioned, we continue to see patients, children, males and females with acute renal failure due to snake bite. The question and I'm asking is that you are doing an excellent job making these but are there facilities in Mithi, in Tharparkar, where this venom is available? What are the storage conditions?
Okay. Yes.
Because you are doing a great job, but is it going to the place where it's needed?
Yes. We have in Dow University Venom Bank, and we collect almost 500 to 40 ml of each venom in our system, and we routinely characterize. A dedicated team is there who work with the venom only. Farah, Dr. Farah is sitting here just in front of you. This lady is actually the venom expert. Dr. Usman is sitting here. He is a person of proteomics. The veterinary doctors of the team at the moment not here with me. So these people, and the Sarish is sitting behind, and that she's a executive of and the manager of quality assurance. And just after you, the quality control manager is sitting. So they are managing the storage in the facility for venom. Not only venom, but the plasma. The production manager, Dr. Shawar, is not at the moment. We have more than 8 PhDs and more than 7 MPhil scholars. And the important thing is that in vivo testing of the venom, the Shazmeen Aslam is sitting here, and the staff scientist, Dr. Midhat, is sitting here. They are maintaining all that quality control practice in the institute. Thank you. Thank you. Thank you very much.
Thank you so much, Dr. Talat, for a wonderful talk. Our next invited speaker is Professor Rabia Hussain, and it's really my honor to be able to introduce her. Professor Rabia Hussain is an endowed professor and former founding head of the Department of Microbiology, the Khan University, and a distinguished national professor of HEC. Professor Hussain obtained her fellowship in clinical immunology at Johns Hopkins University and later held a faculty position there. At Johns Hopkins, her research focused on characterizing ragweed pollen allergy. At NIH, she identified the fundamental role of IgG4 antibodies in blocking allergies and received the Bersaniello Award in 1981 from the American Institute of Nuclear Medicine, USA, for the best paper on the development of a highly sensitive qualitative assay for detecting IgE antibodies to parasite allergens. She has to her credit 175 full-length papers in international peer-reviewed journals with high impact factor. She has multiple prizes to her credit. She has received a Lifetime Achievement Award from Pakistan Societies of Pathology. She has received Tamgha-e-Imtiaz, Sitara-e-Imtiaz by Government of Pakistan. She is an elected fellow of Royal College of Pathologists, Pakistan, Islamic Academy of Sciences, the World Academy of Sciences, where she's also served as a council member and vice president of the Central and South Asia. She was elected Life Patron of Pakistan Society of Immunology. Professor Rabia Hussain.
Thank you, Kiran, for that long introduction. What I'm going to talk about today really has a very wide impact or an umbrella of immunology. You'll see that it has impact on every question you ask, specificity of antibody. And so let's get started. Thank you. Okay, slide, pointer. ये dead है नहीं आ रहा. Thank you. Okay, so let's get started very quickly. And what I'm going to talk about is really recognition of conserved and specific epitopes, which has broad implications in both disease progression, autoimmunity, and protection. So let's see how all of these come together. Can I have, I have to do it. So there are three issues that I would like to address. Role of antibody in mycobacterial diseases, where we'll talk about how it relates to disease severity, disease protection, the specificity of antibody responses. And I've used this mycobacterial disease as a model. It can apply to any diseases, so it will be relevant to all diseases that we study. And then, is there a connection between mycobacterial induced or BCG-induced immunity and cross-protection for other unrelated infection and recently introduced COVID-19? So these are the three main issues that I'll focus on. Very quickly, leprosy, actually Tore Goodall said that, and I will end it with that leprosy was a God's gift to immunologists. Why? Because it was the first disease in which you could make a clear-cut diagnosis in terms of bacterial load, and the clinical spectrum related to how much bacteria you had in the body. And that could be ascertained by biopsy. So that allowed an immunologist to really dissect the immune response and relate it to the bacterial load. And in 1970, for the first time, Tore Goodall was the one who showed that, I'm finding it very difficult without a pointer, but we'll have to do it without it. You'll have to follow the slides. So what you show up here, what he showed was that T-cell mediated response was indirectly related to bacterial load. So as the bacterial load increased, the cell-mediated responses decrease. And therefore, all attention was then paid to T-cell responses, cellular responses, and antibody was forgotten. The role of antibody was totally forgotten. The only thing that was shown was as the bacterial load increases, the antibody response increases. So it has no role in protection. But what about other roles of antibody? Um, so when I joined Aga Khan, I switched from parasitic diseases to mycobacterial diseases. And we had a very nice control program being run by Dr. Fowler, which I think every one of you must have heard the name of, the most successful program. I went to her and I said, I want to study immune responses in leprosy. And the first question she asked was, how is it going to help my patients? So I had to rethink about my contributions and where I should start and what I should do. And what I did was I started off with looking at antibody responses, which was my specialty. And you can say I did the tia pancha of antibody response in every aspect. So the first thing we did was, how do antibodies relate? But not only antibodies relate, we looked at all the subtypes of IgG. We looked at IgM, and we said, which one? Do all of them increase, or are there certain specific antibodies that increase? And the answer was yes, there were differential responses, and the antibody that was associated with bacterial load most was IgG1 antibody. For IgG3 also had a role, but the reason we concentrated on IgG1 is because it plays an opsonic role in uptake of bacteria into the macrophages. So that started us on as to what's happening, In the absence of T cell, you have so much antibodies. What are these antibodies directed to if there is no T cell response? You know that T cell responses are activated to processed antigen, and innate responses, non-T cell responses are activated to unprocessed antigen. So that was the most important thing, and that we, kya hua bhai? So basically, just to make the point of how good the relationship was between IgG1 antibodies and bacterial load. It was incredible. We had a lot of patients, and all of the bacterial load was done in London School, and we did all of the antibody isotypes. And this led us to ask the question of what are these antibodies recognizing, in the absence of T cell responses. That was the key question. What I want to say is, yes, we contributed in terms of role of antibody in several different areas which had been neglected almost for 20 years. And we had several publications, the implications of diagnostic markers in leprosy, immunopathogenesis, immunoregulation. I'm not going to talk about this because this is all published data. I'm going to just make main points that we did. So coming to this, in the 1980s, it was not very easy. The technology has really moved very fast, and we moved with it. So what are these subclasses recognizing in the absence of T cell responses? That was my question. And I'll try not to give the detailed things because these are published, but the main points that came out was that IgG antibody binding, we used SDS electrophoresis, immunoblots to identify what are the antigens that are being recognized. And it was very interesting because what we found was that IgG1 antibody recognized mostly the region between 25 to 40 kDa. IgG2 was also restricted, a little bit more restricted, 25 to 36, again the same region. IgG3 antibodies interestingly recognized 5 to 7 kDa, and this has been recognized to be a T cell epitope. This is the GRO-ES, what we call the ESAT6 antigen. And at that time it was not known, it was much later recognized. And IgG4 antibodies were binding to a totally separate region, again more restricted. I can't even see to a 28 kg region, so it was even more restricted. So it was the IgG1 that attracted us most in terms of what its role was and what are these antigens which are being recognized in the absence of T cell. When we looked at this region a little bit more carefully, we found out that this was where the most of the heat stress proteins were present. What are heat shock proteins or stress proteins? In 1988, Doug Young had shown that these are the major targets of immune response, both T and B cell. So it made sense. And then I had a PhD student who went on to characterize this even more. She said, okay, what is this HSP25 to 36 residue? And what was, when it was defined further, it was found out that it had an integral protein which is about 90 amino acids long, or 10 kDa protein. And it was bound, it had a very long tail. And that's why originally when it was thought it was at 25 kD, it was really that 100 amino acid protein that was the dominant target of immune response. So we went on to crystallize. This is the crystal structure of this. And it forms the central portion of a core. And this is the finally the crystal structure of sound. So we said, okay, let's look at this protein. It seems to be really the most dominant protein. And what we found was, let's first talk about heat shock protein a little bit. What are they? They are expressed under stress, under heat, physiological stress. They are chaperones, and they are very important in folding and unfolding of proteins. So you see it's a common function which is shared across all species. Highly conserved for that reason, because if they are not conserved, they would not be able to do the function. And they represent actually a group of broadly recognized antigens which are recognized by the innate arm of the immune system, what are called as PAMs, pattern associated recognition, molecular pattern, right, thank you. So it belongs to that category. So if it is such a conserved protein, of course the next question is it the conserved epitopes or is it the specific epitopes in this that is being recognized? Okay, so BCG stress proteins, 10K, LPS, BCG, which I have encircled, are the ones that produce, we have to also look at cellular activation and cytokine responses, are mainly IL-4, IL-17. You know, IL-4 is a surrogate marker for B cell activation. And this BCG 10K is the one that produces, which really induces it most. And then we come to interferon gamma, IL-10, which are surrogate markers for T cell activation, but also NK cells, gamma delta T cells. So you see there is a lot of redundancy in response. And what people thought was when they were looking at interferon gamma and IL-10, they thought they were looking at stimulation of T cells. But these cytokines also come from innate cells. But innate cells don't have memory. So how do they get activated in vitro? And we'll address that issue. So we come to next. We next analyzed 10K protein, and we picked up these cytokines to look at activation. Is LTT really a T cell response or not? Is interferon gamma, IL-10 coming from innate cells or adaptive arm of the immune response? And the B cell, which is a surrogate IL-4 is a surrogate marker. Why are they activated? Is it because of the IL-4 from the innate arm and not the adaptive arm? And that will all depend on what is being recognized. So what we did was, at that time, ML10K, this 10K stress protein had been purified both from MTB and mLepre. And we addressed the question, is there any concordant recognition? Are both are recognized? Or is mLepre recognized more in leprosy than in TB? And vice versa. So we looked at that issue. And what you see up here is we looked at it in lepromatous leprosy, which is the severe form of the disease, tuberculoid leprosy, which is the localized form of the disease. So disease is in its track, pulmonary tuberculosis, TB, and we have this endemic controls, healthy endemic controls, which represent BCG vaccinated, MTB exposed group. And to make it easier, I made a table of it. And what you see up here is that in the endemic controls, you see the highest response to both antigens. But it's selective, IL-10, IgG1, and IgG4. When you look in patients, what you find is the red is the ML, the black is the MT. And what you find is that in tuberculoid leprosy, yes, you do get cytokine responses associated with T cells. You get it in endemic control, but you don't get it in leprosy patients. So the T cell response is absent, but antibody responses are in all the groups. So we went to the next question, and that is, what is the epitope in these two that are being recognized? What I have shown in red are the conserved epitopes. What I have shown in white are the specific epitopes for either leprosy or TB. And then we looked at responses. Thank you. We looked at responses to these epitopes. Now, this is a very busy slide. I don't expect you to know what's going on at every level. But these are 15 mer peptides, 5 amino acid overlapping, which span the entire 10K. And we looked at each peptide. And in the red are the responses for interferon gamma, IL-10, and in the blue are the responses for antibodies. Okay? And you see at the tail end, what is being recognized are conserved epitopes plus one epitope in the middle, which is showing the most response. So I summarize this again to make it easier for you. And what I show up here is conserved epitopes, leprosy patients, pulmonary patient, endemic control, all recognized. And the epitope that is recognized is, actually it's 41 to 50 mer, but this is what is the conserved epitope. And the tail end is being recognized by G2, G4. So even the conserved epitopes are being selectively recognized by antibody subtypes, an important thing. But it's all conserved epitopes. While interferon gamma, IL-10 are recognizing more specific epitopes. But is it really coming from T cells? No, because there is no T cell activity in leprosy disease. But in endemic control, yes, probably it's coming from T cells. And is this really a sign of, is it really a protective response or what? We'll look at that. So, one other very silly thing I did was to look at completely different non-related pathogen responses in leprosy sera. And what I found was there was very high titers of T. gondii antibodies. Even people from London challenged me. They said, this doesn't make sense. But I had I'd done all the experiments to show that this is really, antibodies are recognizing T. gondii in a specific fashion by inhibiting it with T. gondii antigen. And I never understood the significance until trained immunity came into context. So revisiting the evolution of immune responses, what we've shown is that as the disease progresses in leprosy, you get you get more and more suppression of T cell responses, so adaptive immunity is shut off, and probably only innate immunity is present. So as the disease progresses from the tuberculoid, T cell responses are suppressed, and at the end of the totem pole, disseminated leprosy, you only have any immune response that you are seeing is probably innate responses. And a huge amount of antibody to conserve epitopes, not to specific epitopes. But, you know, can they contribute to immunity? Endemic controls, yes, there is a lot of evidence that only 1 in 1,000 exposed individual gets leprosy, only 1 in 100 exposed individual gets TB. BCG does not protect against pulmonary disease, but it does protect against extrapulmonary disease. And remember that innate immunity's main function is surveillance. That is, it stops the antibody it stops the pathogen in its track at the site of infection. So that made sense, and that was the only reason why BCG was continued by WHO. There were all other things. So basically, are these responses really, you know, of any importance to us if they don't have memory? But then in 2018, there was a paper published by Nitya's group showing that yes, there is memory in these responses, in innate responses, and this is because of reprogramming of some of the cells. I'm not going to go into the detail, but that was the basic message. And that made a lot of sense to us. So the third question that I'm sorry, I'm going to run a little bit over time. The third question was, what is the relationship between mycobacterial diseases and immunity or cross protection? Because these conserved epitopes are across the pathogen. So do they play any role in cross protection? And very quickly, yes, BCG induced mortality in childhood, reduced childhood mortality because it was continued, thank goodness. In control programs, several control programs showed this, not only that it reduced the severity of the disease, but it also protected against other infection. Childhood mortality, viral infections, respiratory tract infections. And then along came COVID. The good side of immune recognition of conserved epitopes. Okay? Much less mortality and morbidity associated to COVID-19 in countries that had ongoing BCG vaccination programs. That was the good side of this recognition, and now we understand why. BCG vaccination induced protection, and everyone started looking at protection to COVID. And of course, now there are several publications that have shown hidden in plain sight effects of BCG vaccination on COVID-19. And COVID-19, RBD is considered to be the most protective molecule, you know, inducing neutralizing antibodies. But when you look at the structure, again, there are conserved epitopes and there are specific epitopes. And recently there was a paper in 22 from Atlanta where they showed that 30% of the antibody was they could absorb out RBD neutralizing activity, and 30% of the antibody was remaining to what? Probably to conserve epitopes. Okay, so a lot of information that was previously acquired is now being looked at in new light. Okay, so maybe I should now very quickly stop because this is already covered. The bad side of immune recognition. Conserve epitopes, autoimmune diseases, long COVID role. And that's why I keep asking the question, what are the specific epitopes that are being recognized, not just molecules? What role do they play? Why is there overproduction of antibodies, and which conserve epitopes? Very important questions. Okay, so we'll leave it for next time. Thank you.
Thank you for a wonderful talk, Dr. Rabia. Probably we can take one question, if there is any, or a comment.
You showed that there was increase in interferon gamma, and there was increase in interleukin 12 as well, and at the same time interleukin 10, which interleukin 10 as well, which is really the first two are associated with Th1 and the second one is with Th2. So that's kind of contradiction there. See, we know that there is, yes.
All the ingredients of the adaptive arm, and we thought not have memory.
Yes. What I Yes. What I wanted to say is that when looking at cytokines, obviously their function, there is a lot of overlap and there is a lot of redundancy. So that has to be kept in mind. And in studying leprosy, there is this great dichotomy. On two extremes, you have tuberculide, good cellular immunity. On the other extreme, tuberculide, yes, tuberculide has good cellular immune response, and lepromatous would be less of cellular immunity and lots of antibodies. And there is this variation of immune response in between all of that. So if we could have, yes, two different groups of patients and then looked at these responses again, that would be one thing. And looking at toxoplasma antibodies, if you get immunosuppressed in HIV, you get toxo-infection. So similarly,
Both are different mechanisms. Anyway, the thing is these studies were done 20 years ago, and a lot of questions now are arising because of the new perspective on responses. And I think we will leave it up to this young group to pick up the questions and ask those questions, relevant questions. मेरी आवाज इतनी बड़ी है, सब सुन रहे हैं, है ना? पीछे तक आ रही है ना? I'm used to talking. अच्छा, उसमें नहीं आ रही है, लेकिन इसमें आ रही है ज़्यादा. अच्छा, ठीक है. लेकिन ये कि I have taken more than my share of time, and I think we should move on, and we should have a wonderful discussion sometimes just on talks later on. Thank you.
Thank you so much, Dr. Rabia.
So let's move to the next few set of talks. The first talk, oral presentation, is from Dr. Shakir Hussain. He's from Indus Hospital and Health Network, Karachi, Pakistan. He's going to talk about evaluating antibody responses to booster doses of COVID-19 vaccine using chemiluminescence microparticle assay and pseudotype lentivirus antibody neutralization assay in Pakistani population. Shakir.
Bismillahirrahmanirrahim. Assalamu alaikum. My name is Shakir Hussain and I am from Indus Hospital, Karachi. Today, my research paper topic is evaluation antibody response to booster dose of COVID-19 vaccine using chemiluminescence microparticle immunoassay and pseudotype lentivirus antibody neutralization assay in Pakistani population. So I don't know. So why booster dose? In many research and many publications, we see that there is a after vaccination there is a decrease in antibody level after some period of time. So in our study, 43 participants are taken, and all these participants are of booster shots, having booster shots. So keeping this in mind that booster dose can increase the antibody level, we design a project, a study in which we evaluate the anti-SARS-CoV-2 IgG level after booster dose of Pfizer vaccine followed by the 2 dose of Sinopharm. Comparison of two methods, one is chemiluminescence assay and the other one is antibody neutralization assay, to diagnose the anti-SARS-CoV-2 IgG level. So we know that this is a list of vaccines which are available in for COVID-19 vaccination. So most of in our population, most of the vaccine we go for is the first is Pfizer vaccine, which are mostly used in booster dose. So this is the whole summary of our study, that we take 2 samples. After centrifugation, serum is collected, and 1 sample is for antibody neutralization assay, and 1 sample is chemiluminescence assay, and after that we analyze our data. So this is the basic principle, that how virus enter into the cells, they bind with receptor. So what is the difference between lentiviruses and the normal viruses? So here you see that the spike proteins, and in normal viruses, the spike proteins and genomic materials, and they have mRNA, and there are some proteins as well. But in lentiviruses, the structure of the virus is same, but the difference is the genomic material. The plasmid can replace the genomic material of normal viruses. So this lentivirus we made in Indus Hospital. This is the same lentivirus which was made in Wuhan during COVID pandemic. So some of the important genes are retained, which are responsible for the packaging of viruses, and some are deleted, which are responsible for the pathogenicity and for the packaging of lentivirus. So this is the process in which plasmids are transfected with 293 cells, and after transfection and incubation of 42 to 70 hours of incubation, the supernatant is collected, and the lentiviruses are then transfected with another special type of cells, which are AS2 293 cells. When this lentivirus infect these cells, the cells turns into fluorescence, and if not infected, the cell remains colorless. So this is the basic principle of antibody neutralization assay. If a patient contains serum, if a patient's serum contains antibodies, the antibodies will bind with the pseudotype lentiviruses, and they will not enter into the cell. And if there is no antibody present in the patient's serum, the lentivirus can bind with the cells in hospital setting, and the cells turn into green. So these are our results. In first, you can see that there is antibodies. That's why the cell not turn into fluorescence. And in other slides, you can see that there is no antibody in the patient's serum. That's why they all are fluorescence. So we use luminescence assay for the optimization of our test. So these are the result of chemiluminescence assay, in which you can see that in y-axis we take log 10 of the antibiotic titer, and you can see that 2.9 is the titer, is the mean of the mean of the antibiotic titer before the booster dose, and 3.87 is the titer after the booster dose. So the value is significance, and the p-value for this is 0.001. And for antibody neutralization assay, the mean was 2.45 before booster dose, and after booster dose it is 2.8. The value is not significant, but it is however 0.0631. But there is an increase in antibiotic titer. So limitation of this study is that actually the first is the low sample size, and the other is at the time of study no calibrators are available. So now WHO have developed some calibrator for pseudotype lentivirus assay. In conclusion, this study support the administration of booster dose and the increase of antibody titer with pseudotype lentivirus assay, and SCMI is observed. So this is our team. In our team, Dr. Javeri Jas and Fozia Nasir, and these are the references. Thank you so much. If you have any questions, please go ahead and ask.
Thank you so much. Do we have any questions?
I'm sorry, I think I missed it out. What was the time period of checking for after the booster dose?
The time period varies, but we actually the sample size is very low. We take only 43 booster dose patients, And after that, we categorize them into different categories according to the time period from booster dose to time of collection of samples. But we not see any difference of antibiotic titer between them.
So the time...
the time from booster dose and from the collection of samples.
Did you have a standard time?
12 months. No, we have no standard time.
Oh, because it's important
Yeah.
when you do the levels after the booster dose.
Yeah, because we in 43 samples we not see any difference between the time periods from booster dose to you can see that time period. एक्चुअली हम चाह रहे थे कि जो टाइम लिमिट है ना वो हम रखें, लेकिन हमने रखा था 12 मंथ तक का रखा था, लेकिन 12 मंथ में हमने ये नहीं देखा कि 5 मंथ में भी लिया गया था, 7 मंथ में भी लिया गया था और 12 मंथ में, लेकिन जो एंटीबॉडी टाइटर था बूस्टर डोज के बाद उसमें हमने कोई डिफरेंस नहीं देखा, क्योंकि मे बी द नंबर ऑफ सैंपल साइज जो कम था, इस वजह से अगर साइज बढ़ा दिया जाए तो मे बी दिस इज पॉसिबल.
5, 7 and 12 months, so that's the answer.
Yeah.
Thank you. The thing is, age and gender has an effect on antibody responses.
Yes.
Did you look at that issue, this
Yes,
variable in your...
Yes, ma'am. Actually, I will tell that if the sample size was increased in our study, we will be able to rule out that it will affect. Yes, there are many variables. We did this in our study and we found nothing between the titer of antibodies, increase in titer of antibodies. But I think, yes, actually we did a try, a new, we developed a new laboratory developed test to, to just calculate the titer of antibodies in our lab setting. So it's a LOD test.
You said you use chemiluminescence assay micro. So
Yeah.
which assay did you use? Like it must be from a commercial supplier. So company कौन सी थी ऐसे, and it was detecting anti-N antibodies थी, S antibodies थी, RBD, S का RBD था, कौन सा ऐसे था ये?
Anti-IgG थी और एबर्ट ने हमें प्रोवाइड किए थे किट इन FOC.
नहीं वो तो है, लेकिन N एंटीजन देखा था, S एंटीजन देखा था, दोनों बनाता ना एबर्ट तो.
IgG N एंटीजन.
N एंटीजन था, तो लाइक देन वी आर कंपेयरिंग एप्पल्स विद ऑरेंजेस, क्योंकि वो N एंटीबॉडी तो वो सर्विलेंस के लिए यूज़ होती है, जो न्यूट्रलाइजेशन के लिए देखते हैं ना वो, लाइक इन योर अदर ऐसे आपने जो एंटीबॉडी देखी वो इट.
सॉरी आई इंटरप्ट यू. देखिए हमने जो देखा था जब हमने एंटीवायरस बनाया था, ठीक है, उसका जो स्पाइक रखा था वो S प्रोटीन का था.
हाँ वो तो
ठीक है, तो उसके अगेंस्ट हमने जो स्पेसिफिक एंटीबॉडीज थे वो हमने डिटेक्ट किए थे, ऐसे ही देखा था.
लेकिन देखो ना फाइजर में व्हेन यू गिव, कौन सा एंटीजन देते हैं फाइजर में यू गिव एस एंटीजन, ठीक है. जो mRNA वैक्सीन है, दैट mRNA वैक्सीन कैरीज इंफॉर्मेशन टू प्रोड्यूस एस एंटीजन. तो आपकी जो सूडो न्यूट्रलाइजेशन ऐसे है गोना डिटेक्ट एंटाई RBD एस एंटीबॉडीज. जो आपकी कैम्युलिमिसेंस है उसमें पता नहीं किस तरह बूस्ट हो गया, वो इन्फेक्शन से हुआ होगा, उसमें तो एन एंटीबॉडी देते हैं, वो वैक्सीन में है ही नहीं. No, like, but he said he did anti-N chemiluminescent. अच्छा वो गलत कहा आपने, वो S किया होगा भाई. हाँ ये ठीक है. हाँ N नहीं कर सकते, N शुरू में आया था फिर S आ गया था. ठीक है. Thank you so much.
Thank you, Shakir.
Thank you.
So we'll next invite Kumail Ahmed from Khan University Hospital, and he'll talk about BCG-trained immunity against cross-reactive COVID antibodies in healthy BCG-vaccinated donors in TB endemic area.
Welcome everyone. My name is Kumail Ahmed from Khan University, and the topic of my presentation is the trained immunity against cross-reactive COVID-19 antibodies in healthy BCG vaccinated donors in TB endemic area. I do not have any disclosure statement. Many studies have been conducted for the efficacy of BCG vaccination. Pakistan has 90% coverage of BCG at birth, and it provides protection against the disseminated TB or the extrapulmonary TB, as Dr. Rabia has already mentioned. and it also provide a non-specific immunity, and it has been used in the treatment of bladder cancers and the melanoma cancer. In terms of this non-specific immunity, a term trained immunity was adopted for these additional effects of BCG in non-infectious cases. So the protection of BCG against COVID-19 is also debatable with conflicting reports. So the trained immunity that Dr. Rabia has already discussed about it. Once the BCG vaccine is administered, the macrophages goes under epigenetic modification, as a result of which the macrophages activated. And when the infection comes, these activated macrophages secretes cytokines of the trained immunity, like the interferon gamma, TNF, IL-10 and IL-17. So this heightened response basically is the trained response. The hypothesis of this study is to BCG provide non-specific immunity against non-infectious disease and exhibit additional benefits by activating the trained immunity network. So the objective of our study is to understand the trained immunity of BCG using cytokine profiling, and also to observe the cross-reactivity of BCG against COVID-19 antibodies. So here's the methodology. Blood were collected from the recruited individuals, and then they will stimulated with the antigens like the BCG and the LPS. After their incubation time period, after 12 and 48 hours, the supernatants were collected, and the cytokine assessment was done by using the Luminix 200C system. While for the assessment of anti-IgG-RBD, we performed the ELISA. For that purpose, the ELISA plates were coated with RBT antigen with 2 microgram per ml. And for the calibration curve, we use the positive pool and the negative pool. The positive pool, basically the COVID-19 patient which having the OD value greater than 1, and the negative pool having the OD value less than 3. So basically these positive pool were titered twofold, as a result of which, and this will read against the OD values, as a result of which we get the high response and the lower response, that titration time points, as a result of which we get the equation which help in the calculation of the concentration of unknown samples. So we recruited 20 individuals with the male and female ratio 10 equal numbers. Our study protocol was approved by the EQERC, and the inclusion criteria includes a healthy adolescents with age and gender match, while the exclusion criteria consists of the post-treatment of the TB, history of any autoimmune disease. Then we compare the then we compare the basically the BCG effect, the BCG scar present in individuals and versus the non-BCG scar. As you can see, there is no significant difference found between the scar positive and the scar negative individual. This was not surprising, basically because the T cell response which is given by the BCG vaccine has been waned with the passage of time, and also the BCG scars and the MT are not the true reflective of BCG vaccination. Then we did the comparison between the cytokine in its spontaneous and BCG stimulated whole blood assay. As you can see, there is a significant difference found between the spontaneous and the BCG stimulated response, which indicating that the recall responses of the BCG, and it is included in all the, it is shown in all the cytokines of the trained immunity. And then we compare the results of BCG and the LPS antigen, and we found that IL-4 and IL-2 give the significant difference, which is suggesting that these cytokines are from the macrophages. And then we did the assessment of the cross-reactive antibodies for the COVID-19 receptor binding domain. As you can see, there is a significant difference found between the plasma sample. These plasma samples were basically from the pre-pandemic time point, and we run this through the ELISA, and you can see the significant difference between the plasma and the plasma having the BCG stimulation. So there is certain type of conserved epitopes which might be hinder or supporting the BCG to hinder the COVID-19 infection. So in conclusion, prior BCG vaccination has no profound effect on cytokine profiling, and the BCG stimulation shows high level in IL-2 and IL-4 compared to LPS, which may be the recall response against BCG. And the presence of COVID-19 antibody in pre-pandemic samples shows the cross-reactive epitope of the BCG against COVID-19. So I'm thankful to Dr. Abia Hussain for the optimization of this essay, Dr. Najiha and Khan University for funding this research, and BCG Japan for the provision of BCG, and Infectious Disease Research Lab for the logistic and partial funding to support the cytokine assay. Thank you.
Thank you, Kamal. Do we have any questions?
Thank you so much, Kamal. So let's move to our last presentation. It's from Fahim Shahzad, University of Health Sciences, Lahore, Pakistan. He's going to talk about decreased classical monocytes, CD163 expression in TB patients, an indicator of drug resistance. Fahim Shahzad.
Bismillahirrahmanirrahim. Thank you so much and good afternoon everyone. My name is Dr. Fahim Shahzad. I am from University of Health Sciences, Lahore, and today I am here to present my research work, that is decreased classical monocytes and CD163 expression in TB patients, an indicator of drug resistance. As you all know, tuberculosis is a major public health problem worldwide. It is an infectious disease. These are the stats from WHO, recent stats. The encounter between MTB and the various components of immune system induce a complicated series of host responses and counter responses by the microbe. The host cellular immune responses determine whether an infection is arrested as a latent TB or progresses to active TB. Peripheral blood monocytes represent highly efficient effector cells of the innate immunity against TB. They are subdivided into different subpopulations by the expression level of membrane antigens like CD14 and CD16. These are the main three types of monocyte subsets: classical monocytes, intermediate monocytes, and non-classical monocytes. CD163, a hemoglobin scavenger receptor expressed exclusively on active subsets of monocytes that plays a major role in immunological homeostasis. CD163 positive classical monocytes has a great potential to be used as a biomarker for disease progression and monitoring of treatment efficacy. Increasing prevalence of drug resistance has become biggest threat to global TB control. Impaired immune response by mononuclear leukocytes may also contribute to the development of drug resistance. Studies have been conducted to elaborate relationship between microbacterial infection and host immune responses that affect progression of the TB. The effect of host pathogen relationship on drug resistance remains largely unexplored and is therefore a key field of future research. Therefore, the present study was designed to evaluate the biomarkers and subsets of monocytes in pulmonary TB with and without drug resistance. These are the objectives of my study. The project was approved by the Ethical Review Committee of University of Health Sciences, Lahore. A total of 240 subjects were recruited for this study, and they were subdivided into 3 groups of 80 individuals of each. Group 1 had 80 healthy individuals, Group 2 had 80 diagnosed TB patients without rifampicin resistance, and Group 3 had 80 diagnosed patients with rifampicin resistance. Cells were analyzed by flow cytometry using FACS caliber, and these antibodies were used for the flow cytometry. Isotype antibodies were used as a background staining controls. This figure shows the gating strategy for the evaluation of different subsets of monocytes and CD163 classical monocytes. Serum level of CD163 was determined by using commercially available ELISA kits, and this is how we analyze the data. This slide shows the baseline characteristics of study participants. In this picture, you can see the comparison between classical monocytes among 3 groups, and you can see here that in third group, that is RAF-resistant TB group, you can see here classical monocytes were statistically significantly decreased in this group. This picture shows intermediate monocytes, and in this picture we can see that intermediate monocytes were decreased in patients without RAF-resistant TB. And this picture shows non-classical monocytes. In this picture we can see there was a difference in the percentage of non-classical monocytes between healthy controls and all other 2 TB groups. In this picture we can see the expression level of CD163 on classical monocytes and serum level of CD163, and we can see here that serum level of CD163 was decreased in TB patients as compared to healthy controls. Similarly, expression level of CD163 was decreased in RAF-resistant TB only. So in the current study, a statistically significant decrease in percentage of classical monocytes was detected in patients with RAF-resistant TB as compared to healthy controls and patients without RAF-resistant TB. To the best of our knowledge, this is the first report showing a decreased percentage of classical monocytes in patients in RAF-resistant TB. Reduction of classical monocytes may have contributed to the development of drug resistance by prolonged exposure of M. tuberculosis to drug without sufficient immune response. However, functional studies are needed to explore the possible mechanism involving this phenomena. Similarly, the expression of CD163 on classical and intermediate monocytes was also significantly decreased in patients with RAF-resistant TB relative to healthy controls and patients without RAF-resistant TB. A decreased serum level of CD163 was also observed in patients with and without RAF-resistant TB relative to healthy controls. Decreased 163 classical monocytes in patients with drug-resistant M. tuberculosis patients possible may have is an indicator of that it may be involved in for the development of drug resistance in those patients. However, further studies are needed to investigate the underlying mechanism. In summary, the current study described changes in monocyte subsets. TB patient decreased frequency of classical monocytes was observed in RAF-resistant TB. Decreased frequency of CD163 classical monocytes and CD163 serum level was also observed in RAF-resistant TB patients. Taken together, the findings of the current study and observation from previous study, it can be concluded that decreased frequency of monocyte subsets and CD163 expression could be an indicator of M. tuberculosis drug-resistant TB patients. Furthermore, the estimation of CD163 level in TB patients could be a useful tool for prognosis and therapeutic monitoring. I would like to acknowledge University of Health Sciences Lahore for providing funds and also to acknowledge all the authors who contributed in this study. With this, thank you so much.
Thank you, Fahim. Do we have questions for this session? Yes, sure.
It was a very good talk, excellent talk. And there is alternatively activated macrophages as well. Did you look at that? Because alternatively activated macrophages are the ones that downregulate the classically activated macrophages.
Yeah, ma'am. It could be another, you can say, way to go through, but we have some limitations regarding funds, and that's why we could not go through. Yeah, but we are planning to go for in next step as functional studies, and we hope to add that in this, inshallah. Thank you so much.
Do we have more questions or comments? All right, so with that, we are done with our session, and I would like to ask Dr. Junaid to close the session.
Thank you so much, Kiran. So we started with two wonderful presentations from two of our invited speakers. We had first presentation from Dr. Talat Rumi, who told us about the importance of locally manufactured multivalent anti-sera against the big fours, like the locally most relevant snake bite venomous victims. And I think somebody said from the audience that NIH is also producing anti-sera, but then we came to know that this is like the production from there is not enough to meet the requirements, especially in the southern parts of the country. And I hope like the Dow University and their facilities contributing towards saving lives of the people. And then we had this presentation from Dr. Rabia Hussain, talked about the humoral response against conserved antigen of leprosy, and extrapolated that how the work she done now, like people producing the similar kind of work for COVID-19, and the use of conserved antigen and the immunity against conserved antigen, and how it protects from COVID-19 like a severe infection. Then we had three presentations from young individuals. First from the Indus Hospital, from Shakir. Shakir talked about that how he used two assays to evaluate that the B cell response or the antibody production after the booster dose, and he compared two assays and showed that obviously I think the neutralization assay is the gold standard. Then we had a presentation from Kumail who showed that though the sample size was small, I think that was the reason that we didn't see BCG effects, which we earlier showed by Dr. Rabia and lots of other individuals. I think they published that BCG have some effects. Then the last presentation was from Fahim, who showed that there's a decreased classical monocyte and CD163 expression in TB patients. His sample size was also small. I'm not sure that we could use the assay at this point, but this is something we should look into and further test to see whether we could use this test to see antibiotic resistance or infection from antibiotic resistant Mycobacterium tuberculosis. So wonderful presentation. I'm also thankful to audience that they stayed and attended all the presentation. Thank you all. Thank you.
So with this, I would like to thank our judges and speakers, and I would like to request our judges to please come forward and present the shields to our respected speakers. Dr. Abia Perujin. So please, Dr. Abia, please, can you? Thank you. Dr. Junaid Iqbal. आपस में. Dr. Junaid Iqbal. Thank you, Dr. Junaid. Dr. Kiran. Dr. Kiran. So may I request judges please stay there and present the memento to our speakers. Judges please. Dr. Talat Rumi. Dr. Professor Rabia Hussain, please.
Dr. Aisha, the moderator, will going to receive the award.
So
So we'll be breaking for lunch right now, and for session 3, I would request you all to please come back by 2 so that we can start on time and proceed further. Thank you. to the post-transplant monitoring. So please take your seat. I would now like to invite Professor Mohammed Mubarak to chair this session. Professor Mohammed Mubarak is a professor and consultant hematologist at SIUT. He's heading the Department of Histopathology over there, and his keen interest lies in renal and transplant pathology. Dr. Mubarak, please take your seats. So you may invite the speaker now. You may invite the speaker.
Good afternoon. So we start this session, and I have the pleasure of inviting Dr. Salma Munir. She is currently working as an associate professor and consultant hematologist, heading the Thalassemia Prenatal Diagnosis Unit and Molecular Lab at the National Institute of Blood Diseases and Bone Marrow Transplantation. She received her FRC PAP from Edinburgh in 2021. She is a CPSP and approved supervisor for more than 5 years and is author of more than 25 research publications. She has special interest in molecular diagnosis and cytogenetics. She has the honor of being a student of late Dr. Tahir Shamsi, who himself is the pioneer of bone marrow transplantation in Pakistan. She is consultant clinical genetist and head of departments of molecular and flow cytometry at NIBD. Currently, she is actively running postgraduate training program for doctors and pharmacists. Dr. Saima, please.
Ji, thank you. Assalamu alaikum. Thank you for inviting me. Let's start my presentation. So, thank you, sir, for the great introduction of mine. And now I'll be sharing, I've made a very simple and basic presentation for basically the resident levels, so you can understand. And I'll share simple data about the NIBD and international data about the role of stem cell transplant in various inherited disorders, focusing mainly on the common ones. So as we know that Pakistan is a country where consanguineous marriages are a norm, and many rare disorders which are reported as rare in the literature are not so rare in our community. Since I'm a hematologist, so I'll be talking about more about the hematological disorders. These are the different disorders that we come across day by day in our OPDs and clinics. And top of the list is beta thalassemia major, followed by sickle cell disease, hemophilia and other bleeding disorders, marrow failure syndromes, immunodeficiencies, and last but not the least, storage disorders and others. So what's hematopoietic stem cell transplant? It is basically the transplantation of multipotent hematopoietic stem cells that are usually derived from the bone marrow or the peripheral stem cells, and rarely, not in Pakistan, but in the West and South Asian countries, they are derived from the umbilical cord. The common indications that we come across in hematology setup for which transplant is indicated, in this, the majority chunk is beta thalassemia major, and the second is aplastic anemia and its counterpart, the inherited Fanconi's anemia or other inherited marrow failure syndromes. Then comes the different immunodeficiency syndromes and leukemias, lymphomas, myelomas. They come under the heading of acquired disorders, but yes, they are also few of the indications that we go for bone marrow transplant. Storage disorders, commonly Gaucher's, mucopolysaccharidosis, HLH, and these are the disorders for which we have performed bone marrow transplant. If I share the data of transplant for benign disorders at our setup, we have performed around 800 transplants up till now, since the beginning, since 1995. Dr. Tahir performed first transplant in 1995, and since then till date, we have performed 800 transplants for benign disorders only, and the major chunk, around 70%, it was done for beta thalassemia, followed by aplastic or Fanconi in 25 to 28%, and then the other disorders that are rare disorders for about 4 to 5%. इसमें the most common two donor types we are doing. One is matched sibling donor or the matched HLA matched transplantation. It remains the best source because, but the disadvantage is that only 25 to 30% of the patients can go HLA matched sibling donor, or in some case we have done transplant that was full HLA matched, but not with the sibling, with other with the parents or with the uncles or aunts. The advantage is that there is rapid hematopoietic and immunological reconstitution, and there are lower chances of infections and GVHD. Haploidentical transplantation advantage is that it extends about the donor population or the donor pool. Around 50-60% patients who lack a suitably matched familial donor, they can get haplotransplant from a donor who is more than 50% matched with the patient. Initially, this kind of transplant generated poor outcomes due to high rates of GVHD, but now the outcomes have improved because of the different optimization of transplantation techniques. So I'll discuss about beta thalassemia, which is the most common indication and for which we have done the transplant. Our center and the other centers working in Pakistan have done the most cases on the benign is beta thalassemia. Carrier rate of this disease in our country, as reported, is 5 to 7%. It may be more, and around 5 to 9,000 children are born annually with thalassemia major. Exact numbers may be more because registries are not there, and the problem is that the only curative option for these patients is bone marrow transplant. Hematopoietic stem cell transplant in this disease consists of substituting the thalassemia hematopoietic stem cells bearing the ineffective erythropoiesis with an allergenic one which is capable of effective erythropoiesis. And it is not only to the disease erythropoietic component, rather the stem cells, they replace the whole of the entire hematopoietic system. So the complications of the repeated transfusions are reduced, and the patient after a successful transplant leads a normal life. So why we need transplant? Why we do not go for conventional treatment? The conventional treatment or the standard of care treatment in thalassemia is still repeated blood transfusions and good or optimized iron chelation therapy. The thing is that there are the complications of regular transfusions, as you all know. There are many. And second thing, the chelation therapy, its complications, the cost. If we compare the cost of the regular transfusions along with the chelation therapy of a 1 year, it's almost equal to a transplant of a patient, full match transplant. So if we invest on the transplant of a patient single time, I think it's much better for treating him for years and years and getting nothing at the end of the day. The Syro classification, basically this is a classification on which we select our patient, and these are the 3 categories which are the risk factors or the risk categories on which we decide that the patient is a good candidate for transplant or not. In class 1 category, there are 3 basically parameters which we have to see. The hepatomegaly 2 centimeters से ज़्यादा हो, portal fibrosis की presence, and history of inadequate iron chelation therapy. Inadequate iron chelation therapy means that if you have not started chelation after 2 years of age, and it should be जो initial classification है उसके लिहाज से injection desferol up to 50 to 60 mg per kg, at least 5 days per week for at least 10 hours per day, because it is given subcutaneously. So it is very difficult in majority of the patient to start the chelation at such a younger age, and compliance is also poor in many patients. In class 1, all these parameters are absent. In class 2, 1 or 2 can be present, and in class 3, all the 3 parameters are present. So the good candidates are those who are either in class 1 or in class 2. The class 3 patients are usually deferred from transplant because there are many complications which we can 4C. The different conditioning regimes, I'll just touch a bit on that. Preparatory regimes on transplant, they must achieve two objectives. Number one is the elimination of the disordered marrow and establishment of a tolerant environment that will permit transplanted marrow to survive and thrive. A myeloablative conditioning regime जो thalassemia में use होती है, that's busulfan with cyclophosphamide, and it has been considered standard of care for transplant in severe thalassemics for many decades. This is an Italian study, पुराना data है, लेकिन still the outcomes are similar. जो class 1 के थे, they are around 80-90% से above होते हैं. Class 2 वाले, they reach around 80% and class 3 around 70%. And adults में जो है इसका outcome, it's not good. This is the data of transplantation till 2006. That was of our own institute. इसमें जो हमने इसमें जो थे usually class 1 class 2 के patients थे, and the outcome was अगर हमने bone marrow transplant किया था, it was around 80%, और अगर हमने stem cell harvesting की थी donor से, तो it was around 70%. अच्छा, this is a graph. ये इस overall survival show कर रहा है जो हमारे पास इस वक्त patients का है, it's around 75%, और उसमें जो disease free survival है thalassemics में, it's around 68%. So some of the patients, yes, they do relapse and the disease come back, but afterwards they come back to the normal life and they live like a normal thalassemic patient. But it's 68% patient or 70% patients, they are cured for lifelong. Now coming to our next indication, most common indication that we come across is aplastic anemia. The literature reports, as per Pakistani, I have found few studies. It reports that around 3.5% per million cases in Pakistan are diagnosed each year with this aplastic anemia. And its inherited counterpart is Fanconi anemia. The Fanconi anemia patients also present to us with hypercellular marrow, aplastic anemia, but they have some somatic abnormalities as well. And there is the main defect or the main pathophysiology in this disorder is defect in the DNA repair mechanism. These patients are at increased risk of infection, bleeding, and of course because of the failure of the marrow, they need repeated transfusions that lead to aluminization, and if we do not transplant them at the at earlier stage, we can lose the patient. These are the few somatic abnormalities. You can see polydactyly, absence of the thumb hoti hai, ulnar deformities hai, and there is cafe olive spots or skin pigmentation abnormalities. These patients are ideally diagnosed on these chromosomal studies in which you can see multiple breaks in the chromosomes because there is a DNA repair mechanism. So the intercross linking agents hote hain, if they act on that in a normal person, we have an inherited mechanism that will repair these defects. But in Fanconi patients, these defects remain unrepaired, and so they come across different complications. Because of this difficult pathophysiology, the Fanconi anemia patients cannot receive the normal conditioning regime, and you have to modify these cells because they have marked sensitivity, hypersensitivity to the alkylaging agent. So the cyclophosphamide dose, which is normally given in normal aplastic patients or the other patients, is 100 to 200 milligram per kg. But in these patients, because of the toxicity, risk of toxicity, the dose is reduced to less than 10-folds. And this is then given in combination with fludarabine, and then the outcome now has improved to 78%. The pre-transplant workup of these patients, you have to do the bone marrow, yes. Once you have diagnosed aplastic anemia, then you have to do detailed examination of the patient, because sometimes the chromosomal breaks, even in these patients, can be negative. And if you expose these patients to the standard dose of cyclophosphamide, then it will be a big problem. So we have to do a detailed examination of the patient, including the scans, because some of them have got absent kidneys, and it can be only single marker which can indicate that this is not simple acquired aplastic anemia, it is because of some inherited syndrome. Best results are then when transplant is done as early as possible after diagnosis, so you can save the patient for complications. Post-transplant follow-ups in all the patients is if it's Fanconi, there's something special for that is in Fanconi you have to go for longer follow-ups to see for the malignancies, because secondary malignancies are common in this disorder. Otherwise, looking for the GVHD, infections and these things, or the monitoring of the disease status, it is common in thalassemia and other disorders. The third common indication is immunodeficiency. इसमें भी यही है you have to go for the transplant as soon as possible. We at NIBD, I think we have done the transplant of the most youngest patient of ours was, I think, 21 days old. She was diagnosed at birth because she had a history of sibling death, and she was diagnosed as severe combined immunodeficiency syndrome. Her HLA was matched by luck with her brother, and she was immediately transplanted, and she is now, I think, 12, 13 years of age, mashallah, well. So prompt diagnosis, infection control, and early transplant is the way forward to immunodeficiency. Now coming just a small tribute to my mentor. This is the history of transplant, Dr. Tahir performing the bone marrow harvesting. He was a pioneer of stem cell transplant in Pakistan, and he performed the first transplant in 1995 on a young patient of acute myeloid leukemia. Afterwards, he trained multiple bone marrow transplant physician and nursing team. Along with that, developing a team and logistics and a specialized center of lab, molecular lab, and the basic lab to perform all those investigations that are necessary to go on for the transplant. Further, the team took responsibility of training the other human resources or to the staff of the BMT units and for developing units in public and private sector hospitals. These three centers have been established in 2009. Dr. Tahir and his team assisted Shifa International Hospital in establishing the BMT unit in 2015. National Hospital Lahore, we established a BMT unit there and performed, I think, around 25 to 50 transplants there. And in 2016, Government of Punjab asked Dr. Tahir to establish a unit there, and the transplant unit is working since 2016 there, performing multiple transplants there. And another transplant center is now at NIBD Lahore. That's it. Another center is working also. To date, more than 850 transplants for various disorders have been carried only at NIBD. The Armed Forces Institutes and the Shifa International and other institutes, they are also working, and new institutes are opening day by day. But still the need for bone marrow centers in Pakistan is much higher because the disease load is still very high, and we still need trained physicians and nurses and logistics to carry on this work. So this is the take-home message, that we have to carry out his work. And that's all. Thank you.
Are there any questions or comments from the audience?
Gigi.
What kind of basic tissue type are you? Initially it was ABDR, which is certainly not enough for bone marrow transplant, but there it is. Your center was carrying out bone marrow transplants on ABDR, and then now the minimum requirement is A, B, C, DR, and DQ. So that kind of tissue type is required, and you know that you are also carrying out one haplotype mismatch transplant. So
yes, we are.
You are looking for DSAs
yes,
by Laminex,
yes,
and so the data and your workup. That should be up to the mark, up to the minimum standards. For tissue type, for effect, I know that still there is just ABDR, which is not enough.
Yes, we are, majority of our transplants are 6x6 mesh, and in very few cases we go for high resolution HLA typing, where yes, we are doing 6x6 transplant.
Not the high resolution one?
Not in every case, not in every case.
So I was just talking about all the loci. Minimum is ABC 10. So 10 out of 10 is minimum for bone marrow transplant. So that's the minimum standard.
Okay,
and
so you are suggesting me that we should go for at least 10. Okay, thank you. Any more questions?
I just want to pay tribute to Tahir Shamsi for establishing bone marrow transplant in Pakistan, and we are very unfortunate that he left us early. And I wish you all the very best to continue the great work that he started here in Karachi and in other centers. May Allah bless him.
Thank you.
Good afternoon. First of all, I must thank Dr. Sabia for inviting me to chair this session. I am executive for this meeting. I am basically a histopathologist. And next, it is my pleasure to announce our next speaker, who is none other than my mentor, my colleague at SIUT, Professor Dr. Mirza Naqi Zafar. He is Professor of Pathology and Head of Research and Publications, SIUT in Karachi. He graduated from University of London in 1975 and completed his PhD in 1979 from the same institute from where I did my diploma in clinical pathology, the Royal Postgraduate Medical School, University of London. Currently, he is supervising chemical pathology and transplant immunology, and in both research as well as routine laboratory work. He is truly a multitasking personality. Please, sir, Naqi Zafar sir.
Thank you very much. I'm going to take a view of immunology from the donor's perspective in transplantation. So this is a model based on donor factors in predicting long-term survival of living-related transplants. Whenever we do transplants, the outcome is also always very important. And the donor plays, the kind of kidney that you place in a recipient really determines what the outcome of that particular kidney is going to be. So there are a number of donor factors that have been identified that impact the long-term survival of that graft that you put in the recipient. This is, of course, the most important is the degree of HLA match, donor age and gender, absolute GFR of the donor, the number of nephrons or the nephron dose that you are putting in. And then there's another important factor. If you are taking a kidney out of an individual who's 50 kg and you're going to put it in an individual who's 70 kg, there's going to be stress on the kidney. So donor-recipient body weight ratio has also been an important factor in determining long-term outcomes of the graft that we put in. In our region, especially CAS region, majority of the activity is related to living-related donor as well as in Pakistan. So majority are living-related donors. So it's very important, and majority of the cases are where we have familial donors, blood-related donors. So this model perhaps would provide an opportunity to select the best donor that is available in the family. So we try to develop a model to predict long-term graft survival in living-related donor kidney transplantation based on these donor factors. I've put this, that donor set, data of 2883 donors, and these kidneys were followed up for more than 12 years. So my focus is on the kidneys that we've put in. The mean age was 35. As you can see, majority of them were 18 to 35 years of age. We are very fortunate at SIUT that we keep a balance of gender, 50-50%, and the body weight ratio was 1.25. Pre-transplant GFR, we look at the uncorrected creatinine clearance, which gives you the actual function of the kidney rather than adjusting it to body weight. Majority had very good uncorrected GFR of more than 111 mil per minute meter square. And as you can see there, majority of the recipient, the matching was 6 and 4 and 5 antigen, about 50%, and HLA-DR match was also very good in majority. And relationship was parents, sibling, majority, offspring, spouses. I think we have done the majority number of large number of spousal transplants. So these were donors who received those grafts. There were 13% were children. Mean age was 29. Here, majority were men. Time on dialysis was 8 months, and the majority had 0 PRA, 80%, so they were not sensitized. Immunosuppression, we used induction in those cases which were poorly matched, but majority of our patients are still maintained on cyclosporin, azathioprine, and steroids. Overall acute rejection rates in these patients was 16%. Comorbidities were diabetes, mainly hypertension, chronic infections was tuberculosis, recurrent UTI and hepatitis, and the primary renal disease, as you can see here, was really unknown in more than 50%. So this is how the donor score criteria was established, that if the donor age is up to 30 years, you would give a score of 0, and as the age increases, you would give a score of 1 or 2. Gender, male seemed to be doing better in long term, so 0 for male and 1 for female. Donor recipient body weight ratio, it's more than 1, that means the weight of the donor was more than the recipient. HLA match identical, and then with the new modern immunosuppression, we can control the HLA mismatch by using induction and tacrolimus, so there's 0 and 1. Uncreated clearance, the maximum 110 is 0, then 1 and 2. So I don't know whether you can look at the bottom. So if you look at the different scores of a donor, you can come up with the summation of what the total donor score is. So this is what the 2,283 donors had. 3.6% had 0 score. These were HLA identical, young donors, male, high GFR, and high donor body weight ratio. So majority, if you can see, had a score up to 3, and very few had 7 score. We were poor match, old donor, female, low GFR, and low body weight. Sometimes when there's only one donor available in the family, you have to select donors who are older in age, more than 50 years of age, poor match, because you can cover that with good immunosuppression. So these are the donor factors. So HLA-6 match antigens, kidneys do much better with 15-year survival of 58%, and they get 5% rejection as compared to 20% rejection in 0 to 5 antigen match, and this statistic is significant. If you look at the donor age, younger donors have lesser rejection. 15-year survival is 49%, 5-year survival is 82%, and as the donor age increases, you get poorer graft survival. And male, 15-year survival is 47%, 34% in female, and 78 and 76% at 5 years. So up to 5 years, it's almost the same, but as the years go by, the survival is poor. And this is the nephron dosing. If the GFR is more than 111, you get the best survival, and the least rejection episodes are really similar, except when the GFR is very low. This perhaps also pertains to older age. And this is the body weight ratio. If you put in a kidney from a high body weight individual to a low body weight recipient, the kidneys do well because there's less pressure on them. So this is the outcome. If you look at the donor score, if the donor score is 0, you get a 15-year survival of 87% with 2% rejection. And if you have a donor score of 7, you get a 15-year survival of 29% and a rejection rate of 27%. So with the donor score increasing, you get lower graft survival and increasing number of rejections. If you group up, like I have done here, group up the 0 to 1 score, group 1, 2 to 3 score, group 2, because of the numbers, I've combined them together. As you can see here, that the rejection rate again increases with the donor score, and the graft survival reduces with the higher donor scores. Now, one independent factor that affects graft survival is if an individual, if a recipient has a rejection episode. And if there is no resection, you get a better graft survival. If there is any rejection, and Dr. Mubarak here is our histopathologist who diagnoses our rejection, and we always argue about borderline rejection, and I'm still saying that here. But nevertheless, rejections do decrease your overall graft survival. So this is group A, where there was no rejection. So there were 1,908 recipients in whom we had put in our kidneys. And so those kidneys which had a 0 score had a 15-year survival of 86%, reducing to 44% if the donor score was 7. And these are those who had no rejection episodes. So rejection episodes have been taken out from the scoring system. And if you combine these two scores, group 1, group 2, group 2, you see the same effect. In this group there were no rejection. You get excellent graft survival in 0 to 1 score of 75% at 15 years. And this is the group of 375 who had rejection in their follow-up period. As you can, I have grouped these scores here. As you can see here, even with the rejection, those kidneys which had a 0 to 1 score had a 15-year survival of 73% as compared to score of 6 to 7 of 12%. So even when there is rejection, the donor factors play an important role in the long-term outcome of the graft of the kidney that we put in. So if you look at the group with 0 to 1 score, you don't see any difference whether they had rejection or not, because the donor factors were all very good in terms of the kidney that we have placed in. So we tried to test our model. This is the 283 on which the study was done. These are the mean survival days at up to 15 years. And then we had a test patient population using data from other patients as to how long they survived, and we applied the donor score criteria. And as you can see here, there were no statistical difference. So the model was able to predict the long-term survival of the kidney that we are putting in. So what's the advantage? And this is the test group and the model that we had made. So in conclusion, this simple model can assign scores to donors available in the family to select the best donor in terms of long-term outcomes. And this is very important in a living-related donor program because we have several donors in the family. And now with the concept of swap donors, that if you don't have a blood match donor in a family, you can ask another family and swap the donors. So this model then allows you to select the best donor that is available in the family to be able to predict the long-term survival. So the relation of rejection withdrawal scores can help tailor immunosuppression also, because if we have a donor and the score is high, and we know the risk factors, we can tailor the immunosuppression because we have a very broad or different choices of immunosuppression now. So donor factors significantly impact long-term graft outcome survival in living-related kidney transplant. A better version of this paper, where we had included also the factors in recipient, was published in Nephrology in 2018 with our collaborator Jermaine Wong in Australia, Howard Abbas and myself. So this model is now being practiced in Westmead Hospital in Sydney and also in Shiraz, where they are doing Tehran, where they are doing liberated transplant. Thank you very much.
Thank you. Thank you very much, sir, for this very interesting and insightful talk. The results from this study show that donor factors are also equally important, if not less important than recipient factors. So the forum is open for questions or comments.
Thank you. Thank you. Very nice talk and a lot of applications. My question is, it may be a naive question because I'm not in transplantation immunology, but does age affect rejection? For example, if you are giving kidneys to older recipient, do they reject less than the younger recipient?
Okay. First question first. An older donor, what happens in an older donor, the cell membrane shrinks and the HLA molecules come out. So there's more epitopes for the T cell to recognize.
But the T cell response is growing.
Yes. In older people, so what we do is that if you put older people have poorer immune responses, so what we do now is if we have an older donor, best to select an older recipient, best to select an older donor as well. And that works very well for them. But putting in an old kidney in a young person, that kidney is bound to have more chances of rejection because of the more exposure of the HLA antigens on the cell surface.
Professor Tahir Aziz, please. Thank you. Professor Naki, thank you very much. Excellent as always. One kind of very discriminatory here. What's the limit for old for transplant? What is old?
Recipient or the?
Both of them. Is
okay.
The 20-year age difference? There is a supplementary question. You said gender is balanced, but 70% of your recipients were males. So gender is not that balanced.
In donor, see, the problem universally is that most living donor transplant centers have more females. Our donor balance is the same. Now coming to why there are more male recipients, because renal failure is more in males than females. That's universal. More males have renal failure, ESRD, as compared to females.
Right. So I don't think that difference is that much, that we have 70% of the recipients. I suppose there is something to do with the provision of this expensive treatment to preference is given to males. I'm not very sure.
Well, you know, we have done 7,000 transplants, yes. And there are no cost involved,
yes.
And everybody is welcome.
Right.
And so we have a control over the donor. When we are selecting the donor, we have no control of the patient who's coming in. So right. There are more patients coming in who are males with renal failure.
And then there's this important question: what happens to donors in 15 years? What about these donors? Did they develop renal disease? Did they suffer from hypertension?
That is a different study, and we are very proud of that work that we are doing. That at SIUT we also follow donors in a dedicated donor clinic. We have had 3 publications so far where we have shown that 0.2% of I think 4,000 donors had end-stage renal disease, and the main reason was that they presented very late to the donor clinic. We are just we have just submitted, in fact, a paper in transplantation where we have followed up donors who have completed 15 years of donation to 30 years. So we have the longest one-to-one survival of donors, and they are like general population. The only difference is they do better than the general population because they are followed up every year. Their hypertension is diagnosed, their diabetes is diagnosed early, and their other diseases are diagnosed early. So if you look at our donor population, average, if the diabetes in the general population is 40%, you'd find it half in the donor, same as hypertension, same as chronic kidney disease. So we don't take donors less than 18. 18 is the last. We have gone up to donor age 70 because their renal function and their health was so good, and they survived. So we have donors who are 95 years of age right now. Sorry?
Recipient donor parity in terms of age. Recipient donor parity.
Age parity? That really depends on the donor that you are getting in the family. But if you look at our average donor age, I think the slides are there, it's about 30, and the average recipient age is about 40. So there's a disparity about 10 years.
But you yourself said that the LA are more displayed in older age groups.
Yep.
So if you give a kidney from an older age group
To a younger person, yes.
more likely to
Yeah. That's why you have more rejection episodes in pediatric transplants, because children get their kidneys from their parents, and so our immunosuppression protocol is totally different for pediatrics. They all get induction therapy with IL-2 antagonist. That's standard protocol, and they get tacrolimus and MMF, more potent immunosuppression.
Thank you.
Your
Thank
donor pool.
Sorry?
Your donor pool, if it needs rejection, it needs transplantation or a kidney. If this one kidney fails, do you give preference? This is an ethical issue.
I think 8 in the last 40 years, 8 individuals have had end-stage renal disease after donation. 4 of them were lucky enough to get transplanted from a family donor. 2 did not have donors in the family because the disease was genetic, and they had long-term dialysis till they died. You have to follow them up because, we generally don't care about our. Yeah.
Thank you very much. With this, we
Okay.
conclude this
Yes,
session.
thank you. Now we have our oral presentation. The first is by Anusha Sikandar Ali, is that correct? And she's going to be talking about frequency of anti-endothelial end cell antibodies at the time of renal graft rejection from the Sindh Institute of Urology and Transplantation, Karachi.
Assalamu alaikum and good evening. I am going to be talking about anti-endothelial cell antibodies. Renal transplant has been the treatment of choice for end-stage renal diseases. And anti-HLA antibodies are one of the very significant antibodies that can cause rejection. However, non-HLA antibodies are also involved in renal graft injury. One of those non-HLA antibodies is anti-endothelial cell antibodies. As the name indicates, these antibodies are directed against the antigens present on the endothelium upon any stress. Whenever any stress comes, for example, tissue reperfusion injury or inflammatory responses, then these antigens are expressed and the anti-endothelial cell antibodies react with them. Basically, AECAs are a heterogeneous family of antibodies reacting with endothelial cell antigens. These are involved in graft rejection and they cause series of immune reactions, for example, inflammatory responses, leukocyte adhesion, and cellular cytotoxicity, which eventually leads to tissue injury and poor graft outcome. This diagram shows that non-HLA antibodies can be present before and after transplantation. And again, as I've already shared, that these react with the antigens present on the surface. Okay, the objective of the study was to evaluate the presence of AECAs in the absence of HLA class 1 and class 2 molecules, class 1 and class 2 antibodies. This was a descriptive, cross-sectional, single-center study that was conducted at SIUT, Sindh Institute of Urology and Transplantation, from July to November 2018. We chose 70 transplant patients with biopsy-proven rejection, and we chose patients who tested negative for complement-dependent lymphocytotoxicity, that is CDC cross-match and flow cytometric cross-matches. The assay was performed via indirect immunofluorescence assay, that was our principle, and the statistical analysis of the data was done on SPSS version 20. Okay, this is the titer plane technique that we used for the assay, and the other diagram shows the principle of indirect immunofluorescence assay, which shows that the primary antigen and antibody complex is captured by a secondary fluorescent labeled antibody that is targeted against that primary antigen antibody complex, and that is fluorescent labeled, so that can be seen via fluorescence microscope. If the results are positive, the picture is going to look like this with a granular green colored fluorescence. So this is the picture of HUVC positive sample. Coming to the results, we had 70 transplant patients, out of which 60 tested negative for HUVC, while 10 tested positive. Among these 70 patients, we had 23 identical patients with identical haplotype match, 21 were more than one haplotype match, 18 were one haplotype match, and 8 were less than one haplotype match. To make the picture clearer, the other pie chart shows that among those 23 identical transplant candidates, 3 tested positive for HUVC. That shows that even after being identical match, there are still chances of rejection, there are still chances of endothelial cell activation and graft failure. So to conclude, I would like to say that despite of HLA antibodies, we should take into consideration the non-HLA antibodies as well. So we can test them pre and post-transplant, so we can improve our graft outcomes and better survival of the graft in our patients. Thank you.
Any questions or comments? Thank you very much. About 20 years ago, we looked at HLA identical transplants who had rejections and who lost their grafts. And at that time, we were not doing these antibodies because we didn't have them here in Pakistan. But the samples were sent to Terasaki lab, and the study was done on HLA identical transplants, and almost 70% of them, because of failure, at different time points was endothelial cell antibodies in HLA identical transplant. So this is something that we need to introduce in our transplant follow-up, even in pre-transplant, because you can still have some viral infections and bacterial can cause endothelial cell antibody prior to transplant. The next presentation is by Ambar Khan, a study of cell culture and growth kinetics of mesenchymal stem cells from BLB cell mice, and a short review of clinical trial up to now in the field of stem cell therapy, Sindh Institute of Urology and Transplantation.
Assalamu alaikum and good evening, audience. This is Ambar Khan here. By profession, I'm a physician assistant at SIUT Hospital. Today, the topic of my presentation is stem cell. It is not a word, it's the future of medicine. So, today the title of my presentation is Isolation, Expansion and Growth Kinetics of Bone Marrow Adipose Tissue Derived Mesenchymal Stem Cells from BLB mice, and a short overview of mesenchymal stem cell therapy. Hallmark. So, what is stem cell? Stem cells are multipotent adult cells which have renewal property. Look at some hallmark of mesenchymal stem cells. It's reduced inflammation, it has anti-apoptosis property, it has cell differentiation property, and regeneration property. So, the objective of my study is isolation and propagation of bone marrow mesenchymal stem cell and adipose-derived mesenchymal stem cells. Also, comparison of growth kinetics of mouse bone marrow and adipose-derived mesenchymal stem cells. As we know that benefits of stem cells speaks aloud due to its rapid therapeutic action and application. It become a useful tool for tissue engineering and molecular medicine. So, the question arises here, how we get the mesenchymal stem cell? Yes, we can grow it in our own lab. So, as per my objective, we grow the mesenchymal stem cells and choose other than bone marrow, another alternative source, that is adipose-derived mesenchymal stem cell, to grow and compare the growth kinetics of mesenchymal stem cells of both those sources. Come to the methodology. We use standard parameters to compare the growth kinetics of bone marrow-derived mesenchymal stem cells and adipose-derived mesenchymal stem cells, including the parameters culture time, growth curve and expansions, population doubling time, colony forming unit, and we see the PCNA gene by PCR. So, come to the results. We see that in population doubling time and culture of the cells, we see that adipose tissue grow more rapidly as compared to the bone marrow-derived mesenchymal stem cells. And when we talk about the population doubling time, it takes half time and less time as compared to bone marrow-derived mesenchymal stem cells. Similarly, when we see a comparison of bone marrow stem cells and adipose-derived mesenchymal stem cells in CFU assay, we see that adipose-derived mesenchymal stem cells show more number of colonies and clear defined pictures of the cells. When we see the growth curve analysis, we see that high peak we can get it from adipose-derived mesenchymal stem cells as compared to bone marrow-derived mesenchymal stem cells. See, in this picture, we can say that adipose-derived mesenchymal stem cells show a PCNA gene that shows that the cells end in a proliferating phase, G1 phase. Conclusion, in a nutshell, we can say that adipose-derived mesenchymal stem cells is an alternative source, a good source for the clinical trial. So let's come to the second topic of my presentation, that is the stem cell therapy. As I am belong to SIUT, it's a transplant unit. So the first consideration point for me is transplant recipient. So look at this, how the stem cell help the how the mesenchymal stem cell help the transplant world. Researchers said that immunocompromised drugs are have adverse effect on a recipient. So with combination of mesenchymal stem cells, it can promote the graft survival, and also it reduces the number of immunocompromised drugs to the recipient. So these are ongoing clinical trial cases, FDA approved, and globally working on diabetic nephropathy, end-stage renal disease, and CKD, chronic kidney disease. So let's hope that these magical mesenchymal stem cells use for here we can also use these magical mesenchymal stem cells to serve the humanity. Thank you.
Thank you. Any questions from Amber Khan?
Yes, we separate it, we dissect the mouse and we get it from the abdominal of the mice, and likewise bone marrow we see the from the bones of the mice, biopsy mice, we use it and cut it and remove this from the lymph from the bones. Yes, it's another study of my seniors on which we do the fluorescent microscopy, and we also run some other markers by which we confirm that these are mesenchymal stem cells. And yes, sir. CD19.
So I have another question. So you compared the growth kinetics of two different kinds of mesenchymal stem cells. So how did you ensure that the starting concentrations of both were equal, and that you have a pure isolate of mesenchymal or what are the other stem cells, pure isolates, and the starting concentrations were equal?
We count the cells of for each parameter, and then we pour into the each and every the method which are using for methodology of growth kinetics.
There must be a minimum count that you count before you plate them.
Yes, sir. For CFU, we use 60 cells for 60 cells, and for growth curve analysis, we use 3 exponential 4 cells of each 24 mm square well.
Does that make sense? Okay. Thank you. Shall I inform them? This young lady is a transplant recipient, and mashallah has been doing very well and was transplanted, working at SIUT, transplanted at SIUT, and now working at SIUT. So that's the benefit of a kidney transplant.
Thank you so much.
Thank you. Okay, the last presentation is by Dr. Sabah Sarfaraz from Dow University of Health Sciences, and an association of dectin-1 polymorphism with candida infection in liver transplant recipients.
Bismillahirrahmanirrahim. Assalamu alaikum. Good afternoon, everyone. Dr. Sabah Sarfaraz, serving Dow University of Health Sciences as assistant professor. Well, I'll be talking about association of dectin-1 polymorphism with candida infection in liver transplant patients. We all are well aware that adaptive immune system is crucial for human survival, but we can't underestimate the importance of innate immune system, which Professor Rabia has also elaborated. If we say that innate immune system is an old game with new players, it won't be incorrect, because innate immune system is rapidly evolving field with novel cell types and molecular pathways are discovering, so the paradigm is continuously shifting. Innate immune system is hardwired with germline encoded receptors for immediate responsiveness against a variety of pathogens. They recognize evolutionarily conserved pathogens in the form of either pathogen-associated molecular patterns or damage-associated molecular patterns. Once stimulated, they cause release of inflammatory cytokines, chemokines, and reactive oxygen species, which play an important role in providing defense. There are 4 families of pattern recognition receptors: toll-like receptors, rig-like receptors, nucleotide oligomerization receptors, and C-type lectin receptors. C-type lectin receptors are again of type 1, type 2, and soluble type. Dectin-1 is one of the type 2 transmembrane C-type lectin receptor, previously known as beta-glucan receptor, which is present on most of the innate immune cells, and they play a very important role against fungal infections. And not only that, this receptor also modulates its adaptive immune system by stimulating naive T cells into Th1 and Th2 cells. Liver transplantation is a therapeutic modality for choice in patients with end-stage liver disease. Pakistan, being third world country, have a heavy burden of these disorders, and around 35% do require liver transplantation. And there are a number of indications starting from autoimmune disorder to hepatocellular carcinoma. Post-transplantations, there is a long list of complications, but being a third world country, infection is our major threat in the form of bacteria, viruses, and fungi. So previous studies have mentioned that the innate immune system receptors play an important role in providing antifungal immunity in not only normal but in transplant individual as well. So genetic, the previous studies has mentioned that the polymorphism in dectin-1 has led to increased fungal infections, which causes decrease in pro-inflammatory cytokines and decrease autoreactive T cells, hence increases the tendency of infections. Here I would like to quote 2 studies, one from Italy and another from Dutch population. They evaluated the same dectin-1 mutation Y2386, and they saw that the mutation in dectin-1 receptor increases the prevalence of increases the susceptibility to aspergillosis after hematopoietic stem cell transplantation, especially when the recipient and donor both have the polymorphism. And the Dutch study, they mentioned mucocutaneous cutaneous fungal infections when they found when they were evaluated that it was associated with the dectin-1 polymorphism. So the objective of this study is to analyze genetic polymorphism in dectin-1 714, which T is replaced by G, in liver transplant recipients, their respective donors, and healthy volunteers. Evaluation of dectin-1 receptor on their cellular expression and the identification of their function, what happens to their functional alteration in terms of the analyzing the cytokines like interleukin 1, interleukin alpha, interleukin 6, and IL-1 beta. And then we evaluated fungal infection in post-transplant liver transplant recipients, and we followed them for 6 months. So the study was conducted in Dow University of Health Sciences. What is a prospective study? Initially we enrolled 118 patients, and the study was approved by Institutional Review Board. And out of 118, there were 60 healthy volunteers and 29 recipients and 29 donors. But unfortunately, due to that of 2 recipients, we left 27 recipients, and due to 6 were excluded due to the differences in the data collection and sampling issues. We were left with 23 donors. So we evaluated the expression of dectin-1 polymorphism. We evaluated their expression by flow cytometry, and then we evaluated the cytokines. So it was found that the most common allele was T, which was present in, more frequently than G, and this was the genotype in 3 population. TT was most common, followed by TG, and then this was the electropherogram, TT, TD, and TG, and then expression of dectin-1 was increased in wild type as compared to homozygous and heterozygous. And then when we evaluated the cytokine analysis, we saw the differences when we stimulated the polymorphonuclear blood cells with beta glucan. So we saw that there was with dectin-1 polymorphism, there was decrease in number. Then we evaluated the patient for 6 months, and we found that the number of infections were increased, especially in 4 and 6 weeks, followed by 2nd and 8 and 12 weeks. To conclude, the heterozygous TG was present 18%, and homozygous variant were present 4%, and dectin-1 polymorphism was associated with increased fungal infections, and cell surface expression of dectin-1 was diminished. And interestingly, the inflammatory cytokines were reduced in the polymorphs in selected cases with dectin-1 polymorphism. So, to the best of our knowledge, this was the first study which evaluated dectin-1 polymorphism in liver transplant recipient. We do have certain limitations like small sample size and predominant liver male patients, but this was the strength of the study, that this was the first study which looked into the dectin-1 polymorphism. I would like to acknowledge Dr. Talat Rumi, which led the whole project, followed by Dr. Shaheen Sharafat, Dr. Shams Nadeem, who was the head of the Department of Liver Transplant Unit, and the Vice Chancellor and Pro-Vice Chancellor of Dow University of Health Sciences. Thank you so much, very much.
Thank you, Dr. Sabahat. Any questions or comments?
Thank you. Very nice talk, Sawad. And it makes a lot of sense because innate immunity's major role is surveillance. And most of the fungal infections are usually on the surface. Yes. So, you know, you were going in the right direction. I just wanted to make that comment.
Thank you so very much, madam. Highly obliged. Thank you.
Any other question? Well, thank you very much.
Thank you, everybody. Thank you, sir. I would request Dr. Rabia Hussain to please come on stage to present the shields. Thank you. I request Dr. Naki to please come and take your shields. Professor Mubarak had to leave because he had a talk in his another session. You have to present both these shields to Dr. Naki as speaker and as chair. So you have to show it up like this. Thank you, madam. Yeah. I'll take this. Dr. Mubarak. Yeah, Dr. Rabi, could just present. सर ये ले लेंगे अब लिखा है Thank you, Dr. Rabia. Sir, please, Dr. Naki, we'll have you present the shield to Dr. Saima Muzzer, if you can come on stage. That's the wording.
So I have to give the concluding remarks. Of course, the best talk was by Dr. Naki Zafar. Dr. Saima talked about bone marrow transplant, and that's something that is new, that is coming up in Pakistan, and you have been the pioneers and continue the good work. We still need to go ahead and do a bone marrow transplant for malignant disorders. Benign disease we have done, but that's a bigger challenge. But that is something that we need to be done, because there are more and more patients of bone marrow leukemia and lymphomas who need this kind of treatment. There were very nice presentations. Endothelial cell antibodies by Anusha. They are a big problem actually, and the more we do the tests, we realize that these antibodies are there even prior to transplant, and it's I think you are going to give a talk on, you have already given a talk, the role of non-HLA antibodies is becoming significantly important in transplantation. And we did a paper with a chapter, in fact, with Paul Terasaki some years ago, where we identified the importance of non-HLA antibodies and kidney transplantation. Stem cell therapy, we have a stem cell lab where this young lady presented her work. The idea is we are going towards growing kidneys from stem cells. So that's the project that is going on for the last some years. It's very interesting how stem cell lab is, and if anybody has not seen it, come and see it. It's a very isolated, protected place, negative pressure everywhere, but that's an exciting field in transplantation, where stem cells are being used for regeneration of organs, and we go on from there. Yes, and very soon we'll be getting 3D printers for. There are lots of genes which predict outcomes in transplantation, and of course, as Sabahat has clearly shown, that genes can predict and tell us about different infections. So this is a new field. Everything is moving towards genetics, and I think that's the way forward. Abhi humko malum hai ke which diseases we are prone to, so very soon we'll have our genetic analysis done कि आपको yoga, 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 and so be it. So thank you very much for your attention, comments. Thank you all. अभी इसके इम्तिहान और भी हैं.
Bismillahirrahmanirrahim. Welcome back, Dr. Sabahat Sarfraz, serving Dow University of Health Sciences as Assistant Professor. Well, we are proceeding with our fourth and last session of today's session. Well, this session is all about allergy, inflammation, and immunotherapeutics. We have two distinguished chairs, three worthy invited speakers, and the session will be followed by 6 free papers. So it's extreme pleasure to introduce Professor Rabia Hussain, who is an Android professor and former founding head of the Department of Microbiology, Khan University, and she is a professor. She obtained her fellowship in clinical immunology and at Johns Hopkins University, and later held a faculty position there. Over there she worked on ragweed pollen allergen, and later on at NIH she identified the fundamental role of IgG antibodies in blocking allergies. She has around 175 full-length papers in international peer-reviewed journals with high-impact Google Scholar Citation Index. She has been awarded with the Tawaz Prize, Lifetime Achievement Award, Sitara Imtiaz, Tamga Imtiaz, and she is elected as Fellow of Royal College of Pathologists, Pakistan Academy of Sciences, Islamic Academy of Sciences, and Central and South Asia. She's also serving on numerous international research steering committees. So we warmly welcome Professor Rabia. Next distinguished chair of the session is Dr. Najia Talat. She is serving as assistant professor and vice chair of research in the Department of Biological and Biomedical Sciences at Khan University. She did a PhD in biotechnology from Karachi University and did a postdoc from Wine Cancer Institute. She has authored several research articles in national and international articles. So please welcome Dr. Najia. And then now the session is over to our distinguished chairs. Please proceed.
Thank you, Sabahat, for a very nice introduction for Dr. Rabia and for myself. So our first speaker is online. So I would request Dr. Rabia if she kindly introduce our invited speaker.
Can you get the person online?
Sorry, can you get? Yeah.
Welcome, Dr. Pavel. Dr. Pavel is our first speaker. Dr. Pavel Gordon. Dr. Pavel Gordon is a consultant clinical allergy and immunology at the Hull University Teaching Hospitals, NHS Trust, Hull, UK. He qualified in medicine from the Medical Academy of Latvia and specialized in both clinical and laboratory allergy and immunology at the Royal Victoria Infirmary, Newcastle upon Tyne, UK. He undertook a period of research studying mast cells and their mediators at the University of Southampton and was supervised by Dr. Andrew Walls. I have been managing patients, this is Dr. Powell's statement, I have been managing patients with immune system disorders for more than 20 years. My clinical interests include allergen diagnostics, allergen immunotherapy, hereditary angioedema, and primary immunodeficiency. I will let Dr. Powell take over the presentation.
Thank you.
Hello? Can you hear me?
Yes, we can.
Yes, we can.
Brilliant, thank you. I hope it all goes all right. Can you see my slides? Can you see my slides? Oh, just a second.
Yes, we can hear you. Are you able to share your slide?
This is what, oh yeah, I can see, yeah. Great. Can you see my slides now?
Yes, yes, we can.
I'm sorry for the delay. So I will be talking on allergen component diagnostics in clinical practice. And as I was introduced, I'm Dr. Gordon, I'm consultant allergy and immunology. I'm based in Hull. Just a brief introduction to where we are. We are on the east coast of the UK. It's north of England, East Yorkshire. And this is our city, Hull. It's quite a flat place. We're no skyscrapers in Hull. And this is where I practice, Castle Hill Hospital. So if you visit UK, if you visit north of England, East Yorkshire, please do come and visit us. So it's a relatively basic talk, but at the same time, maybe complicated. So if you have any questions, I will be very, very happy to answer. This is my first slide is about classical approach to allergy diagnosis, and this is what we do in our allergy clinic on a day-to-day basis. And the allergy diagnosis always starts with good history. Without good history, we can't get correct diagnosis and ultimately correct management of a patient. After we collected good history, we will think about investigations. And traditionally, what we do in the allergy clinics, we do skin tests, and skin tests can be done with commercial extracts of allergens, or they can be done with natural or so-called raw allergens, where we use, sometimes we would use nuts, we would use fish or shellfish in the clinic, and sometimes we get more accurate and more predictable results with raw allergens, in particular with foods. And blood test is a relatively new thing, what we can do for diagnosing allergies, and blood tests were based on detecting specific IgE antibodies against allergens of interest. And after we collected the history, we did investigations, we did skin tests, blood tests. Then we would sit down with the patient, would correlate the history with investigation results, and we will define the management plan. Specific IgE antibodies. So this is, as I said, relatively new thing, what we can do in the clinics. In the allergy clinics, we can do blood tests to detect specific IgE antibodies to whole allergens of interest. And these tests have been around for several decades. More recent advances in allergy diagnostics, we can also detect specific IgE antibodies to allergen components. And this is what we call as allergen component diagnostics or component resolved diagnostics. And the advantages of allergen component testing, I will present the advantages in more detail on the next slides, but the main advantages of doing components is to distinguish primary sensitization from cross-reactivity in patients, particularly in patients with multiple allergen sensitivities. It can also help to assess the risk of patients developing serious or systemic reactions, such as anaphylaxis. And component testing can also help us to select patients for allergen immunotherapy or allergen desensitization. So this is what we do in the clinic. So this, if we see somebody with a history of allergy to peanut, so we collect history, then we proceed to testing, and there are different types of testing. So the peanut, if we do an extract from peanut, it's we call it soup of allergens or soup of antigens. So there will be multiple protein molecules which can induce allergic reactions, and they are displayed here in different colors. So in the top right corner, this is a more traditional approach. This is what we've been doing for many years. We can do tests, skin tests with whole peanut extract, or we can do blood tests. We can detect specific IgE antibodies to the whole peanut extract. But when we do that, we don't know which particular allergen components or which particular proteins the patient is sensitive when we get positive results. And here in the bottom right corner, this is what we call component diagnostics. We can separate allergens to individual allergens and then detect specific IgE antibodies only to that particular allergen or protein of interest. And this is normally done by detecting specific IgE antibodies. This is not normally done by skin testing. So what is the difference? So when we use the whole allergen extracts, because there are multiple molecules, multiple proteins, they're very sensitive. These tests tend to be more sensitive, and they're very good for screening and identifying allergens. On the other hand, when we do component resolved diagnostics, when we detect specific IgE antibodies to individual allergens, these tests may be less sensitive, but they're more specific, and they help to differentiate primary and cross-reactive sensitivity, and as a result assess a risk of systemic reactions. So this is just an example, what we call by specific and cross-reactive allergen components. And here it's done on plants and plant-derived foods. So we have birch, we have peanut, we have hazelnut. If you do allergen extracts from birch pollen, peanut or hazelnut, we get again this kind of soup of allergens or antigens. And there will be allergens or antigens which are unique to that particular species or that particular allergen of interest. For example, for birch, these light brown allergens, they're only present in birch extract, but they don't present in peanut or hazelnut extract. And the same will be true for peanut and hazelnut. We have unique allergens. But at the same time, we have so-called cross-reactive allergens, these protein molecules which are labeled here in dark blue. They will be molecules which are very similar across different types of allergens. So in birch, peanut, and hazelnut, we will come across protein molecules, allergenic protein molecules, which are very similar. So here you can see specific allergen components which are unique to each allergen, source of allergen, and at the same time we have cross-reactive allergen components which will be very, very similar. So these are just examples of specific allergen components in different sources of allergens: bee venom, birch, peanut, timothy grass, egg. And as it states on this slide, these allergens tends to be more stable. They're resistant to heat or digestion. They usually indicate primary sensitization. They are often storage proteins in seeds and nuts. They have limited cross-reactivity, and they are normally associated with more severe reactions. So if we detect specific IgE antibodies to allergen-specific components, then patients are more likely to have anaphylaxis. On the other hand, we have several groups of cross-reactive allergen components, and here I present 3 of them. One is called profiling, then PR10 proteins, and LTP or lipid transfer proteins, and several examples from peach and peanut. And cross-reactive allergens, they're generally speaking, they're less stable. They're more susceptible to heat or digestion. they usually indicate secondary sensitization. They usually lead to predominantly local reactions. It would be unusual for cross-reactive allergen components to result in systemic allergic reactions such as anaphylaxis, and obviously where cross-reactive allergens, they are the reason why we sometimes see extensive cross-reactivity between different types of allergens. So this is, here I just wanted to present several protocols, how we use component testing in our practice. And we don't test specific IgE antibodies to all allergen components which are available to us. Here there is an example of peanut. So if we suspect peanut allergy, our primary test is whole peanut extract. We can do skin test or we can do blood test, but the primary test is always whole peanut extract. This test is very sensitive. It's a good test for screening, but the positive result doesn't tell us much about the risks for our patient, about the risks of anaphylaxis in particular. So if we receive positive specific IgE antibodies or positive skin test for whole peanut extract, our next step is to test for specific IgE antibodies against allergens which would be associated with a high risk of systemic allergic reaction or anaphylaxis. So in this particular case, with peanut, if the specific IgE or skin test positive to peanut extract, our next step is to proceed to detecting specific IgE antibodies to RH2 protein, which is a storage protein. If it's negative, we proceed to testing RH6 and then RH1 and RH3, which are all storage proteins, which would be associated with a high risk of systemic or anaphylactic reactions. And only if all these 4 allergens are negative, we would proceed to detecting specific IgE antibodies to cross-reactive allergen components such as RH8, PR10 protein, or RH9, lipid transfer protein components, which would indicate cross-reactivity. They would usually indicate secondary sensitization to peanut, and they would be normally associated with a relatively or sometimes very low risk of anaphylaxis, or sometimes no clinical significance at all. So this is the protocol for peanut, and this is what I just said. So if somebody is testing positive to RH1, 2, 3, and 6, this indicates primary peanut sensitization, and these patients will be at high risk of systemic reactions. On the other hand, patients monosensitized to RH8 and also to a certain degree to RH9, they will be at low risk of systemic reactions, and more commonly they will present with oral allergy syndrome, where the symptoms are limited to the oral mucosa, mouth, throat, but low risk of systemic reactions, anaphylaxis. And this is just a summary slide, so you can see this is on the example of peanut with RH8, RH9, PR10 protein, LTP. Cross-reactivity is relatively high, but the risk of systemic allergic reaction or anaphylaxis is relatively low. As we move to storage proteins, cross-reactivity decreases, but the risk of systemic allergic reaction, anaphylaxis, increases dramatically. And these will be the patients who will be advised to carry EpiPen. These are the patients who will be advised to strictly avoid nuts. This is another example on egg, hen egg. Again, the principle is the same. We are screening for allergy by doing whole egg extract. If a whole egg extract is positive, then we proceed to components. And again, first what we do, we measure specific IgE antibodies to the component, which would be associated with a very high risk or relatively high risk of systemic allergic reaction anaphylaxis. In the case of egg, this is GAL-D1 or ovomucoid, and only if ovomucoid is negative, we proceed to testing specific IgE antibodies to other allergens such as GAL-D2, GAL-D3, GAL-D4, which would be associated with a significantly lower risk of systemic anaphylactic reaction. Okay. And patients who are not sensitive to GAL-D1 and sensitive to GAL-D2, 3, and 4, they usually can tolerate well-cooked egg or well-cooked egg products. Occasionally, they can still develop symptoms on consuming products which are made with uncooked egg or very lightly cooked egg. So again, doing components here helps to assess the risks for a patient and provide appropriate management advice. And just another example with cow's milk. Again, if we suspect cow's milk allergy, first what we do with our primary test is to do whole milk extract. If a whole milk extract is positive, we measure specific IgE antibodies to allergens which would be associated with a higher risk of systemic reactions or anaphylaxis, such as beta-lactoglobulin, BOS-D5, and casein, BOS-D8. And only if BOS-D5 and BOS-D8 are negative, we would measure BOS-D4 and BOS-D6, alpha-lactalbumin and bovine serum albumin, which would be associated with significantly lower risk of anaphylaxis. Here I wanted to present two unusual allergies where allergen components would help to diagnose specific allergic conditions. And before we knew about allergen components, we didn't know much about these conditions, so these conditions remained largely undiagnosed. So one of these components is omega-5 gliadin, which is a component of wheat grain. And specific IgE antibodies to omega-5 gliadin have been shown to be associated with wheat anaphylaxis in children, but a rather unusual condition, so-called delayed wheat-dependent exercise-induced anaphylaxis in adults. And the second condition I wanted to talk about briefly is alpha-gal sensitivity, or sensitivity to galactose alpha-1,3 galactose, which is an epitope frequently found in meat fiber, in particular red meat. And this particular sensitivity, again, it's unusual because it presents with delayed anaphylaxis, and there are several cases I just wanted to run through quickly. So the first case is a case of wheat-dependent exercise-induced anaphylaxis, and this particular gentleman, he's a 40-year-old salesman, and after playing squash for 1 hour, he felt that he developed some tingling sensation and swelling in his lips, and then very quickly he developed very itchy, large wheals all over his body. So essentially he developed urticaria. He had difficulty driving home because his legs also became swollen. And when he arrived home, his wife was frightened because his lips were very, very swollen, grossly swollen. He could hardly speak. An ambulance was called. He was treated with adrenaline, and he was taken to the emergency rooms, where he was subsequently given some steroids and observed for 4 hours. And that gentleman had, on collecting history, which is again highlights the importance of history, that he did say that previously he had similar urticarial rash after playing squash, but symptoms were much milder and resolved on their own. And he has history of mouth eczema in childhood and normally takes no regular medication, so he's healthy. So what we also found out that on that particular day when he had this severe reaction, he was running late. He was rushing home from work, and he didn't have time to eat at the time when he would normally eat. So normally he would eat 3 hours before playing squash. On this particular day, he had a cheese on toast approximately 1 hour before playing the game. Okay, and the second case is 37-year-old gentleman, and he's apprentice in organ building, so he works at a local church. And on that particular day when he was walking from home to the local church, it was cold morning, winter morning, and on arrival he developed widespread itchy rash, and he felt lightheaded and generally very, very unwell. And because he had previously similar reactions, he was already carrying EpiPen, and he self-administered EpiPen and then was taken by ambulance to hospital. And on history, we discovered that he had previously very similar recurrent episodes where he had symptoms of urticaria and anaphylaxis after exercising. And also he commented that stress, temperature changes have previously triggered symptoms. He had no history of any other specific allergies, and he wasn't aware of being allergic to any foods. Again, on the history, we discovered that particular morning he had a fried egg with sausage, and he had bread with butter and jam. Okay, so this is just a summary of several patients we've seen with this condition called weed-dependent exercise-induced anaphylaxis. So all these 5 patients, they had this condition for a very long time. You can see that they had multiple attacks during the year. You normally presents with urticaria, angioedema occasionally can proceed to anaphylaxis. And the food associated with reactions would normally be bread, pancakes, rolls. So it's essentially food which contains high level of wheat flour. And we also had, some of them had multiple admissions to A&E. So the only way to diagnose this condition was to detect specific Ig antibodies to omega-5 gliadin. And the prevention is the key for managing symptoms of wheat-dependent exercise-induced anaphylaxis. We would generally avoid wheat for 4 to 6 hours before or after exercise, and it's very important we discontinue exercise and we stop training at the first sign of symptoms. The second condition I wanted to present today is alpha-gal syndrome, or tick bite meat syndrome. Again, this condition we could only diagnose when we discovered specific Ig antibodies to the component alpha-gal. This particular gentleman, he's a 60-year-old farmer, and he has presented to us with a history of recurrent attacks of widespread itchy urticarial rash. On a number of occasions he developed lip cyanosis and felt very dizzy and unwell, so very likely had anaphylactic reactions. And this has been happening only for the past year or so. And we did spend a lot of time collecting history here, but what we discovered that last year he was bitten by 2 ticks in the field, and the attack started approximately 6 or 8 weeks after he was bitten. He couldn't identify any specific immediate precipitants, and he could eat any type of food with no problems. But then on collecting history, and he kept food and symptom diary, we were able to see that the symptoms only occurred after he consumed red meat. But there was a significant delay of at least 5 to 12 hours. And this particular patient was strongly positive for specific Ig antibodies to alpha-gal. So he currently avoids red meat. These reactions can be very severe. So this is a syndrome associated with significant impact on quality of life. And the only way how we can manage is to advise our patients to avoid red meat. So it will be beef, rabbit, lamb, but also some of the patients who are extremely sensitive to alpha-gal, they can also react to food which was cooked in animal fat, or pharmaceutical products or food containing gelatin. And they will be generally safe to eat poultry, so birds are fine, chicken is fine, eggs, fish, seafood absolutely fine, because other sources of protein, other sources of meat do not contain alpha-gal. So this was a quick run through, and just in summary, just to summarize why component diagnostics are important and where they help us. So they help us to identify the primary and most relevant allergen sources. They also help us to understand patients' risk of systemic or life-threatening reactions on exposure to specific allergens, and help us to design appropriate management plans where we would be in patients with primary sensitization to allergens which would be associated with high risk of anaphylaxis. We would be certainly recommending the use of adrenaline auto-injectors and more stringent avoidance measures. They also help us to understand cross-reactivity and secondary sensitization. And secondary sensitization and cross-reactivity not always associated, in fact, very rarely associated with systemic allergic reactions, more commonly associated with local reactions or no reactions at all. And the last thing, which I haven't been able to cover today, but detecting correct components, detecting primary sensitization to most to the species-specific or allergen-specific allergens, also helps us to select patients for immunotherapy or allergen desensitization. Okay. So this is the end of my talk, and I'm very happy to take any questions.
Thank you very much for a wonderful presentation, Dr. Powell. And taking the privilege of a chair, I think I'll start with the questions. I think it has been known for a long time that people were, those who were allergic to groups, similar groups like plant origin or insect origin or food origin. And your results make a lot of sense in terms of common antigen recognition that may be responsible for this group reactivity. My question is, in case of testing, would you use common antigen? And in case of immunotherapy, would you use common antigen? Because those are most likely to give more hypersensitive reaction or anaphylaxis. Thank you.
Yes, I think for sorry, I couldn't hear. So the first, what was you asked about immunotherapy desensitization treatment, and I missed the first.
No, what I was asking was that people are usually allergic to groups of antigens, distinct, either from insect origin or from plant origin, and probably this is because of common antigen among that group, which is what you are showing as well. My question was that if you give common antigen for immunotherapy, there will be more likely to be more anaphylactic reaction, which is what you were showing as well. So what would you recommend for immunotherapy? Would you recommend distinct antigens for testing versus different antigen for immunotherapy?
No, it is a tricky one. I think, yes, you're absolutely right. I mean, we see patients who are allergic to multiple groups, and there is a lot of cross-reactivity between different groups. If you give patients as a treatment vaccines which contain cross-reactive allergens, they wouldn't necessarily develop more severe reactions. So I don't think there will be high risk of severe reactions. Again, even for immunotherapy, we still use whole extract for a lot of our patients, pollens, animals, or venoms. But I suspect in the future we probably will be moving towards the immunotherapy where we're using more specific components. Because when you start including cross-reactive components, which are usually less relevant, the effect of immunotherapy is likely to be lower. So because we know that the allergy is predominantly driven by sensitization to main allergens, not cross-reactive allergens. So in particular for treatment, for immunotherapy, ideally we would probably want to get rid of cross-reactive allergens and focus on the allergens which are primary and specific to that particular allergen.
Yes, thank you. That makes a lot of sense. Any more questions?
Dr. Powell, excellent presentation by you. Sabiani is over from here. Dr. Powell, in your diagnostic algorithm, you said that if these, for example, for the ARA-H and GALD1, you said that if these allergens, these allergens will be negative, or the specific IG for these allergens will be negative, then we will proceed for the other allergens. So what is the significance of this algorithm? Why aren't we testing all of them at one time?
I think it's important to understand what we're trying to achieve, because, for example, in the example of a peanut allergy, if we suspect peanut allergy, the most important question for us and for our patients is to detect severe form of allergy to peanut, because that is likely to have more significant impact on life and potentially can lead to life-threatening allergic reactions. So these are the patients who will be carrying EpiPen, and they will be advised to very strictly to avoid peanuts or sometimes all nuts. And it's also cost, because if we, in theory, we could do specific IG antibodies to all components, but in patients with allergic sensitivity to main allergens or primary antigens where the risk of anaphylaxis is very high, knowing that we're also allergic to other less relevant cross-reactive allergens is probably not very important. This is why we, in our algorithms, we don't just test all available components. There are even more peanut components now. We probably will be, in the next 2-3 years, we'll probably have 10 or 15 different peanut components we could possibly measure. But I think it's important for us as clinicians to know what is the main aim of our testing, what we're trying to achieve. And in the case of food allergies, the most important thing for us and for our patients is to identify patients at very high risk of anaphylaxis.
Any other questions? If not, we move on to the next talk.
Thank you, Dr. Powell.
Thank you.
Next, Dr. Usman. तो मैं ऊपर दूँ इंट्रोड्यूस. So our next invited talk is from Dr. Munira Naz Baloch. She has been working as an associate professor in the Department of Microbiology, University of Karachi. She has been proficiently teaching various disciplines of immunology, including immunobiology, immunotherapy, and immunogenetics. Her research has been focused on beneficial attributes of lactic acid bacteria, and moreover, she has keenly exploring the probiotic potential of lactic acid bacteria, especially for immunomodulation and also for the plant growth promotion. She has done her PhD from Karachi University, during she studied bioactive agents of lactic acid bacteria. And during her postdoctoral research, she has produced 2 PhD and 2 MPhil under her supervision, along with 10 master's projects to her credit, who have presented their work at many conferences. So I would now invite Dr. Munira to give her talk.
Assalamu alaikum. First of all, thank you so much, Dr. Najia, for the introduction. And firstly, I would like to thank
the Pakistan Association for Pathologists to invite me here with you distinguished guests and speakers over here and renowned scientists to share my part of knowledge here in this on this occasion. I am very proud to be presenting my part of knowledge under the chair of Dr. Rabia Hassan and Dr. Najia. Dr. Rabia Hassan and Dr. Najia both are from our department. They are the alumni from our department, and naturally they are so well known and established in their research. And in front of them, presenting my part of work, I hope that I managed to do well. So here we're talking about the probiotics, and now how we relate these probiotics with their immunomodulatory potential. Normally, whenever we talk about probiotics, we've heard about their being good and beneficial in fermentation or food preservations. So usually the people who are related to the food industries are much more focused in studying about the attributes or the beneficial aspects of the probiotics. So I would just like to put some light on what are these probiotics, because naturally all the sessions which we had since morning did not discuss about these organisms, but I'm sure everybody is familiar that these are basically the live microorganism which when taken in sufficient number will give you a good health benefit, right? And the proposed number of bacteria that is taken in is about, starting from 10 to the power 6 and onwards, we give a sufficient number of microorganisms to benefit from these organisms. Now, how we relate these organisms towards their immunomodulatory potential, and how these being good in enhancing immune responses of our body, they could be beneficially used to influence positively immunotherapies, right? Sorry. Now, it's a very amazing fact that the human gut has probably 10 times more number of prokaryotic cells than eukaryotic cells. And the most predominant phyla which are present in our gut or inhabit the gut include the proteobacteria, fucibacteria, actinobacter bacteria, spirochetes, and the most important which are occupying the gut, 90% of the predominant phyla include Firmicutes and Bacteroides. And amongst these gut microbiome, which we call, those having probiotic potentials include the genus Streptococcus, Bifidobacteria, Enterococcus, Lactobacillus, Lactococcus, and Pediococcus, and many others as well. But these are the most predominant ones. Out of these, Lactobacillus and Bifidobacterium are usually commercially being used as good probiotic products available around the globe. Now, first of all, we'll just have a little view on the beneficial aspects of these probiotics or the attributes. We all know that the probiotics are very good in preventing a number of infections, and they are, they're able to inhibit the number of viruses, parasites, and bacterial species as well. They have been proved very good in, fighting against they have been very good against the severe conditions even in case of COVID. So COVID-2 also has been inhibited by the probiotics. Recent studies have shown that how they block the binding receptors of the COVID-2 in the lungs. And secondly, the probiotics have the ability to adhere to the intestine or the epithelial cells, and this adherence is naturally by means of the pattern recognition receptors. And why they are able to do that, the inhibition of the pathogens, they basically inhibit the pathogens by producing a number of antimicrobial metabolites. And these antimicrobial metabolites could be bacteriocins or could be the bioactive agents, which are the organic acids, and also certain neurochemicals as well. Now, for as far as the immunomodulatory potential is concerned, since they have been very good in strengthening the whole host immune system by activating a number of T cells and the innate immune responses as well. So they are good at activating or enhancing the adaptive as well as the innate immune responses. Further on, how they can compete with the pathogens or eliminate the pathogens from the gut is by competitive exclusion. So they outnumber the pathogens, and since they have the ability to attach to the epithelia, they can easily colonize in the intestine and produce their beneficial effects. So for doing all these beneficial effects, a probiotic must be having number of attributes that it should be able to pass through the digestive tract. It should be able to transit through the digestive tract, having a role to withstand the acidic condition of the stomach, and also withstanding the physiological concentrations of bile. Now, when we know that these probiotics are good in modifying our immune system, there are some strains which be good in stimulating the immune responses, and the others are good at suppressing the immune responses in a number of ways, where they activate the macrophages, the natural killer cells, and produce a number of cytokines which may be classified in those cytokines which are pro-inflammatory and some of them which are anti-inflammatory. And in doing this work, the actual aim and the objective of these strains is to decrease the cancer-specific proteins and also to degrade the pro-carcinogenic enzymes. So they do work on decreasing the pro-carcinogenic enzymes like the glucuronidase or the nitroreductase, which can convert the pro-carcinogens into carcinogens. So that is the very important role that they're playing. They also increase the antioxidant formation, and basically when we aim these for immunotherapies in cancers, so we aim in promoting apoptosis, and our main aim would also be in stopping or decreasing the proliferation of the cancerous cells. So these days, fighting against cancer, the medical practice is usually shifting from the chemotherapies towards the immunotherapies, which are still under trials and clinical trials and preclinical trials, but they are giving good results, and they are augmenting a number of other therapies as well. So since in case of the chemotherapies, we know that they have some life-threatening side effects, and no doubt they are damaging the cancerous cell, but along with that, being non-specific, they are also targeting the normal cells of our body. So shifting towards immunotherapies using the immune checkpoint inhibitors, where we are augmenting or enhancing the immune response, or making the immune cells more active towards the tumor cells or the cancer cells, but the response of the immunotherapy in the patients has been a little lower, and it has been towards like 30% of the patients are responding. Today, according to the research available, that most of the people are not responding very well to the ISIs, and so many reasons could be behind this. It depends from, it varies from person to person, and to facilitate the response or to increase the response of these immune checkpoint inhibitors, we can use these probiotics, because we know that these probiotics are good at enhancing our immune system or making our T cells or the other innate immune cells more stronger against the tumor cells. So we'll see how these probiotics are helping the immunotherapy. Now, these two molecules, the monoclonal antibodies, the anti-PD-1 and the anti-CTLA-4, these have changed the landscape of immunotherapy or therapy against the cancers. And basically, they reactivate the anti-tumor activities of the T cytotoxic cells. So they make the T cytotoxic cell more strong against these by blocking the T cell signaling. So that is the main aim of the immunotherapy. And why we see a negative prognosis in case of immunotherapy is, as I discussed, that many patients are not responding well, and due to many reasons. It could be, one of the reasons could be the antibiotics which are being used that lead to dysbiosis inside the body, and the immunotherapy also has certain adverse effects which include a number of conditions, like it could be increasing the damaging the liver, the kidney, or the lungs, and also maybe leading to cardiac failure and a number of other conditions. So when we use these immune checkpoint inhibitors along with the gut microbiome, and these gut microbiome could be the probiotic species as well as the gut microbiota, which we could use in the form of fecal microbe transplant. It could be a very good approach or a strategy to optimize immunotherapy. So a number of research is being done to optimize immunotherapy by using microbiota or probiotics. We'll see later on. So some probiotics modulatory effects include that they increase the cytokine production or mucin secretion as well, strengthening the intestine, increase the activity of the phagocytosis, and the activation of the natural killer cells. So we know these things. So I'll skip this to make things fast. Stimulation of IgA. And the cytokine production, some of them are the pro-inflammatory cytokines, and the anti-inflammatory cytokines could be the interleukins and the transforming growth factor, the tumor necrotic factors, and interferons. Now, how these probiotics work in augmenting or in facilitating the immunotherapy, or they also work very good in cancers. So these these probiotic bacteria, since we say they are beneficial bacteria, why they are beneficial bacteria? Because they're producing certain antimicrobial compounds and which are good against the pathogens, and they are producing a number of anti-cancerous agents, which are including the metabolites, the CLAs, conjugated linoleic acid, and short-chain fatty acids, amongst which butyric acid is the very most important one that inhibits cell proliferation of the cancerous cells and also induces apoptosis. And besides, we know that they have the immunomodulatory potential, and they can decrease or reduce inflammation while producing anti-inflammatory cytokines, and also they can modify the intestinal microbiota. When we take a pronounced number of probiotics, then they are outnumbering the pathogens, and since they are beneficial, so they would be proving to be good in those conditions. So these short-chain fatty acids, which are basically produced by the fermentation of non-digestible fats, what do? These are the metabolites from the probiotics, right? So what do they really do? They regulate the intestinal barrier function, they strengthen the intestine, they regulate the T cell function, they also induce the IgA production, and they also inhibit the intestinal macrophage proliferation, and basically by the cytokines, which are the interleukin 6 and 8 and all of these. These short-chain fatty acids aim to improve the anti-tumor response of the T cells, and by activating the CD8 cells, and they promote apoptosis, and they suppress the proliferation of the tumor cells. So one of the best strategy and the recent thing to do research on would be to see the synergistic effect of these short-chain fatty acids and the immune checkpoint inhibitors. Where the butyric acid, while it's regulating the inflammatory cytokines, is regulating the activity of the proteins involved in the apoptosis, it's basically, or ultimately, it's enhancing the immunogenicity of the tumor cells. Now here we see the outcome of the metabolites produced by the lactic acid bacteria or by the gut microbiome, and all these are the, I'm trying to use the pointer. but I think it's not working. So I'll just proceed in a sequence. So the microorganism belonging to the bifidobacterium, which is a probiotic, and Mucinifera, which is a gut microbiome, they produce inosine, and this inosine is naturally bringing down signals across the lamina propria, where we have diffused or organized lymphoid tissues, and the cells are present there, and these cells are naturally getting activated and producing their beneficial effect or immunomodulatory effect. Then we have short chain fatty acid produced by probiotics as well as the gut microbiota, and they are also transducing signals that are activating the CD8 cells and increasing the anti-tumor activity, and that is the increasing apoptosis. Then we have anachoric acid, then we which is also doing the same purpose, that is activating natural killer cell and increasing apoptosis. The two metabolites, bile acid and the tryptophan, which are produced, they're basically attenuating the tumor anti-tumor effect. So these may be produced by the other bacteria, especially the Clostridium. They might not be helping in the augmenting or modulating the immune system, but they might be reducing the activity. And on the other hand, we have the peptidoglycan, the PSA, which is the polysaccharide, and the outer membrane vesicles, which are also activating the immune cells. The inosine, which is produced by the probiotics and the gut microbiome in the intestinal lumen, they are basically helping the tumor antigens to be presented to the T cells and to increase the ISI efficacy. Further on, the inosine is also activating the interferon gamma and interleukin 12, which is activating the T cells to give a pronounced effect. And inosine is also giving alternate carbon source for these. So these are basically the ways in which the metabolites from the gut microbiota are giving their good effect or enhancing the immune cells. And you can see on that side, immunostimulatory effect has been seen in the tumor microenvironment when we give inosine with ISIs, and alone they are giving an immunosuppressive effect. So basically, how these two interact, how the probiotics or the microbes which we are using beneficially interact with their microbe association molecular patterns, MAMs. They interact with the toll-like receptors. We have heard about the toll-like receptors, so I'll skip, running short of time. And here we see the interaction of the pathogens, the suppression of the pathogenic bacteria by the probiotics on one side, because they're producing their short-chain fatty acids, and underlying the mucosa, we see a number of activities going on, and these activities are basically for their immune responses. So we have they will activate the T regulatory cells. They will also activate the other cells which are releasing the pro-inflammatory cytokines. And at the bottom, you can see the interaction of the toll-like receptors with the probiotics, and they are regulating the tumor necrotic factors as well. So we need to focus our attention more on activating the PRRs rather than seeing the inhibitory potentials of the probiotics or the gut microbiome. And this is to evaluate the PRR activation profile. We screen for all the TLRs, and we also see that whether they have a co-stimulatory effect. So we need to see the response of the molecular patterns here to the TLRs, and also how they're dependent on the myeloid differentiation factors or proteins in their signaling. So various approaches could be done, and we've seen that the gut microbiome vary, or they are different in their response, in the interaction with different TLRs. So we need to see that which probiotic is interacting with which class of toll-like receptors. We also need to see the MYD88 dependency, and next we need to determine use the cell lines. We can use the cell lines. We can inhibit the MYD88 proteins and see whether the toll-like receptors are dependent on these or not. We can finally be having insight in the specific TLR signaling of the lab, and we can predict for which application we may use these probiotics. So the here I'll come to this one. The labs proceed in both the pathways to increase apoptosis, the intrinsic pathway as well as the extrinsic pathway. And in the intrinsic pathway, they are activating those gene products which are actually activating the caspase and leading towards apoptosis, whereas in the extrinsic pathway, they are activating through the Fas ligand, inducing the tumor necrotic factor receptor, and finally activating the caspase for the apoptosis. And I'll skip this, running short of time. Okay, so the therapeutic strategies, basically we need to set some strategies to use the microbiota-based therapeutic strategies in which we can use fecal microbiota, we can use probiotics along with the ISIs to augment this. Now these are the approach which we can use. We can combine, we can use a combination of therapies, we can use probiotics as discussed before earlier. We can use fecal microbiota transplants, and these are the gut microbiota, and usually we use this from the ISI responding patients. And we can use engineered microbiome according to our preference, that we can do the gene editing for a specific molecule that would target the immunotherapy. So it is also important to understand the factors influencing the gut microbiome and explore the therapeutic responses. So this is one of the studies where they have used the fecal microbiota, and they have used the probiotic bacteria along with some prebiotics.
I would like you to wrap up.
Okay.
Last minute.
I'm sorry. So they would be, using prebiotics which are increasing or enhancing the growth of this, and it has been seen that using the anti-CLA and the anti-PD antibodies for the immunotherapy, these probiotics have been used beneficially to enhance the activity of the immune cells and give enhanced effect of the in the tumor microenvironment. Okay, so this one I would like to share this for the last thing, that this is the timeline from the discovery to the application of the gut microbiota, starting from 2007 and 13. First time it was known that the gut microbiota is stimulating the anti-tumor activity, and further on in 2015, some preclinical trials were done, and these preclinical trials showed that using Bifidobacterium and Bacteroides vaginalis, these two microbes have basically increased the immunostimulatory potential and facilitates immunotherapy or the ISI efficacy. In 2018, these were used in humans, and the humans we used from the non-responders and from the responders. And
stop you now. Please, last slide.
Okay, last slide. And we can see that when we're using the fecal microbiota from the responders, we are getting a good immunostimulatory effect. Whereas when we use, and we've also seen that the antibiotic-induced dysbiosis does not get a good effect on the immunotherapy. So our aim would be to use fecal microbiota plus or the probiotics to see our immunostimulatory effect in the tumor microenvironment. So that is the recent research which is being done, and there are these companies which are working on the third. They are on the third phase of the trials and using these fecal microbiota or gut microbiota, and still it is not approved by the FDA. These some probiotic products which are available, so we need to focus more on the probiotics or this thing. Okay, I'll stop here. Sorry, I've run out of time.
You run over time. Yes. Thank you. Thank you very much for this interesting review of literature, but I would like to know what experiments have you done to look at any of this? Which arm are you looking at? Are there any results that we can see?
Okay, these research which I've just shared are the recent researches, and what we are doing in our lab, basically I have started my research work in working about the bioactive agents of these probiotics.
You don't have any results to show?
No, not right now, of course.
Fine. Okay, and I just want to add, because I'm in this business of microbiome and FMT, so FMT is not as simple as you are suggesting. So there are various ethical concerns, DRAP issues, DRAP approval, We are still working with OpenBiome, a company which provides FMT throughout the world. So just to give you that it's not that easy to get FMT here in Pakistan.
No, it's not easy to get FMT in Pakistan, of course. I was just telling you that these are still on trials, on clinical trials, and
it is used for C difficile. It has been successfully used for C difficile.
It is approved for the C difficile by the FDA, yes.
So there's a question from Dr. Powers, actually, he must be listening to you. And he asked that if there are any risks associated with using probiotics or fecal transplants in patients with impaired immune system due to cancer immunotherapies?
Usually these probiotics are not associated with any pathogenicity. And if you talk about the cancer or the conditions under cancer, how we use these probiotics, since some of the strains are leading to the pro-inflammatory responses. So if we require to modify our immune system in producing pro-inflammatory responses, then naturally I don't think probiotics would be giving some unusual or not bad effect against the cancer patients if we use the probiotics. Thank you.
Okay. Thank you, Dr. Munira. So now we have free oral presentation. We had in our plan for Dr. Usman Yusuf, but because of shortage of time, we have to skip this. And now I would like to invite Dr. Kiran Iqbal from Aga Khan University to give an oral talk on dynamics of IgG antibody response to SARS-CoV-2, reveal insights into immunity during the early pandemics period in Pakistan. Dr. Kiran, you have 5 to 7 minutes to wrap up. Thank you.
Dr. Najma. All right, thank you for giving me this opportunity to present the research work of our group on this platform. I have nothing to disclose. All right, so pandemic hit the world by around 2019. And since then, it has claimed roughly around millions of lives all around the world. And during early phase of pandemic, many countries reported high case fatality rates, among which Italy reported 6.2%, Iran reported 3.6%, whereas Pakistan, fortunately at that time, did not go beyond 2%. So at that point, we asked this question that what are the mechanisms that are responsible for protective immunity in our population? So what we did was that we recruited study subjects, and those were into 3 groups. So we recruited healthy controls. These were the volunteers from the hospital, from Aga Khan Hospital, as well as some attendants who were coming with COVID patients, so they were exposed with COVID. We had COVID-19 patients, and we had some pre-pandemic controls. The samples were collected before the pandemic started. And what we did was that we looked at the B-cell markers and the T-cell markers. So to study the humoral immune responses, we looked at IgG responses to spike and RBD, and we also looked at interferon gamma responses through ELISpot to look at the T-cell responses against spike. So this is the first set of results, and in this slide, we are showing the levels of IgG responses to spike and RBD in COVID patients as compared to healthy controls, and we see that the levels were significantly raised for both IgG to spike and RBD in COVID patients as compared to healthy controls. We also did the correlation analysis between IgG to spike and RBD in both the groups, healthy controls and COVID, and we saw a strong positive correlation in healthy controls, whereas in COVID patients it wasn't that strong, which may indicate that the receptors that are recognized in the cases may be similar or distinct. So then we looked at the dynamics of the IgG responses during the pandemic period, and these are healthy controls, and we could see the raise in the IgG responses during the period when we saw the peak in the wave, that is between December to January, and then the seropositivity came a little bit down, and then it increased again with the increase in the peak in the 3rd wave. And that's what we saw for RBD as well. And in figure number C, the same is denoted by the percentages. Then we looked at the temporal association of IgG to SARS-CoV-2 antigens in COVID cases as well. And we did see the IgG to spike responses that were maintained over the period. The responses of IgG to RBD were comparatively a bit lower, but they were sustained. Similar is shown in terms of percentages in figure number C. Then having seen that, we also looked at IgG antibodies to spike and RBD in pre-pandemic controls, and we did see few responders in there. In pre-pandemic controls, there were few responders to spike as well as RBD, whereas the correlation was not very strong. This could be due to the cross-reactivity due to circulating coronaviruses, the seasonal coronaviruses, or Pakistan has other disease states. The EPI is also different from other countries, so it could be due to multiple reasons, the cross-reactivity. Then having seen the humoral immune responses, we further went on to see T-cell responses, and we did this by measuring interferon gamma responses through ELISpot assay. And we looked at COVID-19, healthy controls, and pre-pandemic controls. So in COVID-19, we did see 6 responders, whereas 12 did not respond. This could be due to the reason that the samples were collected pretty early, like 48 hours after the diagnosis, and this is the time point where we do not expect much of the T-cell activation. In healthy controls, we had 2 responders, and in pre-pandemic control, we had 1 responder. So we wanted to see if these T-cell responses correlate with the humoral immune responses. So we further looked at these responders, and we saw that the pre-pandemic control, which was positive for the T-cell, was not positive for B-cell. So basically, IgG antibodies to spike proteins were independent of T-cells in this particular group. So just to sum up the findings of the entire study, the higher IgG levels in COVID-19 cases may reflect the antibodies that were present were specific to viral infection, and they could have evolved from the reactivity which was already present there. Increasing antibody frequency and magnitude to spike and RBD in healthy controls could be due to SARS-CoV-2 exposure. It could be cross-reactive antibodies. Spike-reactive T-cells in pre-pandemic controls spike-reactive T-cells in pre-pandemic controls support the role of cellular immunity, which could be introduced again by cross-reactive antigens, and it is likely that humoral and cellular immunity driven by cross-reactivity due to pre-pandemic exposures may enhance protection against SARS-CoV-2. Thank you. Acknowledgements.
Excellent for wrapping up in time. Thank you. So we have some time for quick questions. So one question that I have is that we don't have any consensus on ELISpot cutoff. So were you able to determine any cutoff or positivity in ELISpot against COVID-19?
Yes, we did. Yes, we did. So there were 6 individuals who actually responded, and they were positive for T-cell responses.
So usually,
in COVID-19 patients,
yes. T-cell ELISpot cutoff is more than 20 CFU.
Yes.
SFU.
Yes.
So
yes,
is it the same?
Yes, yes, it is the same. It is the same. As per the kit, as per the manufacturer.
Okay.
NAVTAC kit was used, yeah.
Okay, thank you. Any other from audience?
Yeah.
Sorry, I missed the earlier part of the presentation, but I have one question. I mean, you said that because of the cross-reactivity, we could have all this. But then have you checked in the patients with breakthrough infections that what is actually happening out there? If they are present outrightly at the beginning, these cross-reactive T cells, if they are protecting our population from the breakthrough infections or not?
So basically, this study was carried out during the initial phase of the pandemic, and the samples were collected from October 2020 to January 2022. At that point, these individuals were not even vaccinated. And as per history, they were not even vaccinated, and as per history, they got the infection. First time it was only 48 hours of diagnosis. So this can be done as a future part, and also we can further, for cross-reactivity, we can further look for antigens for other coronaviruses as it's been done in other studies. It's not done in this part of the study.
Something to your comment. Actually, I have also been part of this study. The thing is that this study, the major objective was to see during the pandemic, how does exposure result or in evolution of immune responses. That's all that this study was aimed at. So the samples weren't collected from the point of view of looking at and understanding breakthrough responses, which is going to be part of the next study, actually.
Yeah, thank you so much.
Thank you, Dr. Kiran. Now we have our second oral presentation, Mariam Ashfaq from Indus Hospitals and Health Network. The title of her talk is The Significance of Humoral Immune Response in COVID Breakthrough Infections in a Local Population. So very nicely followed by the question of Dr. Sabiha. You will get some answer from your talk.
Assalamu alaikum everyone. I am Mariam Ashfaq Khan from the Indus Hospital. Today I am here to present our study entitled The Significance of Humoral Immune Response in Breakthrough Infection in Local Population. There is no financial and conflict of interest. What is a breakthrough infection? As per definition of CDC, a person with a breakthrough infection is someone who has a positive COVID-19 test 14 or more days after receiving a full course of COVID-19 vaccine. Although COVID-19 vaccine drive significantly reduced the COVID-19 cases, still vaccinated individuals got infection. Breakthrough infections develop irrespective of the type of the vaccines administered. Why these infections are caused? Because no vaccine is 100% effective. Secondly, fading vaccine-induced antibody levels. Moreover, emergence of new variants and their subsequent escape from the immune surveillance. Objectives to study the dynamics of anti-COVID antibody levels and its significance in breakthrough infection. This is the methodology. We divided our vaccinated participants into two groups. Control groups were the participants who did not develop any infection, and the breakthrough infected group were the participants who developed infection after the full course of vaccination. From this group, we collected 2 samples of blood, one during the first week of the infection and second after the first month of the infection. And from the control group, we collected the sample within 4 to 8 weeks of the vaccination. ELISA was performed for anti-COVID IgA and IgG, followed by statistical analysis. These are the demographics of our study. Mostly participants received Sinopharm. 70.5% of our participants did not have any history of COVID infection, while 29.5% of participants have previously infected with the COVID-19. 88% of the participants had mild infection, while 6% have asymptomatic and 6% have moderate infection. Comparison of anti-COVID S-IgA and anti-COVID S-IgG positivity rates between control and breakthrough infected group. As you can see that breakthrough infected group, both IgG and IgA levels were higher as compared to the control group. For IgA, we found significant difference between control and breakthrough infected group. Comparison of anti-COVID NP-IgG and anti-COVID NP-IgA positivity rates between control and breakthrough infected group. For NP-IgG, we found a similar pattern, that is, breakthrough infected patients have higher positivity rate as compared to the control group, while for NP-IgA, we did not have any positive sample in control group, and only 3 samples were positive in breakthrough infected group. Sorry? Okay. 34. Breakthrough infected participants were 34 and control 29. I have mentioned in the methodology. Comparison of anti-COVID S-IgA and anti-COVID IgG rates at 2 time points in breakthrough infected groups. When we see the effect of the time or after time, we see that we found that for S-IgG, all the samples were positive, while for S-IgA, although the number of positive samples increased, but still there were some samples that were negative. Comparison of anti-COVID NPIgA and anti-COVID NPIgG rate at 2 time points in breakthrough infected groups. For NPIgG, positivity rate increased, while for IgA, the number of positive samples decreased significantly. Comparison of the titers between the breakthrough infected groups and the control group. We have the significant difference between SIgG control and breakthrough infected group, similarly for NPIgG as well. While for NPIgA there was no significant difference, and SIgA we have no significant difference. Comparison of COVID IgA and IgG titers between time points. Basically there is a mistake in heading. We found no significant difference in the titers because for IgG, because they had higher titers at initial phase as well. What we concluded from this study, our findings are consistent with the previous study that breakthrough infected individuals have higher antibody level as compared to controls. They have higher antibody levels due to vaccine exposure and infection. Lower IgG levels in infected individual was the reason for breakthrough infection. Increasing level of IgG will provide a long-term immunity. Heightened antibody levels in breakthrough infection prevented severe illness. Limitations, we had a small number of participants. We are thankful for Mr. Mohammed Skaini of ASKU group and Mr. Masood Ahmad for providing us ELISA kits. Our team, thank you.
Thank you for finishing on time, Mariam. So nice presentation. So any question from audience?
There's only one audience that's talking. I have one question, and that is more what you think about COVID infection and breakthrough infection. Do you think this is a reactivation? Or do you think this is due to re-exposure?
Re-exposure.
Breakthrough infection, of course. Why do you call it breakthrough? Can you evaluate if it was a breakthrough infection because of exposure, fresh exposure, or because it was reactivation?
I think fresh exposure.
Yeah, most more. Is there any report on latency of COVID-19?
That's what I'm asking you.
No, I don't. No. Viremia goes off. Yeah. So.
And another question is, did you do nasal IgA when you say serum IgA?
Serum.
Okay.
You didn't do nasal IgA. Because there is a difference of 100-fold. So if you want to compare IgA to IgG, IgA gets degraded in the serum. That is the limitation. That's the limitation. So you can't do comparison with the DNA. Yeah, but you just look at the lab to see if you can predict or can find the product. And what about long COVID? We haven't done any study on that.
We are starting. We are starting.
I don't know if it is breakthrough or if it is.
So
that's the reason I was asking if there is latent infection.
No. We will starting. We will start soon. Yeah.
In long COVID, why long COVID? Are they harboring a latent infection?
No. This is all about the collateral damage that already done during the acute phase.
And we have seen this kind of syndrome like the chronic kidney syndrome and other viral infections. So you think that is an autoimmune component?
Could be, because there has been reports about the autoantibodies against. Yeah.
This is getting out of hand.
Okay, thank you.
Do we have any more?
Yeah. So we have next speaker, Medhat Batul. Her title from Dow University Health Sciences, Medhat Batul Zaidi, sorry. And the title is Investigation of the Wound Healing Potential of Photoactive Curcumin Zinc Oxide Non-Conjugates in an Excisional Wound Model. So I'm reading title to save your time.
Thank you. Assalamu alaikum and greetings everyone. I'm Midhat Batul Zaidi. I have done my PhD in molecular medicine and currently serving UHS. So what we are presenting here is we investigated the photoactivated curcumin zinc oxide nanoconjugates effect on wound models. Contents. Wound healing is a complex process dependent upon 4 phases in which various growth factors and cells play a significant role in a complex manner. However, the available treatment and therapies fail to completely regain the skin integrity and functionality in chronic and complex acute wounds. So, traditionally, polyphenolic flavonoids have been used in herbal and Ayurvedic medicine due to their strong antioxidant, anti-inflammatory, and anti-infective properties, such as curcumin that reduces the inflammation, increases the proliferation, and support the remodeling while healing. However, its therapeutic efficacy is restricted due to its poor availability in a biological environment. So in advanced technology, in advanced formulations, metallic nanoparticles has been used that increase the availability of bioactive compound, and they also have antibacterial, and they are also reported to have antibacterial, anti-cancer, and wound healing properties. So in this study, we use zinc oxide activated with a specific wavelength of phototherapy, and these zinc oxide were loaded with curcumin bioactive compounds. And this is the chart showing advancement in wound treatment, and in 2027 4.5% CARG is expected to have an increase. Objectives of our study: we synthesized the photoactive curcumin zinc oxide nanoparticle, characterized it, and studied its antibacterial and preclinical properties in wound healing. Our methodology comprised of synthesis, characterization of the nanoparticle, and then we perform its preclinical studies to study its antibacterial and healing efficacy. Our results and discussions. Results indicated that these particles have high antibacterial effect as they reduce the biofilm formation by S aureus and S biogens. Later, the preclinical studies on the wound model, excision wound model, showed they proficiently healed the wound when applied on an excised skin. And there was a clear difference in the illuminated nanoparticles and non-illuminated particles. Later, the H&E staining supported our findings, and pink granulation tissue formation was seen at day 16, corresponding to the remodeling phase of wound healing. However, in non-illuminated group, scar tissue formation was observed. And the immunohistochemical results also showed that is not quite clear here, but formation of blood vessels, and that was studied by using CD31 marker. Later, we also studied the inflammatory markers by gene expression analysis, and the reduction in inflammatory marker IL-1 beta, TNF alpha, and MMP9 was noted in the group treated with curcumin zinc oxide nanoparticles illuminated with a specific wavelength. We also studied the remodeling properties of this agent, and the hexauronic hydroxychloroquine and hydroxyamine was shown to be increased in these treated groups. So here I conclude that the nanoconjugate formulation coupled with visible light illuminated with a specific wavelength can increase the healing potential of the non-healing wound by reepithelialization, angiogenesis, and collagen deposition at the site of wound. So I acknowledge the dedicated contribution of our authors in the completion and compilation of this study. Moreover, we are now working on the clinical translation of this study, and currently one of our group is working on the development of a MedPen light therapy for the activation and the packaging of this formulation in the form that we can provide it to the market. And that group is led by our material chemist Dr. Salma Mirza under the supervision of Dr. Talak Rumi. These are our references. Thank you for your attention.
Thank you for excellent talk and being on time. So, yeah. Any question from audience? I see a lot of people are just end of the day you expect, but
actually I have a comment. When I was 5 years old, my mother used to put haldi on my cuts. It's nice to know what the mechanism behind is, but the role of haldi in wound healing is centuries old.
Yes. And
but if you can improve that formulation,
yes,
and know the active ingredient,
yes,
and look at what's happening, that would be very, very good. It's a very useful study actually, but it has to go further.
Yes, indeed. Thank you.
Thank you. Nidat. And now we have another speaker, Mr. Ali Raza from SIUT, and the talk is title is CYP3A5 gene polymorphism and its impact on tacrolimus dosage among kidney transplant patients, a systematic review and meta-analysis.
Assalamu alaikum. Thank you, Pakistan Society of Immunology, for giving me an opportunity to present our work lab. Today I will talk about one of the important pharmacogenomic variants, CYP3A5 gene, and its role in pharmacokinetics. Tacrolimus is a very potent immunosuppressive drug. However, it has a very narrow therapeutic index. Mean to say, even a small change in the drug concentrations or blood concentration result in the very damaging effect that could result in the nephrotoxicity or graft rejection. Cytochrome P450-3A5 family is reported to be a very major contributing factor in the tacrolimus pharmacokinetics. Single nucleotide change in the splice variant in the intronic 3 region of this functional in the gene result in the alteration of the functional of this protein, which actually affects the tacrolimus concentration in the blood. Patient with the homozygous GG allele of CYP3A5, also known as the non-expressor, actually it is the poor, or also known as a poor drug metabolizer. However, the another CYP3A5 expressor are the rapid drug metabolizers, and they need the high tacrolimus drug to maintain their trough levels. So with this background, we systematically reviewed and analyzed to explore the effect of CYP3A5 expressor and non-expressor on tacrolimus trough level to drug ratios, and also the risk of allograft rejection among the Asian and the European populations. So this is our systematic and review method. We use the medical subject headings with different electronic databases. And this is our inclusion criteria. Primarily, we focus the studies having a CYP3A5 genotyping with the pharmacokinetics, that is the tacrolimus trough levels to the drug ratios in the expressor and non-expressor, and also the incidence of rejections amongst the expressors and the non-expressors group. So this is our exclusion criteria, in which we actually didn't include the combined transplantation. We only include the renal transplant patients, and we didn't include any studies that have insufficient data. So this is our result. In total, we get the total of 52 studies. 27 studies containing the tacrolimus expressors levels between the expressor level, while the 25 studies reported or investigated the effect of CYP3A5 effects on rejection episodes. We subsequently classified these studies into the Asian and in the Europeans. So these are the results. The included studies contain the European population shows the expressor percentage of A allele is 14 to 24%. However, the Asian population shows the frequency of 43 to 46%. This indicates the European population has a low frequency of functional CYP3A5 expressors allele as compared to the Asian populations. For meta-analysis, we use a software review manager software to actually analyze the tacrolimus trough levels between the expressors and the non-expressors. And we also actually find out the effect of CYP3A5 expressor and non-expressors effect on the rejection episode. The overall effects of the study is actually calculated by the z-scores, and the z-scores greater than 1.96 or p-value less than 0.05 is considered as the significant. So this slide shows the Asian populations at week 1, in which you can see the expressor groups in the top and the non-expressor group. Tacrolimus levels were compared, which is calculated for the standard mean difference between the two groups, and the statistical p-value was indicated. Z-score indicated the significant value. This can also be seen by the forest plot, which shows that the expressor group have a cumulative tacrolimus low level as compared to the non-expressors. Interestingly, the week two of the studies among the Asian population had the same actually finding, that the expressor groups have the low level of the tacrolimus level. We further look at the different time intervals at 3 weeks, 3 months, 6 months, and even the 1 year. They all show that the same pattern, the low level of the tacrolimus level compared to in the expressor groups. And even further interesting point is this: even the European populations show the same pattern, that all expressor group have a low level of tacrolimus level compared to the non-expressor groups. So result, the tacrolimus levels ratios was significantly found to be lower in CYP3A5 expressor group as compared to the non-expressor group in Asian as well as the European. So with this finding that expressor groups have a low level of tacrolimus, could this be result in the risk of the rejection? So what we did actually, we actually collected the studies that shows the risk of renal allograft between the expressor and non-expressor. We didn't find any kind of association with the risk of rejection with CYP3A5 genes. However, interestingly, when we stratified this data into the Asians and the European population, so we find the significant association of rejection with the Asian population. Here you can see the Z value is significant. However, the European population didn't show any association with it. I am finishing. I understand you all are actually tired from listening to so many things. Yes, I am finishing. So interesting thing, what I am just to summarize, the frequency European is lower compared to the Asians population. Number one thing. Number two, the tacrolimus trough levels was significantly lower even in the European and the Asian population among the expressor group. The interesting point, the Asian population may have at more risk due to the prevalence of this high percentage of expressor group. Just to add, in Caucasian CYP3A5 explains only the 42 to 50% of the variability of tacrolimus. It does not actually explain the total variability of tacrolimus among the population. So it's better we need to evaluate the Asian population for the SIP and check the variability among our populations. So just to conclude, Pakistani population need to be explored for the prevalence of variants in drug metabolizing enzymes and drug transporters, which could have a putative clinical consequences on drug efficacy, safety, to have a precise medicine. So this work is actually the combined efforts of our lab, and this work has been published, and so far within the 2 years it has more than 30 citations so far. So thank you. With this, I thank you all of you for your for listening this.
Thank you, Ali Raza. Do we have time for a question, or shall we move to the next?
I would like to make a comment. Ali Raza, an excellent study and very meaningful. So I'd like to congratulate you on this study.
Thank you.
Ji, short question. Yeah. This is our actually the pilot. This is actually the data collected for the putting in pilot project. Recently, we have actually designed a project in order to check this thing in our population. So far, what we are actually thinking of taking the liver transplant patient, mainly because the tacrolimus is the main actually immunosuppressant in the liver transplantation. So we actually making a project, a pilot project, just to see the effect in our population. Based on this knowledge, we population are high in prevalence of this SNPs or variants.
Liver transplant, there is minimal need of immunosuppression. What about renal transplant? Our center uses tacrolimus in routine. I think SIUT doesn't, but there the impression is very strong that lower doses of tacrolimus would maintain high levels, comparatively high levels, serum levels. So that there's that bit of information for thousands of patients. So that has to be looked into.
Yes, we will discuss this with you so that you can guide us better. We come to know there are certain patients that actually even giving the double dose of the tacrolimus, they are even unable to maintain the specific levels.
Those are exceptions.
Those are
very
exceptions.
Few.
So particularly these cases recently come to our knowledge by the liver transplant team. They are asking for this test to be performed at our lab, so that we can do this and we can actually offer this.
Thank you.
Thank you, Ali Raza. Last talk is from Asma Khawer from Dow University of Health Sciences. Her title is Folate Conjugated Simvastatin Nanoformulation Modulates OPN Expression in Atherosclerosis.
Which is titled as Folate Conjugated Simvastatin Silver Nanoformulation Modulate Osteopontin Expression in Atherosclerosis. Atherosclerosis is a chronic inflammatory disease of vessels. It usually appears at the age of 50 to 60 in the form of coronary artery disease, stroke, and peripheral vascular disease. In the year 2020, it is reported that globally 27.9% of all deaths were due to cardiovascular disease caused by atherosclerosis. Pakistan is the 6th most popular country, and WHO has reported that the mortality attributed to non-communicable disease is approximately 50%, from which 19.6% is due to cardiovascular disease, primarily due to atherosclerosis. Fibroblasts present in adventitia is reported to play an important role in vascular remodeling and several diseases such as atherosclerosis. Osteopontin, among many mediators of atherosclerosis, osteopontin is one of the main mediators. It acts as a pro-inflammatory cytokine, and induction of oxidative stress is probably the main mechanism to promote atherosclerosis. Osteopontin causes oxidative stress, which activates, proliferate, and differentiate fibroblasts, and then it migrated to intima, where it changed the kinetics of other cells, and in this way it contributes in the pathological process of atherosclerosis. Simvastatin is the drug of statin family, and it is reported that it downregulates osteopontin expression in plasma, but clinically the drug shows some limitations, like it undergoes first-pass metabolism, poor biodistribution to the site of the action, and rapid elimination from the circulation through hepatic clearance and renal clearance. So there is a need of a smart drug delivery system to enhance the efficacy of the drug. So nanoparticle act as a targeted drug delivery system to enhance the effectiveness of drug in inflammatory disease. Folic acid is used for further enhancement of targeted delivery of joint therapeutic drug in the inflammatory disease through folic acid receptors. It is hypothesized that simvastatin and simvastatin nanoformulation decrease opin expression and production in fibroblast cells, due to which opin-induced ROS production would be decreased, and in this way it decreases the atherosclerosis. Our research objectives are to explore whether opin expression and production decreases in adventitial fibroblast cells with simvastatin treatment and its nanoformulation, to check whether reduction of simvastatin dose and its toxicity while using its nanoformulation, to check whether ROS production induced by opin will be decreased with simvastatin and its nanoformulation. These are the treatment groups. The fibroblast cells were grown in 96 well plate and treated in these following groups. Adventitial fibroblast cells were isolated from the thoracic aorta of star rat. Then the cells were cultured in DMEM media with 10% FBS serum until 70 to 80% coagulancy was reached. In our methodology, first fibroblast cells were treated with simvastatin, folate conjugated silver nanoformulation of simvastatin or spironolactone for 1 hour, and then it exposed to aldosterone for 24 hours, and then opin in fibroblast cells was detected, protein expression of opin by ELISA and mRNA expression by quantitative PCR, measurement of ROS by spectrophotometer. Viability and treated fibroblast cells were assessed through MTT assay. Data was analyzed by SPSS version 19. Statistical difference between intergroups were evaluated by using one-way analysis of variance. P-value less than 0.05 is considered statistically significant. MTT assay results showed that the aldosterone and spironolactone both significantly decrease the viability of adventitial fibroblast cells, whereas simvastatin, especially simvastatin nanoformulation, showed significant dose-dependent increase in the viability of adventitial fibroblast cells. Gene expressions showed that simvastatin and simvastatin nanoformulation both showed significant decrease in opin mRNA expression in fibroblast cells. Protein expression showed that the treatment did not cause significant effect on opin protein expression. Measurement of ROS by spectrophotometer results showed that none of the treatment caused significant alteration in ROS level as compared to the control. So from these results we concluded that Simvastatin, especially its nano formulation, demonstrated protective effect in cellular model of atherosclerosis. It decreased OPN expression through its better pharmacokinetic profile. These are the references. I especially thankful to my supervisors, Dr. Muhammad Kashif, Dr. Talat Rumi, and Dr. Uzma Zaman for their continuous help and support in completing my study. Thank you so much.
Thank you, Asma Khabar, for completing on time. And we have some questions from audience. If not, then we can conclude this session.
On behalf of Pakistan Society for Immunology, I am extremely thankful to our distinguished chairs who actively participated to make the session successful. I'm also thankful to the invited speaker and the free paper presenters who actively participated in the late hours. Thank you, all of you. Now may I request Professor Tahir Aziz to distribute shields to our distinguished chairs, Professor Rabia Hussain and...
When I need to conclude the session, summarize the session as well. I know.
Okay,
yeah.
I'm so sorry.
Yeah. Okay. Thank you. It's a very daunting task for me to conclude, but I will try to conclude. So, Dr. Powell, I don't know if he's still online. Okay. Thank you, Dr. Powell, for a very nice talk. And basically, he gave an overview of a specific allergen and cross-reactive allergen, and basically gave us an algorithm for this panel, allergic panel, and how effectively this panel can be used for the identification of most relevant allergen for life-threatening conditions, and also identify the correct component for diagnostic purpose. The second talk was from Dr. Munira Naz Baloch, and she gave an basically discussed on the potential of probiotics on immunotherapy. She presented a review of probiotics and immunotherapy and certain modalities where we can use probiotics in cancer. Then we have a session for oral talks, and first off from Kiran Iqbal from basically she discussed a very important topic of dynamics of IgG antibody during SARS-CoV, and the important thing that she also discussed, which we heard in the first session, about the pre-pandemic cross-reactivity of SARS-CoV antibody. So it shows that there are some cross-reactivity in pre-pandemic samples. If laboratories have those pre-pandemic samples, they can check, and I think we should have stronger data to show that there was some circulation of coronavirus family viruses, and therefore we have some kind of cross-reactivity. Then we had this talk from Mariam Ashfaq, and she discussed on humoral immune response in COVID-19 breakthrough infection, and basically she discussed or compared the level of IgG and IgA in the breakthrough infection. And then we have a very nice talk from Midhat Batul Zaidi on the role of curcumin zinc oxide, and she basically gave an overview of how nanoparticles and other modalities can affect the wound healing of this. And then there was a very nice talk about of meta-analysis by Dr. Ali Raza, who basically showed the level of tacrolimus, which is used in as a drug for transplant, and he basically compared this with the level of CYP polymorphism of CYP3A5. And then lastly, we have a talk by Asma Khabar, who discussed the role of simvastatin on the production of osteoponin, and basically she suggested a role of nano formulation that basically enhance the potency of simvastatin in atherosclerosis. So I think it was a very nice session. And thank you all speakers for their talks, and invited speaker, and also the oral speakers.
I would also like to thank the audience to be in this graveyard session. And I think we have to laud their persistence in listening to the end. So thank you very much, and thank you for all the speakers who have done a marvelous job.
Thank you, Dr. Powell, especially for being online and attend and listen to all talks. Thank you so much.
So now, extremely thankful to the distinguished chairs who made the session successful and invited speakers and presenters. May I request Professor Tahir Aziz to please give shields to our distinguished chairs. Professor Rabia Hussain. Dr. Sabia. Dr. Najeeha Iqbal, please. Thank you. Next, Dr. Munira Naz. Last is myself. Sorry. Thank you, all of you. On behalf of Pakistan Society for Immunology, I thank you, all of you, for being with us throughout the session. Thank you so very much.